happy that you're here, and please get started.
Thank you. Can you hear me?
Yeah, we can hear you. You're good.
Good. All right. So let me share my screen. Start my presentation. All right. So it's a pleasure to be here. Let's see, use the laser pointer. So I'm going to talk about interpreting interpretable machine learning for quantum emergence. And I'm going to start by defining what quantum emergence is. I don't quite know um, who my audience is, and it's kind of unusual that I cannot just look at people's eyes and try to get a sense of like, am I making, getting through or not? I'm going to try to be accessible. I'm going to try to make my subject accessible. So I can run into the danger of over preaching to many different types of experts. And so you just have to bear with me because I expect the audience to be broad, with a broad, diverse background. Now, interpretable, I think, is really key for successful application of machine learning to the subject of quantum emergence. But let me start by defining what emergence is. So this is a collective phenomena of people making a wave, and this is something that you can observe by eye. But now if you go to the hands of one of these people, there is a different scale, very different scale, again, involving enormous many number of individual degrees of freedom behaving as a collection in macroscopically observable way happening. And that's inside somebody's cell phone here. Somebody's holding a cell phone to take a picture of the players. And inside the cell phone are materials, and inside the material, there are these electrons living in this environment that they're in. And like the people in the crowd, these electrons are exhibiting something that you can only observe when there is a whole crowd of them. Only that the crowd, when it comes to the number of electrons is so much larger, even inside this palm of this one person compared to the people over there. And that's why it can be very difficult to try to understand them. But as you can see from this cell phone somebody's holding in, it's obviously very important to understand their behaviors, electrons' collective behavior for many reasons, for societal benefit as well. Okay, so why is it difficult? Well, when it comes to the information content in our solid state systems, the material systems that we would like to understand, the information content in one of these systems can be, let's see, how do I, there, can easily compare to or be greater than the total information content in the Library of Congress. This is if I take into account different defects, there can be different orientations, etc. in this crystal. So in the quantum condensed matter physics, the long quest has been, let's try to extract as much information as possible from material by doing various experiments. And over the course of, 100 years, people have really developed new types of tools, new types of measurement, trying to understand and control. So what do you want to understand? We want to understand why when carbon atoms are arranged this way, they look like this. That's a property, their look, their feel, how they conduct, these are all properties. And when the same atoms are arranged this way, they look like this. The two are obviously very different, and they have different importances. And more sophisticated example perhaps would be this high temperature cuprate, YBCO, when these atoms, particular atoms are arranged in this way that they exhibit what's called high temperature superconductivity. So we would like to understand and control these materials. And that's what we've been after as a community. And we've been trying to come up with newer and newer ways of measuring and better ways of measuring. So what are and or with the huge developments in the experimental techniques and the detector technology, what we're finding is that the goal originally, the drive had been, let's try to collect more data. Let's try to collect new type of data. But once we're sitting on the rich of this data, and we're recognizing there are data-driven challenges. And one example that I would like to go over is that of X-ray scattering. Now, X-ray scattering is a particularly old kind of tool, and it's been around for over 100 years. In 1913, the way X-ray diffraction was done, this is from the paper published by Bragg and Bragg, by father and son, in Proceedings of Royal Society in 1913. And X-ray technique had been around already for a while at this point, but the reason why they made a groundbreaking contribution was because they figured out how to analyze the data. And they analyzed the data, which looks like this, which looks very primitive compared to the data that I'm going to show you later. They analyzed this sort of data by making a conjecture. This was a conjecture they made. They thought, looking at this curve, okay, if the atoms are arranged like this, and if the X-ray beam is coming in at this angle and scattering that way, we can understand from these distance between the peaks that, well, the distance between the atomic plane is a certain and very small number. And this was an important contribution that for the first time enabled people to unleash the power of X-ray to look at subatomic scale resolution. Now, that's 100 years ago. But for the last 100 years, the mode of analysis of X-ray remained largely the same because of what's called phase problem. When we collect photons, we only see their amplitude and we don't see the phase. So it's hard to figure out exactly where the atoms are unless you have a model. So this is what we are now teaching at an introductory physics called Bragg condition, which was at the core of the model. But now we're looking at much more complicated materials, and we don't necessarily have this kind of model. Now, the kind of settings that we're now in, when Bragg was doing the measurement, they would set up the sample, make the measurement, and then rotate the sample a little bit by hand and make the measurement. But today, the way the measurements are done is by, let's see, this is a movie. The movie won't play. I'm not quite sure how to get it working. But the sample is rotating automatically, and it will span the whole large number of Brillouin zones. This little honeycomb shape that is a different, what's called Brillouin zone. That's a reciprocal space point. So the data volume has now gone up tremendously and complexity has gone up. And now we're looking at data like instead of a single peak that Braggs could see, this is thousands of Brillouin zones, tens of thousands of Brillouin zones, over tens of thousands of peaks. The data volume is now approaching terabytes. And this is the one example of data-driven challenges that the community is facing. That we have now convinced the nature to reveal itself more to us, but we don't know what to do with that data, even though we managed to get our hands on it. So my group has for last 4 years, my group has been looking at many different types of these kind of data-driven challenges, and we've been trying to embrace the data science tools, machine learning tools, to actually connect the data to understanding, data to theory, data to hypothesis. We've looked at scanning tunneling microscopy data, which was published in Nature. We looked at resonant ultrasound spectroscopy data. And we're looking at scanning tunneling electron microscopy data. These are just, a variety of settings that we're looking at. My group's attitude has been, okay, if you have a data-driven challenge, bring it to us. We'll try to learn how to learn from the data together. What the approach that generally we've been taking is that, there are these larger volume or new types of experimental data, and we want to make connection between those data and theoretical understanding. This is some model for how a doped Hubbard model would behave. This is perhaps more familiar to many of us because undergraduates are supposed to know this in physics. This is Helmholtz free energy as a function of energy and entropy. These are kind of fundamental understanding. And we would like to go from complex and rich and hard-earned data to these kind of understanding. And we would like to use data science tools such as probabilistic modeling or some neural network-based approaches and get to something fruitful. In this effort, what I've found particularly challenging is the sophistication of my subject area, physics. Generally, irrespective of which discipline you're looking at, when we say understanding, we want to go from some observation that is generally complex reality of data to some simple principles. And I think this is pretty universal, not just physics. And you want to be able to go from your principles to prediction so that you know what you think you've understood actually means something, that it has power. Problem of the domain of physics in particular is that it's a subject that was born simultaneously with the invention of calculus. It's a very old subject because through that history and subject, it's been very successful. We've been able to model real motions of balls bouncing using this very simple equation if you also learn calculus. But because of this history of the subject, for some modeling to be accepted by the community, therefore can actually contribute to the progress, we need to be able to put it back to the traditional paradigm, the language that we know how to talk about. And that is what interpretability is going to mean for us. We need to be able to think about what the machine learned in the language that we know how to think about. And in any machine learning context, you know that the more expressible the architecture is, harder it is to interpret. So what we found ourselves gravitating towards was that, okay, we will try to be as minimalistic as possible so that we can interpret. So this is kind of my general philosophy. This is a powerful commercial grade blender. Of course, it can blend anything. However, what you're after is not just blending, but you would like to know how to make better guacamole. Better tool for you is not this blender, but kind of a Stone Age tool. Because when you use the Stone Age tool, you know what you're crushing, and you know when you taste it, what you want to do better on, what's causing your guacamole to taste better or worse. So we just found ourselves keep going to, let's find simpler and simpler models so that we can interpret, and let's see how we can find the synergy between the tradition of physics, our theoretical understanding, our insight, and what machines can offer. So now I'm going to go over two specific examples. And the first example is going to be, so we want to be minimalistic and integrate the key physics principles. And the first example is going to be unsupervised machine learning for high volume X-ray diffraction data. That's why I started with the X-ray example and Nobel Prize winning, although to our eyes today look so simple, but simple things can be powerful. The Nobel Prize winning analysis or modeling of Bragg's. So we named our approach X-TECH, shorthand for X-ray diffraction temperature clustering. So you're going to see that temperature plays an important role. And that's something that I think we came up with because physicists and machine learning people were talking to each other. So I would like to first acknowledge my collaborators. This work was really heroic, epic effort by my former graduate student, Jordan Vendeley, and my current graduate student, Mike Meddy. And we were helped and inspired by Jeff Pleiss, Cornell computer science student, and Killian Weinberger, my colleague in computer science. Experimentalist colleague Jacob Ruff was actually the first person who said, I think maybe we should focus on temperature. And that was his major contribution. Also, he supplied us with data that we used as guinea pig for developing our tool. And Matt Krogstad and Ray Osborne are my collaborators at Argonne National Lab. They run the facility there at APS, but they've been also involved in providing us with these terabytes of data and helping us think about this together. Okay, so what we mean by X-ray temperature clustering, the clustering X-TECH. X-TECH is to look at temperature-dependent X-ray intensity data. This is an example. I'm showing you only 27 Brillouin zones here. This intensity is distributed in three-dimensional space. And until I saw this data, I thought, well, three-dimension, I know how to visualize. Well, I don't know how to visualize 11 dimension, but three, I know how to visualize. That's what I thought until I saw the data. The data is a distribution in a three-dimensional space of intensities that's not forming a recognizable form or shape. So to dig into this data and find interesting events that are only small portion of it is kind of looking for a needle in the haystack. And the manual approach is time consuming and painful. It takes less than a week, actually just an hour or so, a couple hours of beam time to take terabytes of data. But then a researcher will stare at it for months and months and months and over a year. And even after a year, they wouldn't have comprehensively studied the data. They would have just looked at some slices. We're going to treat this data that's taken at different temperature as a series. And we're going to treat this as if we're looking at sort of a history. We call it temperature trajectory, history of intensity at individual points in Q space, as if it is a population, somebody's history that we're trying to look at. Let's say health history, because that's how we started talking when we first thought about this problem. So the idea is if we're after some important collective phenomena that is detected in this data, and we want to do this in an unsupervised way, so we want to go in and make a discovery. We know that coming back to this F equals E minus TS, we know that temperature is what sets the balance between energy and the entropy. So if there is something collective happening driven by energy, we're going to see those regions in Q space to behave differently as a function of temperature. And we will just try to pluck that out. So this is the entire Q space of data, and we are now going to have several of these, and this is even a subset, but we're going to have several of these because we're going to look at a whole series of temperature. And we're going to try to look at subtle orderings. When we first looked at this problem, how can we analyze this high volume data without throwing out anything in a comprehensive way and discover when there is something interesting happening? It's a small, we know it is a rare portions of reciprocal space. It just happens in particular wave vectors. So it's kind of looking for that rare piece. And it's kind of like trying to look for that special piece in a pile of Lego set which I'm spending a lot of time doing when I'm not giving a talk these days. And looking for that little piece, if you try to do a manual approach of, okay, I'm going to look at one piece until I find the right piece, you can give it a try. You'll quickly discover that's not the right way to do it. It's very easy to miss. You get very tired quickly. The manual approach of X-ray data analysis that the community has been applying, has been employing for a long time, looking for some new peak associated with charge density wave, is kind of like having this sitting in front of a humongous pile of Lego sets and plucking out one piece at a time and trying to see, is this what I'm looking for? What I've learned very quickly trying to sort through trying to find the piece that my kids are asking me to find in a pile of Lego set is that, well, I want to first start by grouping them. So I want to start by first grouping them in colors, and then I will just have to look inside that particular bin if they're looking for a particular color piece from a particular manual. Now, what you need when you try to group is the sorting criteria. So for the Lego set, what I used was color. Depending on your objective, it could be the shape of the piece that matters more, then you'll sort by the shape. But what you need when you're going to go into a high volume data, trying to just go after what you are interested in, is some sort of sorting criteria. So now the problem that I'm interested in really is not that of Lego set, but it is of some collective emergent phenomena. And for that, the sorting criteria is going to be this equation here, that Helmholtz free energy is energy minus temperature times entropy. And energy is where all the interaction is. Entropy is what is driven to dominate. The randomness dominates when temperature goes up. So if there is something interesting in energy, we will see that that interesting thing happens when temperature becomes low enough so that entropy is suppressed and we can see interaction doing something. And in some points in the momentum space, maybe there isn't anything interesting interaction associated with that point, then we won't see anything special there. So this was our initial insight. And we said, okay, then let's look at the intensity of X-ray at a particular Q point in reciprocal space and temperature. And we look at different population of Qs. We look at all those points that's existing inside that cube of the reciprocal space as if it is a population we're going to try to follow their evolution in temperature, not in time. And when we came down to this idea of we want to really look at temperature evolution, and I brought this problem to a discussion with Killian, he said, well, that kind of sounds like, you want to do clustering, and it's kind of like, trying to verify speaker. So this is voice audio signal. And if you have these three signals and you want to automatically verify speaker so that you come out with the understanding, okay, these are, this red one is from female speaker and this blue one is from male speaker by looking at many different data without having a model to model going in that this is what male speaker looks like, but you want to come to the understanding of that model. You want to use some clustering approach. And what we decided to do was to use like the guacamole tool, very simple clustering tool, that's Gaussian mixture model. Now, our whole process starts by pre-processing to remove some of noise because the entire volume of data is still too big to even use a simple-minded tool like Gaussian mixture model. So if we just look at the data, raw data, this is intensity at all points in reciprocal space plotted as a function of temperature. It looks like a mess. Or it looks like a mess. You cannot just look at this and pick out, okay, this is my particular Q that is interesting. This is my order parameter. So what we do is we first have some thresholding scheme and use some rescaling. Our thresholding scheme is to reduce the volume that we have to look at. The data distribution in the reciprocal space is sparse. These are what's called Bragg peaks, and you have some intensity near Bragg peaks, but there is a large region of empty space that there is no activity happening. So we want to remove them from the population, those regions from the population that we have to track. So we threshold out those regions. Now we look at only these yellow regions. That's kind of like where this is the residential area that we're going to look at. And the scale is really huge. There's huge discrepancy in scale. At the peak of the Bragg peaks, the intensity scale can be 10,000 times larger than away from the Bragg peaks. So we also normalize in order to put it through a Gaussian mixture model. After this pre-processing, we employ a simple Gaussian mixture model. So the pre-processed temperature series for each QI that has survived the thresholding as population living in a residential area, we form a whole temperature trajectory associated with this particular Q. There is a whole temperature trajectory of intensities associated with that Q. And then we conjecture K distinct multivariate normal distributions. This number K is the only parameter that we set going in. That's the number of clusters that we assume. The rest is discovered. So this multivariate normal distribution model, probability distribution, has hyperparameters to be learned. That's the mixing weight and pi and the means and variances. And those parameters can be learned through expectation maximization algorithm, EM algorithm. And it's kind of cute because for physicists, the way how we use this EM algorithm to find these values are a little bit like doing mean field theory. That's sort of a side comment for physicists. At any rate, so we learn those parameters doing EM using an EM algorithm. Once you learn those parameters, what you have is for that momentum space point Q of I, we have this cluster assignment. That's the probability that particular QI belongs to the particular cluster K. So now we are going to apply this to some real data. And the material that we have chosen was what's called pyrochlor cadmium rhenium oxide. It is a material that is known to have two transitions. It's, the way we can recognize thermodynamic transition is by looking at specific heat. So there is this specific heat anomaly here, which is classic second order transition. And then there is another kink, little blip here, which is associated with first order transition. And there have been some models conjecturing, okay, this must be the phase in the middle, the structure in the middle, this is the structure in at the lowest temperature. What these structures are showing is that we have these tetrahedras. This is a tetrahedra. These are corner touching tetrahedras. The bonds, when it's all black, they are all the same. In this intermediate phase, different bond length become different. That's a structural distortion. And in the lowest temperature phase, they have yet another arrangement. Recently, there were some new kind of optical tool applied to this transition. And they said, these authors said, they think there may be two different things happening at this upper transition. There's a secondary order parameter. That was a sort of a puzzle that they have thrown to the community. So we decided to look at this data, X-ray scattering of this data. Nothing about what is happening in these transitions had so far been known from the X-ray perspective. And we have looked at about a terabyte of X-ray scattering data, looking at 100,000 Brillouin zones. And we're going to apply our simple Gaussian mixture model clustering. So this is the result of clustering. This is the mean that we learned in the Gaussian mixture model, and this shaded area is the variance. We've tried several different clusters. In this case, what I'm showing is a result of two cluster clustering. The cluster number, the total number of cluster is the only thing that we are fixing going in. And we've tried several different types to see whether what's the actually natural clustering in the system. And two seem to be the right number, because when we make, say, 4, one or two more clusters show up sort of within the variance. That tells us you want really the two clusters to be separated. This is a meaningful clustering result. So what this shows is this is the representation of the entire multiple terabytes of data set. That is, there are some Q points whose intensity follow this kind of overall trajectory. This is extracted because it's the mean, and this GMM mean. And there is other points that follow this kind of trajectory. And I've said, well, from the principles of thermodynamics, we expect something that is undergoing interaction to show a special temperature. And this is a special temperature that is showing up. This algorithm knew nothing about TC. We just gave the data, and it came back to us saying that, well, we're finding something behaving like this that looks like a classic order parameter. Now here comes the promised interpretation step. The interpretation step is we look at where those Q points came from. It's very simple. So these are all the purple points here. These are the purple points. And all the yellow points, these are the yellow points, and there are these blobs of yellow points. By looking at where all the purple points came from and all the yellow points came from, we can read off their reciprocal space coordinates and say, oh, this is a transition that's involving a particular type of structural change. This is the order parameter. This particular Q forms the order parameter for this charge density wave for structural transition. But I said earlier that there are two transitions here. So there are two transitions. There's one transition up here around 200 Kelvin, another transition around 125 Kelvin. And our original clustering, which treated all the intensities near a particular residential area to be, it's kind of like electoral college vote, right? This is a whole one electoral college. And we only learn how this area is different from this area. We don't learn what's going inside. And in this approach, we only learn the upper transition. That informed us that, oh, we shouldn't treat this whole area in the Q space as one block, but we should try to see what's going inside. And when we looked inside, we found 4 clustering to work better. And now these are 4 clusters. Now we are looking at much larger number of Q points in effect, because we are letting the Bragg peaks be all within the Bragg peak to have individual opinions. So this is looking at what happens near the lower transition. And then we now go to interpret. We look at where these different clusters came from, and we find, oh, the red cluster came from sort of the halo of some of these Bragg peaks. There is a regular pattern. And the blue cluster came from the halo of some other Bragg peaks. And then there's this black cluster, which is in the center of the Bragg peak. So when I treated this whole thing as one block, I couldn't see the distinction between red halo and blue halo because it was dominated by the black behavior. Black is the Bragg peaks that are allowed. And so we did this clustering, and we found, oh, there are these halos that are behaving differently for different Bragg peaks. And then we went back to our X-ray experimentalist experts and said, look, there is a halo of a Bragg peak that is behaving very differently. And please look at this Bragg peak and that Bragg peak because they are behaving differently. At that point, our colleagues went and looked at the raw data, because once it narrowed it all down to just go look at these two Bragg peaks, they are different. They looked at these two Bragg peaks, and for these experimentalists, our colleagues at Argonne National Lab, they were not used to trying to look at inside the Bragg peak. They always thought of Bragg peak as a Bragg peak. And we said, but look, if you want to be able to study what's happening inside the unit cell, that information has to be inside the Bragg peak. We got to study inside the Bragg peak. And when they looked inside, they found, oh, so in this red halo region, that's the halo of this Bragg peak, the diffuse area, that's the language that my experimentalist colleagues use, the diffuse area does nothing. But in the blue halo, this diffuse area is very visible. The vertical axis is temperature here. The diffuse area is visible in the intermediate temperature range. So this diffuse region, that's blue halo, um, I think maybe my assignment might be flipped. So there is a halo that goes up, becomes visible, and then it goes down in this intermediate phase, a phase in the middle between the two transitions. And then we were able to further reason, oh, this is showing these peaks with the halo is telling us about the fluctuation, the soft mode that is going to drive the transition lower. And that allowed us to study, this is the vertical motion that is driving this transition. And after this transition, the internal arrangement inside this really big unit cell, let me just go back to, this is the unit cell. Going back to carbon, there are only 2 carbons per unit cell in a honeycomb lattice. There are a ton of atoms in the unit cell in this material. Because there are so many atoms inside the unit cell, when things happen, but this whole unit is repeated, it's really not easy to see from X-ray. But then there aren't any other probes that can really tell us either at this kind of length scales. So this whole exercise that we went through, we are just finishing and working in the process of writing up, was really eye-opening to all of us involved. It was an epic effort on my group's part, because we knew nothing about the material. We knew nothing about how the measurements are done. We had this vague idea, it must be possible to connect F equals to E minus TS to how we discover phase transitions and what's happening inside the phase in this high volume data. That was a year and a half ago. And we talked to our colleagues in computer science, and going through many different analogies, we were able to communicate what we are after and came to this rough idea of temperature clustering. And then we had to access data and convince our X-ray colleagues that this is the right way to look at it. It was an epic effort, and I think it was biggest adventure in my entire career. But it's very, very satisfying. And we are convinced that this is going to revolutionize how we look at X-ray data going forward, because as you could have seen, looking at what I presented, it's very simple. The model is very simple. It readily admits interpretation, and it is going to be now, oh, why is it flying? It is going to be now integrated into how the measurements are done, because the whole thing takes a matter of 10 minutes instead of a year, 6 months. So this is going to be integrated into how measurements are done at facilities, so that our experimentalists can enter the mode of take the data, get a cup of coffee, and look at what discovery they made instead of waiting 6 months to a year. Now, I think I'm running out of time, so let me tell you just a little bit about another model. I don't think I have time to go into detail, but this is what we call correlation CNN, CCNN, for quantum images. And the problem, these are my collaborators. This effort was driven by my amazing graduate student, first year graduate student, Cole Miles. And we've had great benefit of working with Ryu-Han Woo, a computer science student, and Killian Weinberger, my colleague. And the problem came to us through collaboration with Annabelle and Eugene at Harvard. What we wanted to do was to look at quantum gas microscopy data. And this is how the quantum gas microscopy is done. We can look at atoms, real atoms, in optical lattices, and they are used to simulate hard to solve models such as Hubbard model. And what you're getting at the end are these images. When I showed this image to Killian, my colleague in CS, he said, well, that kind of looks like a Go board. That's got to be a very simple image to analyze. You shouldn't need convolutional neural network to study that because the volume of data is actually not that big. It's just black and white. What are they trying to simulate? They're trying to simulate the Fermi-Hubbard model, the problem that has remained unsolvable for tens and tens of years. And we believe this is sort of a key to understand high temperature superconductivity. And the question is whether, what kind of models that people are putting out have relevance in real data. So colleagues like Marcus Greiner at Harvard have worked hard to set up these atoms to simulate the model. And they can now make these kind of images. And this is an image of antiferromagnet where what's shown are one spin species. So where you see color, where you see bright is one spin and the other is absent, is opposite spin. And this checkerboard type is called antiferromagnet. And one would like to figure out what happens when you try to take out some of those spins. And these are conjectured theories that we would like to tell apart. So the problem is there is this specific problem that we are looking at here. But this is sort of a general mode of how we look at experimental data. We look at wonderful data and we want to figure out which theory best describes that data. And for this particular problem, there is this a model called geometric string and this other model called pi flux theory. And both are trying to model what happens when you remove some spins. And they look different, very different in pencil and paper level theory. But when you simulate them, the conventional approaches of looking at magnetization make them look very similar. Spin-spin correlation also make them look very similar. So Annabelle and Eugene and colleagues thought, how about we use CNN? They used off-the-shelf CNN, which has a problem of over-parameterization given the simplicity of data, and it's a black box because of over-parameterization. Now, I'm not going to go into the detail, but what we have now come up with is a simple architecture that is kind of designed to really work with correlations. So we have a phase where we do the convolution. Now, if you are familiar with CNN, this might be a simpler way to look at it. This is a stage of doing convolution. That's the filter, that's the input. But then we make further higher order in input filters that can be obtained all from the same filter. One filter gives us a whole cascade of higher order filters. And these higher order filters allow us to interpret because they actually measure the correlation function. So after training the neural network with labeled data associated with two theories, we did the regularization path analysis and learned, oh, this correlation function really tell us apart. And that was a fourth order correlation. And then we were able to look at, oh, let's look at what that filter means when we look at the fourth order correlation. And we see these patterns. And then we were able to say, oh, you know what, this pattern is actually present when we make a string. So this is a geometric string cartoon, and we see these kind of parallel arrangement in the wake of this hole, and we see this kind of arrangement when we have a hole nearby. And this is a pattern that we can understand. These are the pattern we can understand as a fluctuation around antiferromagnetic pattern. So in this work, we were able to, the architecture that we are proposing is sort of a variation on CNN, but instead of just doing a regular convolution and having the regular nonlinear step or even making it deep, we're proposing to build in what we physicists care, that is the correlation function. And this will allow the experimentalist to go after measuring these correlation functions themselves to try to tell different hypotheses apart. So I'm going to now summarize. This is a summary. I've talked about two examples, spending much more time on the X-Tech, but the idea is what I hope you can take away from this talk, irrespective of your domain, is that my message, at least my takeaway message, is if you want to we believe to make progress and contribution in scientific understanding and scientific discovery, integrability shouldn't come as an afterthought. It should be right there from the beginning. But when you try to interpreting is now not a matter of whether you can see which neuron fires when you see pizza, but it's more in this sort of a sophisticated framework of that scientific domain. We need to be able to put it back into the framework. So for X-Tech, we focused on roll temperature play as the key universal control parameter that controls between the good old randomness of entropy and system-specific interaction and energy. And that will allow us to discover, and we believe this approach can be generally applicable not only to all X-ray, but also to other diffraction techniques such as neutron. And the second thing that I talked about was for real space data, trying to make use of correlation function, that we care about correlation function, and using that information in convolutional neural networks so that we can, when we discover something, when we discover distinct features, we can back out what that means in terms of correlation functions that we know how to think about. So with that, I would like to thank you for your attention, and any questions, I will take any questions.
Yeah, thank you. No, that was, excuse me, that was spectacular. Everyone should clap, and people who have questions should raise your hands. I want to guess use my prerogative as a moderator to ask the first question. So the first part, this X-Tech thing is spectacular. I mean, I'm really quite stunned. It's so simple. And I guess there's so many questions. I guess one is that, I guess I have two questions. So one is, I mean, do X-ray facilities actually store temperature dependent? I mean, is there like all of this data to now go mine using this method? Because this, protein, I mean, we can sort of make a long list of things that you can use this way of thinking to go after. And I just wonder if it's now like, if this is something that can be explored. That's question number one. Question number two is, you talked about interpretability, about using simple models. And of course, the Gaussian mixture model seemed like exactly the right thing. On the other hand, you didn't end up having that many different groups of Q vectors. I mean, there were 2 or 4 or whatever, right? And one could imagine that if one started using this methodology for more complicated materials, it could really get much more complicated. And if I understood you right, the most important thing for your method was to identify the different groups. That is, you don't actually, the interpretability of the model was less important than picking out the groups and trying to see what happens. And so I just wondered if you can comment on those.
So I guess there are two questions. First is, where else can this be useful? Like, do facilities store temperature dependent data? Is this kind of data generally available? And second question was, what about different models? So the first question, as the data volume got bigger and bigger, experimentalists actually do not go home with data. The data remains at the server at the facilities, and they have really invested in storing the data and how to queue the data. So if the measurements are done with temperature dependence in there, yes, they are available. But this is another thing that we learned as we try to do this. People didn't necessarily, it's not difficult, but just they didn't see the utility of having, multiple temperature data, and didn't see the utility of having, data over a whole range. Maybe they would do measurement right near the temperature that you think is interesting. So as we integrate this approach, our analysis, X-Tech, into facilities, that will also change the modality of experiments, how the experimentalists do the measurements. And we started discussing this with one data set that we used as a guinea pig. And then we applied the data set that I showed you today was not what it was initially developed on. And we are now looking at several other data sets that have temperature dependence. But I really thought that there's a potential that this kind of approach can change how people do measurement, how they look at data, and that's why we reached out to work with Argonne Group, because they are right there at the facility. So I think this can be useful in many different phenomena that involves any kind of thermal transition. And it doesn't even have to be temperature. If you have a parameter that you're controlling, because that's majority of how experiments are done in condensed matter physics. You have a tuning parameter, temperature, pressure, composition, and you try to see what happens when you change that. So in all of those, one could envision doing this kind of series clustering when you have a control parameter. Now, the second question was...
Una, can I just interrupt?
Yeah.
Before you answer the second question, would you
Yeah.
mind sharing your screen so that the next speaker can put hers
Oh,
up?
yes, yes, absolutely, yes.
Sorry.
Yeah. Okay,
So,
good. Yeah.
anyway, but please, second question. Yes, simple
Yes.
model.
The second question was, yes, we initially started with Gaussian processes as another example, which is kind of related to, it's a more sophisticated model than GMM. And for the X-ray data, because with the volume of the data and the sparsity of the interesting regions, it was hard to train Gaussian process. It needed really strong prior. So we started with Gaussian process, so we ended up with GMM because we could make GMM work. But I think it really depends on the kind of structure of the data. There's nothing about this whole philosophy that is tied to GMM, really. And even with GMM, when you have multiple clusters, you can discover that too. But in the crystalline data that we are looking at, it just turned out that we tried up to 8 and didn't make a difference. So we kind of learned what's the right number of clusters that's inherently in the data set. And we would like to try to kind of automate that too. That can be kind of explored and learned.
Good. Well, thank you. That was really a spectacular talk. You got us off to exactly the right foot. We have to move on to the next speaker now, but everyone should clap. You have to clap virtually because otherwise it's very lonely for the speaker who has no idea how inspiring they've been. So anyway, but thanks, Eun-Ah. And I guess I'm going to turn it over to Scott, who's going to introduce Barbara.
Okay, great. Well, yes, thank you, Eun-Ah. That was an amazing talk. And we're thrilled to have as our second speaker, Professor Barbara Engelhardt from Princeton University. Prior to coming to Princeton, Barbara had studied as an undergrad at Stanford and then received her PhD from Berkeley, and then spent 3 years at Duke University as an assistant professor before moving to Princeton. Interspersed with all of that academic experience, she spent some time working at JPL, at Google Research, a year at 23andMe, which is a DNA ancestry service. She's received numerous awards, including an NIH K99RO Pathway to Independence Awards, Sloan Faculty Awards, and NSF Career Awards. And her research focuses on statistical methods and machine learning methods for analyzing high-dimensional biomedical data, which is exactly what the type of thing we're excited about for this workshop here. So her lab studies genetic variation and co-expression networks, spatio-temporal models for single-cell transcriptomics, electronic health records, the list goes on, really fantastic research. And she runs the Princeton Beehive, which is the Biological and Evolutionary Explorations Using Hierarchical Integrative Statistical Models group at Princeton University. I have to wonder if the Beehive is really a backronym for her initials here. So something to consider. We're thrilled to have you here, Barbara, and very excited for your talk. So please take it away.
Thanks so much, Scott. And thanks to the organizers. Can everyone see my slides okay?
Yes.
Good. Okay, great. Okay, great. So today, in conjunction with the purpose of this workshop, which I think is super exciting, I'd love to tell you about some of the data using unsupervised learning. And I should say my disclosure is below. I'm actually on leave of absence this year, and I'm on a number of scientific advisory boards. Whoops. Let's see if I can move. Okay. Okay, so let's start by sort of defining what unsupervised learning means. So I think about this actually with respect to exploratory data analysis. This term was coined by Tukey in 1977, actually, as he was working at Princeton. So the real idea, I think, behind exploratory data analysis is that you explore your data for the purposes of discovery. So, given a matrix, for example, here, we can find patterns, we can find trends in time series data. We can look at correlations among features, among the samples, and figure out what's going on underlying the data. Using these exploratory data methods, we can then develop data-driven hypotheses where we actually have to go and collect new data sets to be able to test that these patterns are reproducible out of sample. I think a lot of EDA models are really focused on interpreting the fitted model, not really thinking so much about prediction, although prediction is often used when trying to validate the results, the patterns found in these models. And often EDA is actually done using a visual analysis. So I'm going to show you a lot of images today. The focus is usually on graphics and plots rather than actually p-values. I think my main argument about exploratory data analysis is that it's incredibly undervalued in sciences generally. I think it's undervalued because it's so hard to do well. So in particular, I think a lot of the assumptions of exploratory data analysis are not well characterized. If you hear someone talking about whether it's TSNE or PCA or other types of approaches for EDA, the assumptions that underlie those methods are actually quite severe. And if the data don't fit them, then you can get weird anomalies that you don't really think about when you actually visualize the data. There's no real null hypothesis. We don't know what this data matrix would look like without these patterns in them, really. And so it's really hard to do any sort of test of statistical significance. Often the conclusions that we come to, these discoveries, these hypotheses that we come up with are based on qualitative evidence, just eyeballing things, going and looking at out of sample data. I think very importantly is that oversimplified models like PCA, like TSNE, may miss biologically important signal. And I think this is important because a lot of the signals, especially that we see in genomics, are very, very, very small. And of course, the data are extremely high dimensional. And so it's important actually to build substantial structure within your model to be able to sort of build the scaffold, if you will, to be able to identify these important discoveries that we need to find. I think also very importantly, given sort of the stage of life cycle we're in with respect to genomics, is that the results of exploratory data analysis are really hard to evaluate. Even within factor analysis, for example, it's hard to compare across factors. They're hard to validate. It's hard to know when you've sort of got meaningful signal or not. And they're also hard to reproduce. So given two separate matrices on different sets of samples, you may, for various reasons, get different projections. And this is obviously concerning when we think about sort of scientific reproducibility. Whoops. Okay. So my talk overview is just going to think about a general hierarchical framework for EDA, which is going to come down to some version of Bayesian factor analysis. I'm going to give two examples of where we use EDA and genomic studies, both of which have produced discoveries that we've gone off and validated in out of sample data. So the first one is thinking about paired observation models to identify what we're calling tissue morphology QTLs, which are actually mutations in the genome that appear to change what images of tissues look like. And the second one is robust nonlinear models for single cell normalization. And what I'm going to tell you there is that using the EDA type models, we're actually able to spatially resolve disassociated cells. So we're able to put on an XY coordinate cells that were just assayed from after being pulled apart from each other. And we're able to understand locality in cells. Okay. So the first thing I want to say, because a lot of the work that's done in machine learning right now is done with deep models where the structure is quite different than the models that I'm going to be talking about today. And so why am I using hierarchical latent variable models for all of the EDA that I'm doing? I have to say there's obviously, as we all know, a sort of explicit trade-off between expressiveness of the model with respect to the structure and computational tractability. In general, in my work, I choose models that are more expressive. As many people in this room, Scott, for example, know, genomics and neuroscience data are definitely not what we consider big data. They exist in this sample limited but feature rich world where we might have millions of features per sample, but only a handful, 100, 1,000 samples. And this basically means that we can't use a lot of the methods which are built on this sort of large and growing feature set, or sorry, large and growing sample set, and not the feature set. So a lot of machine learning and statistics rely on large numbers of samples and fixed numbers of features. Again, I'm choosing to do this hierarchical latent variable approach because of this situation that we're in with sort of these high dimensional data sets. And again, just because the number of features are so big relative to the number of samples, I end up adding a lot of structure to the models just to make sure that small signals of association or patterns are identified within these very high dimensional data sets. The fitted models now have mechanistic interpretations, which is really nice. So we can go back to biologists and actually ask them what they mean. Also, uncertainty is explicit, which ends up being a really important thing in genomics, because we do end up taking these discoveries and asking questions about them downstream, and being able to quantify uncertainty and push uncertainty downstream is super important. So then the obvious question, if I'm going to add structure, is how do I achieve computational tractability? And again, there's many ways to do this. I'd say, a majority of my actual mental time is spent on thinking about computational tractability, but it's not going to come up at all in this talk. I want to talk about our discoveries instead. Diving into the Bayesian latent factor models that I'm talking about. So here's a very vanilla version of just a factor analysis model. So throughout this talk, I'm going to be referring to Y as our observations. These are going to be genes on the rows. I'm going to say there are P genes. And remember, again, genes is going to be pretty big in humans. Genes, there's about 20,000 expressed genes. Samples is going to be much smaller. This is on the columns. We're going to call that N. And so what I'm going to do with this Y matrix, which is the only part of this figure that's observed, is I'm going to project it down using weights lambda, or these factor loadings, down to some subspace X. And X is going to be a K by N matrix, where K is going to be much smaller than either the number of genes or the number of samples. So I'm essentially going to do a low dimensional projection of Y. And then very naively, I'm just going to assume that we have Gaussian noise at the back end of this, independent Gaussian noise. And so if you think about a single element of Y, right, a single Yij down here, it's just going to be a draw from a Gaussian, where the mean is just going to be a linear combination of the genes and samples, the low dimensional projection of this in the latent space. And I'm just going to assume noise that's associated, variance that's associated with the genes. So there's going to be one variance term per gene. A lot of the different types of dimension reduction methods that we use are just latent factor models, but with various constraints. So principal components analysis is just this exact model where the factors are orthogonal and the loadings are orthonormal. Admixture models, or LDA, where the loadings, the columns of the loadings and the factors both sit on a simplex, so they're non-negative and they sum to 1. Non-negative matrix factorization is just count data, where again, all of the elements of lambda and X are assumed to be non-negative. And then factor regression models is basically this exact model, although now you're going to add a handful of fixed covariates. Say if you know everyone's age, you can add that to this model just as a linear term. So there's a lot of challenges with these sort of vanilla versions of these factor models. In particular, the data likelihood, or if you write out the actual likelihood of this model, given the formulation that I've told you, the likelihood is invariant to orthogonal rotation. So I can rotate lambda and X by the same orthogonal rotation matrix. I can scale each of the lambdas and X's by A and inverse A. And then label switching as well, where I can actually just permute K for both the genes and the samples simultaneously and get the exact same likelihood. And so these invariances mean a couple things with respect to how we can interpret these models. So number one, they mean that the factors could be correlated. I've never explicitly talked about orthogonality with this model. Correlation may actually be a good thing when some of these signals are correlated, but that's a different story. The factors will not be comparable either within or across applications because of the scale invariance. So this is kind of disturbing sometimes. It's hard to label the factors in a meaningful way. And this idea of labeling factors is going to come up again and again when we think about actually extracting sort of meaningful signals and discoveries from these models. So our entire goal is basically to look at these each of these K factors and figure out what they represent, what patterns they represent in the underlying data. And this requires a lot of domain-specific information usually. So there's a handful of, there's a handful of ideas to be able to address these problems. So number one, we could add sparsity, which is in general something that I'm going to tell you about. Number two, we're going to study structure that's invariant to rotation. So you could actually compute the P by P covariance matrix using these estimated terms, and that's invariant to rotation. You could study that directly. And then I think another thing that's incredibly useful is to actually take the patterns that we find and validate them using held out or out of sample data. So very briefly, I'm going to say that we add a lot of sparsity to these models to aid with interpretability. And the way that we do that is actually fairly heavy-handed. And I can talk about why it's important to be fairly heavy-handed about it, but I'll tell you what we do first. So we actually have 3 layers of essentially a horseshoe prior to regularize these models. So first we regularize lambda by putting a global regularization factor on every single component of lambda. This basically tries to shrink everything in lambda, every element of lambda as close to 0 as possible. Then we actually put a factor-wise regularization term on it, also a horseshoe prior. And this sort of selectively rescues a handful of the K components that we have in lambda. So it ends up actually zeroing out a lot of the components that aren't necessary according to the data. And then finally, we add a local, a factor-specific, an element-wise horseshoe prior also, so that within these sort of preserved components, we'll actually have sparsity there. So at the end of the day, we're hoping, just like this sort of this picture shows, we're hoping to get only small subsets of genes represented within each factor. So then we can understand what those genes represent with respect to the signal that we're looking for. So one thing that was pointed out by both Leek and Story and a handful of papers from Alan Wilsky's group is that when we think about scientific structure, structure and scientific data, it's often a combination of very dense factors, so components of the experiment that essentially affect all genes, and very sparse factors. So it's a handful of things, handful of components, environmental factors say, that only affect a small number of genes. I'll give you some examples of this, but we actually end up putting a 2-component mixture model on these priors so that we can get either dense or sparse factors and estimate those directly from the data as well. And that turns out to be important. So what do these factors represent? So, the factors are sort of the discovery that I'm talking about. So as I alluded to before, we can think about the covariance matrix, a gene by gene, P by P covariance matrix. So this is a 20,000 by 20,000 dimensional matrix that's really hard to estimate from small numbers of samples. And when we think about just running PCA, we can actually pull out this covariance matrix using lambda transpose plus the residual covariance matrix that we have, which is just a diagonal. And this, again, gives us a low-dimensional representation of this covariance matrix, which is sort of a phenomenal thing. More importantly, I guess equally importantly, this is a non-singular matrix generally because we essentially add this really quite heavy diagonal term. So we can manipulate this matrix. We can usually invert it and do some other cool things with it. So there's a lot to think about with respect to this, but it's really nice that we can pull this information out of our latent factor model essentially for free. Another way you can play with these models is by actually adding sparsity also to X. So this is referred to as Bayesian by clustering, where now if you think about a single component in lambda and X, you have now a subset of genes and a subset of factors. So this is kind of nice, again, in thinking about interpreting these patterns, figuring out if a pattern in a subset of genes is also sort of localized to only a subset of the individuals as well. So let me give you a deeper dive into that idea. So when I actually think about these patterns, this is the picture that I have in my mind. So as I told you at the beginning, each element of Y is actually going to be essentially a sum over the outer products of all of these lambda and X terms. So when we think about a doubly sparse, it doesn't need to be, but let's just think about it in this way, a doubly sparse lambda and X, we have something like this covariance term right here, or this outer product right here, which is a sparse lambda and a sparse X. So again, here we're basically saying within these samples in Y, within these genes in Y, there is some covariation occurring. So our job now is, this is the pattern I'm talking about. These are the discoveries that I'm talking about. And our job now is to understand what's special about the subset of samples. Maybe they're all smokers. And what's special about these genes? Maybe these are all genes that are dysregulated in your lungs when you smoke. Why do you not see this variation outside of smokers? So these are the discoveries that we want. And again, it does take a lot of domain-specific knowledge to be able to sort of label them with what those discoveries actually represent. So let's actually dive into that a little bit with respect to the data that I'm interested in. So when I think about labeling these latent factors, the labels could be one of many, many things. And you really have to be creative when you're doing this labeling. So for example, there are a lot of technical covariates. So the #1 contribution to variation in gene expression levels is actually literally the lab technician who did the experiment. So all the variation is due to who did this experiment. But batch, library prep, all of these contribute to variation, unfortunately. And those are signals, obviously, we want to remove as much as possible. Biological covariates influence a lot. So what tissue are we talking about? How old is this person? Is this a male or a female? What's their BMI? Smoking status, as I alluded to before, actually has come up in a lot of our models, which is sort of surprising, I think. Population structure comes up a huge amount. So the ancestry of the individuals ends up being a really important component here, not necessarily for genetic reasons, often for sociological reasons, unfortunately. And then genomic covariates, which is sort of the thing that I think of as most interesting in my work, where there's a particular mutation in a handful of our subjects, our people in the study, the samples, and that might affect only a handful of genes. So that's where we might get this doubly sparse, very, very sparse representation of covariation, where again, a handful of samples has a mutation that only affects a handful of genes. So if you can imagine, with all of these different components, we have to have a very large number of K, and usually K is actually even larger than the number of samples that we have in practice. Okay, so I'm going to dive into the discoveries now after already taking a lot of time. Scott, if you want to give me some sign when I have a few minutes left, that'd be great. So first I'm going to tell you a little bit about tissue morphology QTLs, which I alluded to. What I'm showing you is something called a histological or a pathology image. So these ideas of these images have been around forever. They're basically the way that most cancers are diagnosed. So what happens is that a doctor will take out a small sample from a part of your body. Here, for example, is a very small piece of colon. And what they do is they slice it very thin. They stain it with essentially iodine. They put it under a microscope and they take a very high resolution picture. So for example, in these pictures, these little sort of black purple dots that you're looking at are the nuclei of the cells in this image. And you can see sort of where they are in space and, sort of what the cell organization looks like. These are incredibly important, like I said, in diagnosing disease. But it's really hard to think about a picture as a trait. And so one thing we were interested in doing is taking these pictures and saying, how do we see associations with gene expression, with genotype, with cell type, associated with actually the images. And so that's sort of where we started with this question. So the exploratory data analysis tool that I've been using for this work is called sparse canonical correlation analysis. So this is exactly the same model that I showed you before, essentially duplicated. So here we have actually two different data modalities. We have Y1 and Y2. The number of samples is the same in both of them. In fact, the samples are paired. So where we have data modality for one individual, we also have that same data modality for the second individual. And so what we're going to do is we're going to use modality-specific lambda matrices to project both of these data modalities down to the identical X. So I've just replicated this X matrix between the top and the bottom version, but it's actually the exact same matrix that we're projecting to. So the idea here, again, is that we're going to take these high-dimensional disparate data modalities on paired samples, and we're going to project them using a linear projection down to a shared latent space. And what we're going to do is essentially look at the weights. So when we look at lambda 1, if we take a particular component here, we're going to say that there's a handful of genes in this case, and maybe a handful of image features that appear to be mapping to a component in this latent space. And that's going to tell us what genes covary with what image features. So to be able to use this model, we need to actually have a set of image features. We obviously can't do it just on sort of pixel representation of the images. Right now, it's unfortunately fairly infeasible still to do manual annotation of these pathology images. We have 27,000 of them or something like that. And image segmentation methods also were fairly naive with respect to these types of images still. So we're not going to use that. So we just did essentially the simplest thing possible, which is just use a convolutional autoencoder as the architecture shown, very, very simple architecture, to be able to pull out 1,024 features for every single image. I should say this isn't sort of the end of the story. We actually had to do this fairly cleverly too, because we can't put the entire image into one of these convolutional autoencoders. We had to split it up into different patches. We actually had P patches, 100 I think we set P to. And then when we think about running each of these patches through the CAE, we actually pull out the empirical mean of that set of features for any single image across all of the different patches. And because each of these are sort of jointly Gaussian, this ended up being a reasonable thing to do, but we did test that this was a reasonable thing to do. Okay, so now we actually have some idea of what we're talking about. So for these Y1 and Y2 matrices, we start out with about 500 samples, let's say. P1, as I said before, for humans, there's about 20,000 genes expressed in a particular sample. If we have a paired histology image, we have about we have exactly 1,024 features, given the results from the convolutional autoencoder. And then again, we're going to look at these components, these factor loadings, to be able to figure out which of the subsets of genes correlate with which subsets of image features. Okay, so I'm going to tell you about some results. So brain lower grade glioma is one type of data that we used. It's from the Cancer Genome Atlas, the Cancer Cell Atlas. No, I'm not saying this right. The Cancer Genomic Atlas. There's about 500 images, and a lot of them are either primary or recurrent tumor samples, and this is what they look like. I should say that these are fairly poor samples. There's a lot of degradation of the tissue here. So we find things like this. So just to orient you, this is a bunch of features or a bunch of images that were very high in the loadings, and this is a bunch of features or images that were very low. And you can see that visually that there's a difference between the two of them. What it looks like is actually these images over here, the sort of darker purple ones, have lost a lot of their axons. Axons are the first thing to degrade as tissue degrades in brain tissues. And so we see that actually the genes that are expressed here, which are described by the terms, enrichment of specific functional terms associated with the genes that are non-zero in this component, appear to represent genes that are expressed in axons. So this is a sort of nice proof of principle that this sort of thing works. Actually, we found something that I wasn't expecting to see at all, but again, these are brain samples from this brain tumor, these brain tumor samples. This is, I think, the 7th or the 9th component. It's somewhere sort of downstream. What we can interpret from this is that in these samples on the left, there's actually a lot of sort of density in there. It's sort of packed. And it turns out what those sort of packing is there's a lot of immune cells in them. Whereas in these cells on the right, or these images on the right, there's actually very few immune cells in them. And the way that we figured this out was by looking at the genes that are correlated with these differences in these images. And these genes are entirely associated with cells that are fighting immune cells involved in fighting infection. And so that's how we sort of interpreted the visual differences in these images. I want to talk very briefly, I know I'm getting towards the end of my time, about GTEx, which is another data set that we have with about 500 individuals, about 8,000 total samples, 7,000, I guess, 2/3 male, 1/3 female across many different tissue types, 44, 49 now, I guess. With a lot of brain, I should say. Again, we have about 2,200 total histology images, and that's going up to about 27,000 in the newest data set that we have, but I don't have those results yet. If you just plot t-SNE for the images that we have, you can see that in the image data, well, I'll tell you what to see, because no one here is probably a radiologist, a pathologist. Over here in the upper left corner is muscle tissue, but besides that, it's actually sort of a really big mess. So these sort of these images that look a little bit like watercolors are thyroid, and they're actually quite unusually spread out pretty much all over this region of the clusters, and it doesn't make any sense. So I guess what I'm saying is these features that we come up with for the images aren't necessarily all that meaningful in terms of splitting the tissue types. So the first thing we do is look at some of the components and see that they're associated primarily with distinguishing muscle tissues from everything else. So here are a bunch of muscle tissues. They look a lot like ham. And then here are other tissues that aren't looking like ham. And then the genes that are also associated with these two differences, these components, are associated with all of the functional terms that represent genes that are only expressed in muscle tissues, basically. We can actually take this a step further, and this is sort of the discovery that I was really excited about. So it turns out if you have a mutation, here's your DNA, my pictorial representation. If you have a mutation somewhere in your DNA, often a QTL, a expression QTL, is something that will actually change the relative levels of protein in your cells, just based on this change in this mutation that's added. And we asked the question whether this change, this mutation, actually the downstream effect was to make differences, to lead to differences in tissue morphology. And so we asked that question very specifically, and to our surprise, the answer is yes, you can see this. So here's a particular genotype, which means here's individuals where there's 0 copies of the mutant allele, 1 copy and 2 copies of the mutant allele. And you can see that with respect to the image feature, there's very big differences across the images associated with the samples with these genotypes. So to sort of fast track to the punchline, it turns out that this mutation is actually well correlated with inflammation in your colon. So through a particular gene that actually is good at repressing inflammation and talking to pathologists that actually can understand these images, it looks like the upper, sort of the more extreme images here are very inflamed colons, whereas the ones below, this one in particular, is a normal colon. And again, testing out of sample, we went to the Human Protein Atlas and found that differences in this particular gene actually have very different survival curves for colorectal cancer. So sort of indicating that this is a reasonable discovery. I should say that we've extended this model pretty substantially in a UAI paper. So now actually, before we were doing just the convolutional autoencoder and then CCA, and now we have an end-to-end estimation inference so that we actually only learn the image features that are going to be predictive of gene expression values. And we're getting quite good results from that. Here's an example, but it's not relevant considering I have basically no time left. I should say another thing we're doing is dense labeling, which I'm extremely excited about, where we have an image and using these models, we can actually put on top of it where we think particular genes are expressed. So if you remember, we have gene expression that's across approximately that sample, but now we're saying within the sample, within that image, where is this particular protein expressed? And again, with outside data, we're validating our results from the Human Protein Atlas. So extremely briefly, I'm going to say that we also work with robust nonlinear models for single cell data, which you've probably heard about. Single cell data is just one of the most exciting things I could possibly imagine, because we can understand how cells interact and influence each other. Okay, so let me just fast forward to the exciting part. There are a lot of statistical challenges. I will say that we use essentially a GPLVM with a student's T distribution on the residuals to account for the outliers in the cell types. This helps a lot. You can do a lot of cool things with it, including clustering and understanding cell cycle, and then obviously dimension reduction in visualization. You can also think about cell trajectories. So when you think about cells developing from a primary cell to a fully developed cell, you can actually model that very nicely in this low dimensional space. This is actually just neighbor joining within 2 dimensions of our latent space, and it seems to work really nicely. The thing that I'm really excited about is this sort of what we're calling semi-supervised models. And this is actually an idea that I came up with, or I shouldn't say I came up with, but was done in collaboration with Jonathan Pillow and one of his students, Anqi Wu. And the idea here is basically to add some metadata to these models, to these GPLVM models. And now the idea is that this metadata can represent anything we know about the cells, whether it's the type of cell that they are, how old they are, how far they are along in development. And the thing that I'm really excited about is actually including XY coordinates. So now we know where in space this particular gene is located. And what that means is that when we have a handful of cells and the lower axis that are found assayed using spatial technologies, we're probably missing a lot of genes, but we do have XY coordinates. Whereas when we have a huge number of RNA sequencing cells, there may be millions of them, but we have no XY coordinates, but we do have 20,000 genes. So what we do is actually do the imputation, both of gene expression levels in the spatial data and the XY coordinates in the disassociated cell data. And so this is the major discovery, I think, that we have in this data set, where now we can take all this disassociated data, map it down to spatial coordinates, and get an idea with a model-based system where each of these cells, these disassociated cells, sit with respect to each other. And so on top of this, we can do a number of things. Like, for example, look at this sort of differential relative enrichment of neighbors for cell types. And here's some brain data. And the idea is that there's a bunch of different types of cells. And we can see that for specific pairs of cells, controlling for their abundance and also controlling for their size, we can say that some cells sit to each other 7 times more often than they should relative to normal. And this is based on disassociated data. So this is a pretty cool result that I'm really excited to bring forward for a lot of the new brain data sets that we have. I should say again that this is originally work with Jonathan and Anqi, and we did this with fMRI data sets, and it was never published, but I love it. Okay, I think that's basically it. So in conclusion, exploratory data analysis often, as I talked to you about, requires very highly structured models. But the biological insights you can get from these structured models are really exciting. So in this work, we characterized gene covariation. We found genetic variants that actually regulate tissue morphology. And we were able to think about labeling cell state and spatially resolving dissociated cells. So when these structured exploratory data analysis models are working, this is the part that I'm so excited about. You know, this is every day, this is why I get excited about my work, is there's sort of these two obligations, right? Number one, we need to validate all of these discoveries that we come up with. We need to really think about how to carefully collect data out of sample to validate each of our discoveries. We can't just rely on these patterns, seeing them in one data set. But then second of all, this validation, this process of validation actually is the thing that yields these biological discoveries. It's so exciting to be able to take these patterns that we find in these data sets and actually build experiments around them. So that's one way I think that machine learning has been so exciting in this context. So with that, I think I'm done. Oh, wait, no, that's not right. Yes, thank you.
We're all clapping, Barbara. That was fantastic. I think one of the biggest things that's missing from these Zoom versions of workshops is that we can't all show our enthusiasm for your talk in the same way. But that was really, really great, and so many fun things to follow up on. I wish we had hours more time to talk about all these things. We have one question from the audience from Sam Yin, so maybe I'll let him start here. Is it possible to unmute him so he can ask the question directly? Let's see.
I think it works.
Oh, great.
Well, thank you for your talk. I want to ask about the very first part when you talk about factorizing the gene by samples matrix. You said something about like using domain-specific knowledge. Like if you know something about like batch effect, then you can. Are you talking about like encoding that in your prior, or just use that information to identify the patterns after data fitting?
Yeah, that's a great question. So you can do it either way. We've tried it both ways. I like keeping the information out most of the time, just because I use that information. I rely on that information very heavily to be able to do this sort of component labeling, as I was talking about. So if I leave out cell type, or I leave out smoking status, or whatever it is, whatever dimension it is, then I can go back later and being agnostic to those labels, I can figure out whether certain components are correlated with those labels. So in fact, we do this in other data sets where we don't use this metadata as supervision or as a prior in any sort of way, but we downstream use it to be able to figure out what's going on, what those patterns represent. But this is just the way that's worked for me. I'm not saying it's the right way. In fact, I really love thinking about using this information within the model. I think that's a really cool approach too.
We have a second question from Sebastian.
Hi. Hi. Yeah, I wanted to ask a question about these images that you had of the tissue. So it seems to me you didn't have that many images. So how did you make sure that the autoencoder learned something useful about the data instead of just memorizing it, which could happen with neural networks?
Yeah, also a very good question. So so it's true that we only had a handful of images when you're actually looking at it overall. I should say two things. First of all, the number of images we have that are unpaired is gigantic. They literally had to reinforce a floor at Memorial Sloan Kettering because there were so many of these images sitting as slides in that room. There's millions of them everywhere. So we can definitely do any sort of pre-training we want, and we do. The flip side is for the end-to-end version that I told you about, we actually have about 10 times as many. So we have, I think, 27,000 images now, which ends up being a fairly nice sweet spot for this. These images are really, really high resolution. And again, like I said, we also take patches and rotate and scale and things like that. So that ends up, sort of making that number be much larger than actually the small numbers that I showed you.
Excellent. Okay, so I think that we'll save remaining questions for the panel discussion, which is going to happen later on today. And I'll hand it off to Michael and say thanks again to Barbara. That was for a very great talk.
Thanks.
Thanks, Barbara. That was great. So my job now is to introduce Sendhil Mullainathan, who is going to be given the next talk. So Sendhil is an economist at the University of Chicago with really tremendously broad interests, and more recently in using machine learning to do data analysis, of a wide range of topics. Today, he's going to tell us about some of his work in computational medicine. He, just to sort of give an idea of his breadth, about a month ago, he wrote with his colleagues a fascinating op-ed in the New York Times about measuring COVID risk, which I recommend everyone. Maybe I'll put it in the chat. But we're really, happy to have Sendhil here, who will provide, a different, and I hope complementary perspective on these questions.
I wanted to talk today with you about using machine learning for scientific discovery. I'll be discussing primarily one paper, and that's joint work with Emma Pearson, David Culler, Jure Leskovich, and Ziad Obermeyer. So let me dive right in. Of course, when you think about science, I'm sure the first thing most of you think about is The Wizard of Oz. The Wizard of Oz is a movie that I hope most of you have, if not seen, at least heard of. Though as I get older, I'm realizing these references get more dated. I tried this in one of my classes, and let's just say the majority of people had only a dim understanding of The Wizard of Oz, but hopefully you're more savvy about culture than they were. So The Wizard of Oz was not just a movie, it was also a book. And Frank Baum, who wrote the book, actually loved Oz so much that he wrote another book, and another, and another, and he wrote a lot of books. In fact, he was so enjoyed and felt so much affinity towards the world of Oz that he had created. He wanted to make sure that he survived even after he was dead. So he tapped another author, who we'll describe in a little bit, Ruth Plumley Thompson, to carry on his tradition. And she went on to write even more Oz books. So this is one of those situations where if someone says to you, you've seen the movie, but have you read the book? Don't read the book. There are a lot of them. So the interesting thing about this, though, from the point of view of the talk I'm going to give, is actually revolves around this book, The Royal Book of Oz. The Royal Book of Oz was written just around when Baum died. It is the last book that Baum wrote. Or is it the first book that Ruth Plumley Thompson wrote? In fact, if you look at it, there are covers that credit Baum and not Thompson, and covers that credit Thompson and not Baum. There are covers that credit both. They're sort of splitting the baby. Because it's unclear who actually wrote this. In the world of literary analysis, while this is not a big issue at the level of was Shakespeare one person, it's a minor issue. And it's an issue, though, that many people have talked about and studied. And the way they would have studied it is by using their expertise of language, how authors use language, how they tend to write, their quirks, to make sense of, well, let's do a close reading of these books and figure it out. Now, this issue was in doubt for quite some time. And one of the papers that discussed this issue, which is particularly relevant for this conference and for what you all do, was not really a typical literary paper. It was a paper that was published in a journal called Chance, actually kind of an interesting journal that deals with statistics and probability. And you'll see that they asked the question, who wrote the 15th book of Oz? But the subtitle tells you this is more up your alley than up a literary alley, an application of multivariate analysis. In fact, I actually like the other thing, the teaser in front. Does only the Oz himself know who wrote the Royal Book of Oz? So what the author here had done was to answer this question, was to take a statistical tool to it. And let me tell you what he did. So his method was fairly simple. Remember, this was a paper back in 2003. So it was, by its time, pretty advanced, though it may look simplistic in today's time. So what he did was he took every book of Oz, both Baum's and Thompson's, everything, and broke up each book into 5,000 word blocks. Then for each block, he vectorized it. So instead of having this, he used bag of words to sort of do a word count and to get a vector for each word. What is its frequency in that block? So a block is now simply a vector. And I'm going to come back to this in a little bit, but he doesn't use that full vector. He focuses on the subvector that looks at the 50 most common function words. I'll say what that means in a second, but basically now each block of text in this book is turned into a vector of length 50. Then he does something really simple. He performs PCA on these blocks, and this is what he gets. This is astonishing. Here are the first two principal components, PCA on that vectorized blocks. What's amazing, remember, this, we've not looked at the labels Baum and Thompson at all. This is just PCA. Yet when you look at where Baum falls and where Thompson falls, without ever having used that information, everything with Thompson is on the right, everything with Baum is on the left. This is a perfect classifier built without actually having labels. Principal components really revealed what was going on. Which raises the obvious question. On here is not any of the blocks from the 15th contested Book of Oz. So where will the blocks from the Royal Book of Oz fall? In fact, they fall on the left. It looks like that's the hearts here. I won't discuss the clovers, the clubs, but the hearts here are now we've added in the 5,000 block texts from the Royal Book of Oz. And you can see every one of them falls on the left, and every one of them belongs to Thompson. This is pretty compelling evidence as to who wrote the book. By itself, it's a remarkable way to look at data to unearth something that experts might not have known was there. But the story is richer than that, and let me tell you why. Remember I said the input vector he used was the 50 most common function words. For those of you who don't know, function words are different from content words. Most of us, when we think about language, we focus on the content words we use. Nouns, verbs, adjectives, adverbs. But sentences aren't just formed from those. We have these little connection things like the, a, and, things that tie nouns together, things that make whole sentences, conjunctions, pronouns. It's often the case that we think in content words, and there's a great book by James Pennebaker called The Secret Life of Pronouns that talks in great depth about this. But in fact, function words actually, though they're ignored largely by the human mind, there's some amazing studies that show people don't even remember what function words are there. They are actually a part of language. And remember, the Oz analysis I just showed you threw away all the content words and looked at only the 50 most common function words. So that in making its analysis, when it looked at words, it only looked at these words. In other words, it was distinguishing Plumlee and Thompson perfectly, without ever looking at any of the words that a literary analyst would have looked at. This is nothing. To see how weird this looks, here's a paragraph I picked from The World of Oz. In fact, I think it's the first paragraph from the first book. Here's what the algorithm used from that paragraph. Even this isn't as much as it used. This is more than it saw. Why? Because these were all vectorized. The word order was jumbled. Everything was taken out. It's just a vector of frequency of these words. But you can see all of the semantic information appears to be stripped away. And yet, using just this, we could see who wrote what Plumlee wrote, what Thompson wrote. which is why I like this study. To me, it's a prototypical, early, fun example of how algorithms can be used to aid in scientific discovery. Science here is what the literary analysts were engaged in. They were trying to make sense of the world around them. They were experts. Yet it found an empirical fact that those experts had no clear way of seeing themselves. That's what I mean by discovery. You can really see they had no way of seeing because it focused on function words, something we barely have an understanding of. This is not a one-off. That's also why I like to study. Even in the narrow space of language, there are many studies now that have done stylometrics, things like this. Who wrote what? Was there more than one author, as in the case of William Shakespeare? But it's also not a one-off in the world of language and just focusing on author attribution. People have used language to predict a set of things that experts didn't know could be predicted. All of these have the feature of finding unexpected signal. And in fact, I like this study a lot because the fact that the algorithm was so simple by modern standards, it's PCA, look at the first two components, only reinforces the potential for data-driven, algorithmically aided scientific discovery. So I think of this as a fun study with a hint of a lesson. Algorithms can find signal in data we are unaware of, even in domains where we have lots of expertise. And to me, it raises the possibility that what we are dealing with is a complementary scientific tool. What I mean by complementary is that it's not meant to unearth and replace existing tools. It's meant to fit into a part of the science pipeline. And pipeline is important because there are many components of doing science. We often focus on the one part of the scientific method, experiments. But the experiment is actually just in this pipeline and is an essential piece. But in the pipeline, there's something that comes before. What am I doing the experiment on? Where am I getting my plausible hypotheses? We leave that to chance or the mind or creativity or genius. But by finding unexpected signal, algorithms give us a data-driven way to formulate novel hypotheses, things we hadn't even considered. That function words are the key differentiator in stylometrics and deciding who writes what. That the signature of an author is not the flourish that they use in describing the trees, but instead the hidden ways in which they use to, the, a, and. That's a pretty unexpected hypothesis. It's a hypothesis we can now dive into. These type of early studies I gave on the Oz, they're not meant to answer the question in its full. Science never answers a big, meaty question at one go, but they're meant to probe and generate whole new ways of looking at the world that we can then probe with other experiments. And that's what I think is the goal of ML as a tool for scientific discovery, to help us see things we didn't see otherwise, to generate interesting hypotheses that can be explored in greater detail using experiments and other data. So today I'm going to give one concrete example of that activity in a domain that many of you may think of as science, medicine. And I want to walk through that in great detail. Then I want to discuss lessons from it for how we apply algorithms, for how should we go ahead and produce more examples like this. And finally, I'll discuss what it means for the kind of tools that we need to develop to do this activity. Some of you watching this are in the business of creating the kinds of machine learning and data tools that are usable in many cases. I want to encourage you to think about, wait, if we're going to use machine learning for this new purpose, what new tools are needed? I'll try to give some guidance on that. Okay, let me start with a concrete example. The concrete example is a paper where the lead author is Emma Pearson. She's a graduate student at Stanford who just graduated and will be joining MSR and Cornell Tech after that. And it's joint, as I said, with these co-authors earlier. So let me tell you the topic we were interested in. Pain amongst people, everyday pain as experienced in common, not extreme pain due to some injury, but everyday pain is actually quite common. In fact, it's so common that if you look at people who are unable to work, it's amongst the most common causes of disability, reasons for not working. Arthritis and back or spine problems are the two biggest. In fact, of this, osteoarthritis, joint problems, which I'll describe in a second, are the most common. Osteoarthritis is the most common joint disorder. Osteoarthritis is, for those of you who don't know, the left here is a healthy knee. There's a lot of buffering between the two bones here as they come into contact. There's a meniscus, there's this cartilage. It just keeps the bones from hitting each other. Osteoarthritis is a set of clear degradation of that situation where there might be some cartilage loss, the joints might be too closer, and then suddenly the bones are bumping against each other, which is incredibly painful. The way we go about measuring osteoarthritis, I want to get into that for a second. The first way we have of understanding that there's a problem is someone says, my knee hurts. Okay, well, we run them through a little bit of a survey, which is aggregated into a single KOOS pain score. I'm going to focus on this pain score today because this is our measure of pain. Now, of course, not all pain necessarily reflects osteoarthritis. So I'll come back to that in a second. The key thing I want to talk about with pain, though, is I want to talk about a striking fact that in the United States, when you look across people, let's look at Black patients versus other patients. Disadvantaged patients, in this case, Black versus others, experience a lot more pain. The gap is about 10.6 units of the KOOS score, which I'll talk through in a second. The KOOS score goes in the wrong direction. I don't know why it's defined this way, but lower means more pain. That'll matter in some things you see later. But by race, on average, there's 10.6 fewer points. Just so that you know what that means, that's 2/3 of an entire standard deviation of the population. That's how big the gap is. It's huge between Blacks and whites on average. Now, why is there this pain gap? One view is there's something wrong inside their knees. Physical ailments are more extreme amongst disadvantaged populations, perhaps because they've had some history of heavy physical labor, perhaps because they've had certain things that were undertreated, that there are problems inside their knees that are more severe. A different perspective is there's something outside their knees. What does that mean? Well, for the same level of injury in the knee, it's possible that being disadvantaged means that you experience that pain more heavily. For example, if you've got a lot going on in your life and a lot of life stress, the same physical pain leads to more psychological pain. And this is true for a variety of reasons. Maybe you just have less access to pain medication. So when you ask people how much pain do you feel, we're seeing net of treatment. Maybe disadvantaged people have less social support to help them. So these are the two ways of thinking about this. How would we differentiate between these? Whether there's this pain difference is due to something different in the knee. Well, luckily, we don't have just the pain to work with. We have the X-ray and we have expertise. We can look in that X-ray and find all the signs of osteoarthritis. So we can apply clinical judgment, look for everything we know, and then say, this is how severe it is. There's a known protocol for this, which is called the Kalgren-Lawrence grade, which I'll call the KL grade. So now we have the ability to say, yes, you said you have pain, but let's look inside your knee. When we do that, we find that yes, disadvantaged patients experience more pain, but let's control for the extent of KL grade. Let's control for the extent of knee problems. And when we do that, what we find is not much changes. On the x-axis now is how bad the knee is according to clinician judgment, according to expertise. And the y-axis is reported pain for everyone in that cell. And now you see that even conditioning on KL grade, the difference between other patients and Black patients is basically still quite noteworthy. In fact, it's so big, basically, if you look on the table here, we've done very little to explain the original gap. We've explained 9% of it, 8% of it, almost nothing. Controlling for severity simply doesn't narrow the pain gap. Now, this is not our finding. This is a common finding in the literature, and people have used this finding to say, wow, we really have a problem in our society, a pain problem. We have to figure out what to do about this excess of pain, but it's a pain problem. It's something about how we cope with pain. But is it though? Does this really settle the matter? Does the exercise that we've done here, which is conditioned on what is experts know and then look at the residual, does that really settle it? Well, keep in mind, medical knowledge is not a complete thing. It's still in flux. It's why we're still doing research and still doing science. We're very far from understanding what knee damage looks like. And we know at some fundamental level that we're not that, it's not like we have a great predictor. For example, if we look at pain, KL grade doesn't explain much of pain. Now you could say almost all pain is in your head. Or you could look at this and say, maybe there's some stuff we don't know yet. So suppose there is something in the knees we don't know about. Then you can see that the results I just told you are differently interpreted. They don't say, oh, it's not inside the knees, it's something else. No, they say, maybe there is something inside the knees we should find out about. Are there overlooked features? How would we go about answering this question? Here's why it matters how you answer this question. If we believed most pain like this for the disadvantage was something that was about pain and not something in their knees, we would treat them with drugs, pain relief drugs. If we believed there was something in their knee, we would look to fix the thing in their knee. Now, that could mean surgical techniques we're using today, but it could also mean coming up with new surgical techniques. So this is not a let's do optimal treatment assignment. This is science. So it's about, wow, there's something in their knee. Even if we don't know how to treat it, well, let's go find things to treat it. So at some fundamental level, the distinction from a science point of view is huge because it tells us where to look for treatments and hypotheses. So how would I use ML for discovery in this context? How could I take this hypothesis? Yes, we know that there is, given our current expertise, it doesn't look like there's anything different between Black and white patients. But we don't want to just assume expert knowledge is perfect. How would we figure out if there's something else inside the knee? Well, good news. The type of things that you all have been working on is perfect for this problem. Let's train a convolutional net. Specifically, let's give it the X-ray. And then let's say, given the X-ray, here's the label. It's pain. You go ahead. You look in the X-ray to decide what pain, to make a prediction of pain. The key here, which I'm going to come back to, is two things. The algorithm only sees the X-rays. It's not going to be much of a surprise if it can predict pain from a bunch of other data. The interesting thing is there something inside the knee that denotes pain? So that's going to be important. The second thing is I'm predicting pain. I'm not predicting expert judgment. And I'll come back to those two issues and really call them out. But the exercise here is give the algorithm an X-ray, predict pain. Here's what our data set looks like. We had a training and development data set of about 25,000 X-rays, and we had this lockbox that we didn't touch until the very end, which had 11,000 X-rays. And you can read about those details in the paper you want. When we do this, what we find is that this algorithm-defined measure from the X-ray goes a significant way towards explaining the pain gap. Whereas KLG only explained 9%, we can now explain about 43% of the pain gap. And to be honest, this is not a big data set by convolutional net standards. So in principle, if we had better data, by better I mean more data, we might explain much more than we see here. But the implication is striking, and you can see here on the left was the original graph that I showed you. On the right is what happens now if I rank by the algorithm's prediction. You can see the gap really diminishes. The implication is there is overlooked signal in the X-ray. Things that we didn't know was there given our current state of knowledge, but appears to be there and appears to explain disadvantaged patients' higher reporting of pain. Think how striking this is from a human point of view. It is in their knees. It is what people have been telling us all along. Whereas currently we sent them home and saying, oh, we looked in your knees, there's nothing there. Must be something, else going on pain-wise for you. Here's some pain relievers. No, it's exactly what people have been telling us. Something humbling about this is that even the best part of science is that we always take expertise as a in-progress situation. Here we had expertise and a human cry for help. And we've seen that algorithms can help us listen to people a little better and be more humble about our expertise. But before concluding all that, let me just walk through a few robustness checks, because as you know, algorithms can also be quite misleading and fragile. So one thing you might be worried about is it just finding some weird artifact? And I'll give you one piece of evidence that it's not. Here's some salience maps that shows you it's exactly looking at the location of osteoarthritis. The other thing you might be worried about is that it might be, okay, you might think, okay, but how do we know it's not including something else, even if it's looking at that particular location? So what we did is we took a bunch of controls and we said, what if we added all of these controls? Does that change how much this algorithmic measure predicts pain? And it turns out it has no effect on that. The second thing you might be worried about is, well, maybe the algorithm is somehow reconstructing race. And again, same exercise, control for those things, nothing changes. But I want to make a pause here. We're used to this problem in one context. In the case of scientific discovery, suppose we did this exercise and we had found, wow, the algorithm is reconstructing race. Is that bad? That's bad news. But is it bad news? It would be a pretty interesting discovery in and of itself if it turned out that race is reconstructible from just an X-ray. It certainly is not something that anyone with a clinical understanding would be able to say, oh yeah, that's obvious. This is just to remind us that in the world of discovery, unexpected correlations are what we're looking for. They're not bugs. They're interesting. Now, in this case, as you would have guessed, you can't reconstruct race from X-ray. That's not what's going on. We also deal with a bunch of other counterfactuals. I don't have time to get into it, but let's just pause it for a minute that the algorithm has in fact found new signal. The implication of that is pretty striking, as I've described already. The algorithmic aided discovery showed us there is something there. And this is, of course, the beginning of the story. As I said, it's not by any means the end. We now need to understand, what is it? Do existing treatments work? Do we need new ones? It's the beginning of a long process of interesting science. That was my concrete example. Let me give you an example. Let me now discuss lessons from that example and how we can understand the key ingredients needed to write papers such as these. I think there are two key ingredients, and they are different from what you're used to, so I want to call them out. The first involves the choice of labels. Remember, we have two looks at the problem, the pain score and the KL grade. We chose to predict pain score, but almost everybody working in this literature would have chosen to predict KL grade. We tend to take X-rays, take judgments, radiologists, whoever, making judgments of these X-rays, and then make that the label and have the algorithm predict that. We do that for two reasons. First, because we're normally not in the goal point of discovery, we're in the point of automation. We want the algorithms to do as well as expert judgment. But we also do that because those labels are easy to acquire. Every time you go and get a data set, this is a CT scan, it typically comes with an expert judgment. So there you have your data on the left, there you have your label on the right. In contrast, pain score is very hard to acquire. There's a reason our data set is not immense. It's because this was carefully collected. Since hospitals don't naturally have pain score. This was from the Osteoarthritis Initiative, where they'd actually collected X-rays on people and did a sampling and actually asked people for pain score. As a result, it's a gold standard data set, but it was very hard to acquire. So part of working on ML for scientific discovery is actually working on acquiring these data sets that have these hard to acquire labels that are about ground truth, that are about the core thing you want to predict rather than expert judgment. Automation is for expert judgment. Trying to get to some closer to ground truth is what we need for discovery. But they're not just hard to acquire, they are hard to define. Let me explain what I mean. This is from a project that Ziad Obermeyer and I did a couple of years ago. We were putting together an algorithm to predict stroke risk in the emergency room. And the idea is pretty simple. If someone comes into the emergency room, if you could look at them and say, oh, this is a person with high stroke risk, then you could triage them quickly and fast track it, because diagnosing a stroke early is really important. And we thought, let's use all the patient's electronic health records before they enter the emergency room to predict whether they have a stroke. Let's assume for a minute that what we're predicting is the ground truth label, had stroke or not. There's no problem there for now. What we did is we did this for 80,000 primary care patients, and the result was a pretty effective predictor. So when we looked at that predictor, we said, oh, well, what is the algorithm looking at? The first two things it's looking at, the two top predictors, made a lot of sense to us. First, the obvious one, have you ever had a stroke before? Second, do you have any other history of cardiovascular disease? Because a stroke involves a blockage, vascular blockage, so that it would be a pretty straightforward thing to see. Okay, so let's now look at the next four. Those were weird. Colon cancers, what did these have to do? So this one says the person came in for colon cancer screening. What does that have to do with the stroke? This person said they were evaluated for some minor injury. Each of these is weird. They don't really have anything to do with stroke. Wow, maybe we just made some amazing medical discoveries. Not one, but four. 4 things related to stroke that we never knew about. Probably not. What happened is that we've defined ground truth badly. Yes, we measured stroke correctly, but we didn't measure stroke in everybody. We measured stroke plus whether they actually showed up at the emergency room. Remember, that's how the data is set up. So we've actually predicted stroke plus what we might call utilization, propensity to show up at the emergency room. Because someone in our data shows up as having a stroke if they had a stroke, noticed the symptoms and worried about it, showed up at the emergency room. So Delta 1 and Delta 2 and anything else that kept them from the emergency room played a key role. So we haven't measured Y star, we measured Y star plus delta. Now, why does that imply that we have a problem? Well, delta is utilization. But guess what colon cancer screening is? It's a measure of utilization. This is a measure of utilization. Each of these is a measure of utilization. So we measured Y star plus utilization, and we put things that are proxies for utilization on the right. So guess what predicts utilization? Utilization. So this mismeasurement of Y shows you how subtle and complicated it is to define the right label. You want the true Y star label. Because in general, if what we have is measure Y star plus delta, and when we go to predict Y, we'll see we'll get some signal and we'll get some confounders. Okay. This is to summarize that in general, medical predictions, for example, are built up from human decisions. And so when we do that, what we do on the left is to use easily available data, potentially with confounders, to make predictions. That's not going to get us to science. To contrast, imagine I built a self-driving car that braked whenever the driver braked. That gets us to driving like a human. Science is understanding how do I convert distances and a bunch of other variables from what's between me and the next car for when you should brake, for what is the right time to brake. That's looking at ground truth. And what I want to make as the first distinction of trying to do ML for scientific discovery is working hard to get labels that are ground truth and unconfounded, not human judgment. And when you go back and look at a bunch of papers, you'll see most of what we do in ML is for automation purposes. For automation purposes, you want to use the ground truth as expert judgment, but not for scientific discovery. Let me tell you the second key to making this work, which was it was important that we blinded the algorithm. Remember, we're looking to say, wow, I thought we had found everything we could in the X-ray. I want to see, is there anything else in the X-ray for predicting pain score? For that exercise, you want to look just in the X-ray. Whereas most ML exercises would be, oh, if you're predicting pain, throw in everything you have. But suddenly that would make the exercise meaningless. The goal here is not to predict as well as we could. The goal here is to isolate signal in places that are unexpected, which often means focusing the algorithm on a subset of the data we have. We had all this other data. We just chose not to use it because we're looking to see where there might be a new fact. So the keys to discovery: invest heavily in creating meaningful labels and focus the algorithm only on inputs where signal will be a surprise. Now let me conclude this talk by discussing what all this means for tool development. Many of you work on building interesting machine learning tools. I hope you will get interested in applying those tools to these areas. And in the process, I think you'll find that you'll have to modify your tools. Here are 2 ways I've found that we need new tools. The first goes back to the question of what is the algorithm seeing, which is, of course, the question of interpretability. I'm not going to say, oh, we've got the answer. We don't have the answer. There's a lot of work needed here. We've clearly found an interesting signal. Now we'd like to know what the hell that signal is. That's the focus of our work. But I want to just give a highlight of what interpretability for scientific discovery means. In general, interpretability is about creating human understanding of an algorithm. But typically, when we do that, we take the human understanding as the ground truth. We use human understanding to debug the algorithm, which makes a lot of sense when we're automating, because the algorithm should align with our understanding. And if it does something weird, we know it's doing something wrong. So for example, remember when I showed you the salient check, I was like, oh, good, it's looking at the knee. That was kind of using the fact that we tend to use salience maps to debug. But why should we use it to debug? For discovery, that's not what we want to do. Imagine it looked up here in the femur. We'd be like, wait a second, it's looking there? That's interesting. Why is it looking there? I wouldn't have thought to look there. In discovery, we don't want to impose such strong priors. We want the algorithm to tell us where it's looking because it's found something that we didn't know about. In the Oz example, we wouldn't have wanted to say, that's weird, the algorithm is looking at function words. Something must be wrong because we know that we should look at these other words. No, we'd say, weird. Function words, I didn't even know that signal could be found there. Interesting. So we are starting in interpretability for scientific discovery by wanting to create ways to improve human understanding. And that's a very different approach to creating interpretability tools. It leads to a set of things that we now will need to ask, how do we create such tools? I don't think it's by taking off the shelf interpretability tools. The second thing besides interpretability is actually relates to algorithm performance. I should tell you I never reported it because I wanted to save it to the end. The AUC of our algorithm is 0.69. Is that big? Is that small? Doesn't look like an AUC of 1. One thing we have to do is to recalibrate our intuitions around what AUC we expect. When you are doing automation, you're expecting an AUC of 1 because you're trying to do what the human did. But in goals of scientific discovery, your benchmark is, oh, humans don't think there's signal there. So an AUC of 0.5 is what we're going in with. So anything above 0.5 is remarkable because we found some signal, something we didn't expect. And so discovery means looking in places where we don't expect any signal at all and finding any signal as a surprise. Yet our tools have largely been focused on getting us from 0.99 to 0.995 or 0.9 to 0.98. We're focused at one end of AUC. We need tools focused at the other end. Just to make it, to give an example, for those of you who read the literature on double descent in neural networks, it's not implausible to, as many people have been saying, that double descent appears to be a feature of very high AUC problems where there's perfect fit. That's very important, very interesting. Now what happens at the opposite extreme in very high noise problems? What properties do these algorithms have and how do we go about tuning them and doing other things? Let me conclude this talk by saying there are many more cases like knee pain. In my own work, I've encountered dozens of them, and that's just me and my collaborators. I'm sure with the creativity that you all have, there are many, many, many more cases. That's in social science and medical science, which I know about, but surely, as other people in this workshop have talked about, it's true of many other sciences. And I think there are some exciting discoveries to be made, but it requires a different relationship between the analyst and domain expertise. Rather than asking ourselves, oh, okay, so domain experts give us the ground truth labels. That's not the relation of expertise. Rather than thinking domain experts tell us the mechanisms at play, again, that's not what we need for discovery, because we're not trying to go in imposing the prior of the domain expert. Instead, we need the domain experts to tell us a different part of their prior, to help us understand what choice of labels do you think constitutes a ground truth that you think your expertise is chasing towards. What kind of input data would you be surprised to see signal in for that ground truth? So the domain expert needs to help craft the experiment, the data frame, and the label, etc. And in that way, by doing that, I think we can combine all of these things to have sort of a very fundamentally new approach to the way we do science, a complementary one. And to do that is going to require a set of new tools and new approaches to the problem. Thank you.
Thank you. Hi, everyone. Sendhil is with us here, live to take questions. But I think we will wait for another 2 or 3 minutes so people can join us from the ICML website or wherever else they're watching. Okay. Welcome back, everyone. And yeah, I don't see questions on YouTube, so I'm going to go ahead and ask some questions of my own, which, yeah, I really enjoyed Sendhil's talk earlier. So, oh wait, actually also, let me just check the chat on the website. Okay, so nothing on that chat for Sendhil. Okay, so my first question to Sendhil was going to be a specific thing with regard to the model. I think so. The thing is, okay, one of the things that you talked about is trying to do explanation when the AUC was a little low. So I think that's something that I think we might want to come back during the panel and discuss about it. But I'm just wondering, so like what kind of explanation methods did you apply specific now? But I think we can come to a generalized version of it later in the panel also.
Yeah, it's one of those things. So what we did was a salience method to just kind of get a sense of where the algorithm was looking. And I'd say there were two bottlenecks. I think you identified one of them, which we can come back to in the bigger panel, but let me try to restate it. So the first bottleneck was, if you imagine you have a predictor, so let's say an f of x, typically we interpret that predictor f of x. But for scientific discovery, we're not interested in interpreting the predictor. We're interested in interpreting the part of the predictor that is signal. So if we're predicting a y, how do we know when we interpret f, we're actually interpreting the part of f that's meaningfully predicting y versus the rest? And that's gets to be a bigger and bigger problem the lower the AUC. And in most of these scientific discovery environments, you don't expect very high AUCs. So trying to interpret the signal and not the noise, I don't think we had any. We kind of ran across the problem, wasn't really sure how to do it, but that, that was left unsaid. The other part that was harder was, I think when you're trying to debug an algorithm that you're trying to get to be near a perfect AUC, it's pretty easy to take a salience method and see what you would be doing, which is you would be flipping through instance after instance and then saying, okay, does this look reasonable? Does this look reasonable? And you're kind of just like going through one after the other. And there it's easy to do because it's almost like looking through something, looking for a bug or an oddity or something that shouldn't be. But here, since you're trying to get a sense overall, where is the algorithm looking? It was a little bit harder to kind of look across all of these and notice any central tendencies, because you'd have to register each X-ray to each thing, somehow make it look like the same point in each X-ray, even though they're each very different. So I think those two issues, the ability of sort of aggregating interpretations is the second one, and the first one is the separating signal from noise. Those were both things that we didn't have a good answer to, but we obviously needed an answer to that in order to be able to do something.
Thanks, Sendhil. Just gonna go and check back. Okay, so the other question that I have is with regard to this model. I think there are a couple of interesting points that you made. 1 second, I think I'm getting some echo. Sorry. The couple of interesting, there were a couple of other interesting points that were made with regard to this model that you were training. So I think you mentioned one interesting thing is doing it on the right labels in some sense. So here the right labels were actually what the humans felt, in the sense of like, so in the case of like braking, it was, okay, it should actually be the distance that you would drive before you brake. And so what is the appropriate distance that you should brake for, as opposed to doing this with, let's say some other human prior. Like if you asked humans what they would do, it's similar to what the kind of labels that people pick for a task might be different from, there are a different choice of labels that you can choose from in order to train your model. And one of the points that you mentioned was we should examine a lot of these labels in some context. And one question that I had with regard to this was, if we take it from multiple human actions, would we be sort of like getting an approximate value towards the real goal, or does it still depend a lot on both the type of label that we're looking for and like how we break this thing, break it in the sense of like to what granularity we are measuring it, as opposed to just picking like some boxes. So let's say severity for some diseases, you just have like the quiz scores. Maybe these are just like integer values, but humans might only be able to rate integers, but there could also be a granularity scale that's there. But I guess there are two parts to this question. One is it enough if we get from enough humans? Would we get to the right whatever is the right label that's supposed to be for the task, would we get to that? The other is, yeah, does the granularity of the label matter? Are humans good at it versus should we just take something?
Yeah, I think that your intuition about let's use many labels to kind of aggregate out truth, kind of a wisdom of the crowds thing. I think there's a deeper issue here than the idiosyncratic measurement error of each human. The deeper thing here is that we almost want to think of human reports as their judgments of truth. And in doing scientific discovery, part of what we want to do is to actually ask, could we measure that truth directly? Does that make sense? So for example, it's the difference in a context like a physician examines you and says, oh, you've had a heart attack. Now we could have 100 physicians examine you, but that's physician judgment, versus a blood test that would show certain molecules which are indicative of heart attack. The human judgment is our representation of clinical expertise. The blood test is nature speaking to us. It's sort of a ground truth. And so I think what's hard in this literature, and it's what's great that you're organizing this workshop, is we don't even have crisp language for talking about the difference between a human intermediated label and a ground truth label. Now this problem becomes a little more complicated when the ground truth is itself a human phenomena like pain. So weirdly, I find myself in the position of saying the patient reported pain is ground truth. The clinician judgment of arthritis is human intermediated, even though both of them have humans inside, it has humans in them. So, but should I, maybe I should move to Rebecca's question?
Yes, let me, yeah, okay. So let me actually read out Rebecca's question. I'm not sure if, or I can also maybe, can we get Rebecca to be unmuted? Oh, actually, maybe I can do that. Yeah, Rebecca, if you want to ask your question, I'm going to allow you to talk. Yeah.
Hi, can you hear me?
Yeah,
yes.
Hi, Rebecca.
Hi, thanks so much for your talk. It was very interesting. I was wondering, you kind of talked about this kind of tricky balance between checking the robustness of your model and also avoiding imposing prior assumptions when you're doing that checking for the robustness. So I was wondering if you had any recommendations for how you might, in a practical sense, distinguish what would be an erroneous model from a model that allows you to make real discoveries.
It's a great question. And I think if you don't mind me opining a little bit, I'll just step back 1 minute. I think it has to do with the fundamental, I think the thing I like a lot about computer scientists is they have a humility about how they work with experts. And I think that in scientific discovery, it's about reorienting the humility of how you work with experts. So I think for me, what's been great in working with medical doctors is in version 1, when we're not doing scientific discovery, we view the expert as the source of the label, as the source of the prior that we're trying to match. I think there's just requires a different nuanced relationship with the expert and for discovery. And it's exactly at your question. So we don't want to impose prior assumptions of what's right or wrong, but we want to impose prior assumptions of where the artifacts might be. Like, this is just a confounder. This has nothing to do with the phenomena that I'm going into. So your question actually ties wonderfully together with Shubha's question, which is, experts are awesome at laying out what would be a good ground truth label. And they're awesome at saying, if you can use this input to predict this ground truth label, that would constitute a genuine surprise. And I think expertise is needed at that level because there are so many things that purport to make a discovery, but you show it to an expert and they say, well, there are plenty of reasons X could predict Y, including measurement error. And so I think part of what will be interesting here is rechanging our conversations with experts to be able to answer the two questions. Yes, that's judgment, but what's a good ground truth label? And the reality is in most contexts, there isn't a perfect ground truth label. There's something with some noise and some error so that the expert can help think about that. And the expert can help think about, like, what would you predict? What input predicting that ground truth label would truly constitute a surprise given where we are, as opposed to a confound or an oddity.
Great, thank you.
Thank you.
I'm next going to unmute William, who can ask his question, which is also quite related to everyone's. Yes. William, you're on next.
Hello?
Yes,
Hi,
we can
William.
hear you.
It's a great talk, by the way.
I think.
I'm just wondering whether or not you made a comment about the difference in sort of high AUC situations and low AUC situations where your baseline is 0.5. I'm just wondering whether or not this is a general issue where you have high AUC 0.9, 0.98. These situations are kind of easy tasks. So you can't discover anything from something so easy. You need a very difficult task, a difficult challenge where 0.5, 0.6 is state of the art for discovery to actually take place. Do you agree with this?
Completely agree. Let me actually build on top of what you're saying. When I talk to grad students about these scientific discovery tasks, they're like, but how do you even know if there's anything going to be there? Why do you even think there's going to be anything there? Like, there's an element of risk that I think is implicit in your question about easy versus hard tasks. These are risky tasks because you go into it saying, we don't know if there's signal. So it's not just the magnitude of the signal, we just don't even know if it's there. And so I think what my reading of the field is that when we had, we thought what we're now calling easy tasks were inherently hard. And there was a period where a lot of great work was done to make them look easier. That is, now we're able to hit AUCs of 0.98, 0.99. So I think we have in mind, like, that's very, very hard. But as that becomes routinized, I think what you're saying, William, is exactly right. Now we have these other tasks that they're hard in two ways. We're going to get low AUCs, and there's just risk. Like, science always has risk. You have to look in a certain place, you have to make a guess, and you have to hope the data complies.
Okay, it looks like that was it. And it also seems like we're back on schedule, on time, and we should go watch Arun's talk. I think the process is going to be the same as the slides and the video, and you can also watch it on YouTube where it'll play the slides only, but you'll also hear the audio. So cool. We will come back here in, I think Arun's talk finishes in half an hour, so we'll be back in 30 minutes or so. But yeah, thank you.
Thanks for the question.
Hello, I'm Arun from Google Research. Welcome to my talk. Today I'll be talking about ML-driven scientific discovery. I'll start with a little bit of background and motivation for a slice of our work here in Google Research, particularly pertaining to medical imaging. As is always the case, the work I'm going to present here is a joint effort from a team of people in Google Research and Google Health. We believe we have a lot of scientific discoveries waiting to be made, driven primarily by interpretable machine learning techniques. And we'll explain with a few examples as to why we think this is the case. One of the first problems we worked on was the diagnosing of diabetic retinopathy. It affects a subset of people with diabetes that can ultimately lead to a loss of vision. It's also the fastest growing cause of blindness in the world. So it's an important problem to work on. In this work, we obtained a very large data set working with several eye hospitals in India and also with IPACs. It consists of 130,000 images. And we trained a model to predict severity of the diabetic retinopathy, as well as referability and image quality and other attributes. One thing we learned early on was that ophthalmologists were quite inconsistent in their grades. As you can see in the picture, each row here represents the grades given by several ophthalmologists for a single image. And this may not come as surprising to many of you who have worked with medical data, but for ML researchers like us, this was an eye-opener. As you can see, a small percentage of images, there seems to be an agreement, but for most of them, there's a wide varying levels of grades given by the ophthalmologist. So the intra and intermediate agreement was somewhere between 60 and 70%. We wanted to reduce the effort of this effect of this variance on our model performance. Hence, we labeled each image at the test set about 8 times, and we realized that we needed to label the test set much more than the training set. When we published the work in 2016, we compared the work with performance with respect to the ophthalmologist. And the subsequent work, we've also compared this with retinal specialists who are much more accurate. And then we often report the performance of these models in terms of area under the curve, or AUC, the sensitivity and specificity for referability of DR. And when we have multi-class classification, we also report a simple or a weighted kappa. Our first work got published in JAMA in 2016. It was well received when it came out and has been cited extensively. And recently, it was also selected as one of the top 10 articles of the decade by the editors of JAMA, which takes into account the viewership, the citations, and the importance of the work. So we were very happy with that, but that was just the beginning. We were also very focused to see this through to the end. For us, this work is not just a research in vacuum. Take India, for example, where there's a shortage of 127,000 eye doctors. As a result of this, there are systemic issues. And nearly 45% of the patients suffer vision loss before they even diagnosed. And this should never happen because this is completely preventable if they are diagnosed early. So we built a service that interfaces with the hospital's IT system, and we actually do these diagnoses live now with India. We refer the patients to retinal specialists for further examination if necessary. We partnered with 2 eye hospitals, one Shankara in Chennai and Arun in Madurai. We have also completed both retrospective and prospective studies using this approach in both India and Thailand. We have an initial pilot deployment of our system with partner hospitals in India. We're scaling them up for wider use now. We've also expanded our work to include several other eye diseases as well. We can identify glaucoma with higher sensitivity at the similar specificity compared to eye doctors. And we can also predict the progression of age-related macular degeneration better than our manual grades from the ophthalmologist. And there's much more need to be done in this space. We've also published a number of works which explain where these models find most of the signal from. For example, integrated gradients provides a per pixel attribution. Here, as you can see in the pictures, it places higher saliency on the lesions that it finds, which are critical to the prediction of DR severity. And these model explanations offer several key benefits. First is to gain the doctor's trust on the machine learning model we use to refer patients, because we don't want them to think this is a black box. And also, it helps us understand when our models fail and make an incorrect prediction. And the third thing we were wondering was whether this can help in assisting doctors in reducing false negatives. For example, one of the things that they tend to miss are microaneurysms, which are very small spots, which are hard to notice. And these are very early stages of the diabetic retinopathy disease. And early detection could be vital in treatment. So we wanted to ask ourselves a question whether the model explanations help improve doctors' accuracy. So in this work, we did a study with both ophthalmologists and retinal specialists, where we gave them 3 different modes of grading. One is the unassisted, which is the typical way they do it. And the second one is we give them the grades from the ML model. And the third way is to give them grades plus a saliency map from our model, which shows the regions to pay attention to. To be sure, we also split the images into several sets and rotated them among doctors, spaced out over several weeks to avoid any memorization. That's possible here. As you can see in the graph shown, general ophthalmologists, shown on the left bar of each graph, are worse than the model, unassisted. But they quickly catch up to the model once the grades are revealed to the general ophthalmologist. But then they don't seem to be making much gains beyond that. In fact, there's a small drop in accuracy as we show them saliency maps. Retinal specialists, on the other hand, were actually better than the model unassisted and had a moderate lift when we showed them both the grades and the grades plus heat map. So we believe there's a much more work to be done in trying to show these saliency maps to the doctors to help improve their performance. A parallel effort to disease diagnosis are the models trained to predict outcomes and novel attributes directly. Here is a sample of a set of papers from the team where we are able to predict, for example, cardiovascular risk, refractive error, and many other attributes directly training from retinal fundus imagery. A common theme among them is that they directly predict measurements that are obtained without human subjectiveness. So they might help us discover novel biomarkers, but that's not new. What is new is that we didn't have to hypothesize what to look for in these images. Their models are directly trained from pixels to outcomes, and there is no need for a human intervention while building the training or training the model. The first thing is the accuracy of these models surprised at first. But more importantly, this presented us with a unique opportunity to learn something about the human eye from these deep neural networks that were unknown to us before. For example, explaining how these models could predict age, blood pressure, or gender directly from pixels eliminates a key ingredient in most of the search for novel biomarkers. It is the human hypothesis to begin with. On the right, you can see several soft attention models that the model's prediction of saliency maps for each one of the targets that are predicted there. And many of the saliency maps are localized to main blood vessels, while a few are on the macula, optic disc, and so on. So we began to see eye as a non-invasive window into the physical health of the person. Take, for example, one particular paper on cardiovascular risk prediction. The model here was trained to predict a major adverse cardiovascular event called MACE in the next 5 years. There are existing methods like SCORE which quantify this, but this was the first time we showed with a large enough data set, nearly 300,000 images, that we could quantify the signal from fundus imagery. And predicting five-year MACE is not extremely accurate, but our methods were close to on par with the existing other methods that used demographic information. As a corollary in this paper, we also showed that we can predict a lot of attributes like age, gender, smoking status quite accurately. And I'll go over one by one. On the left, you can see the prediction of age, for example. It's our model's fit and the ground truth shown there. And the mean average error for age prediction is about 3.2 years. And these were confirmed with a couple of independently obtained data sets, one from UK Biobank and then from IPAX. Talking to ophthalmologists, we knew they could identify roughly young retinal fundus images from like the older people's. But there's no known result that we could predict them with this much precision. On the right side, you can see the graph for the refractive error prediction, and our MAE was about 0.560. We can also predict the self-reported binary gender with an AUC of 0.97 on held out sets. This result is based on a model trained with several ensembles using pictures from both eyes and so on. And the result has been validated on hundreds of thousands of images received from several independent sources of data. In fact, in the cardiovascular risk paper, one of the reviewers sent us a new test set on which our performance was verified by the reviewer before acceptance. We still don't know how these models are actually able to make these predictions, so we dug deeper into it. So for gender prediction, we tried some of the local explanations like integrated gradients from the literature. As explained earlier, these models provide a saliency map scoring all pixels between 0 and 1 and provide their importance. Qualitatively, it didn't give us a specific answer. You can look at the pictures here. Here are two examples, one from male and one from female. And the green spots there show the regions where the model has things that's importance to the prediction. And to be sure, in these pictures, the model is very confident and correct. We have some theories on why this is the case, but nothing conclusive yet. We also tried, for example, soft attention, which was shown in the original earlier slides. And here you can see a few examples of soft attention maps for predicting gender. And we went one step further from looking at just qualitatively. We hired a group of ophthalmologists to ask them to label every one of the pictures as to what physical anatomical landmark that they think it represents. And on the right, you can see these landmarks like veins, optic discs, etc., plotted against the frequency with which the ophthalmologist thought they were highlighted in these images. And as you can see, there isn't a sort of consensus even among ophthalmologists on what these models are trying to mark. So there is like a lot of gap between what we think the models are doing and the models are actually doing in this case. We did some obvious checks to make sure we eliminated some of the possibilities. So one of the possibilities that we thought of was maybe it's measuring some distance between landmarks and things like that. So we did It turns out that scrambling patches, for example, on the first row of images, you can see the left is the original image. On the right is a scrambled part of the image, where we took the image and broke it down into 8x8 grids. And we scrambled all the patches. And we did this during the process of training. So every single instance that the model sees is actually a new instance of these randomization. So the model has no way to measure distances between landmarks beyond a very local scale. And we varied this spatial scale over different levels. And we found that up to like 8x8 grids, it had almost no influence on the AUC of the model. So it seems to us that the model is using something local and then accumulating the information. And to be sure about the frequency domain information, we actually built an autoencoder, removed the low frequency information from the model, so that on the right you can see the last set of images. You can see the high frequency information retains most of the AUC of the model, original model that predicted gender. So we're barely scratching the surface here, but our best guess is that the model is accumulating signals from high frequency all over the image, enough to reach a high AUC of 0.97. And we think these are the exact sort of problems humans find hard to distinguish by simply observing them, because we're not trained to do these. We think we need a significant rethink of how we approach model explanation for these kinds of problems. And remember, this is just one of the many different attributes we're trying to predict. And there's so much more to learn from these. To give you a sense of how a typical model explanation technique is used in a disease diagnosis, the top part of the slide explains this. So you have an image, set of images, and then you have humans label them. And then you train a deep neural net on it. And then you have a prediction, you try to explain that and use the explanation as a form of verification. And this is the sort of thing we showed initially, where we showed the lesions and we showed the attributes, the integrated gradient saliency maps that match up with the exact lesions that are labeled by humans. And we sort of have a confidence that the model is looking at the right thing. When we look at novel signals, like in the bottom part of the slide, you don't have a human label there. So we have a prediction and we try to explain it. And we believe that explanation that come up with this from these kinds of problems, we could actually discover basic science. And we need a lot of tools to measure, to make sure that we are actually correct in these discoveries and follow up with clinical studies as we go on. So that brings us to the main motivation of the talk, which is the ML-driven discovery. On the left side, you see a typical scientific hypothesis testing. So we started with the hypothesis, such as, there's small variation between two classes of demographics for a specific attribute of the eye. And we do experiments to measure these features, and then we validate or invalidate the hypothesis. And these are typically done in low-end regime because it involves feature generation either through manual process, or in case of automated process, we have to go through correction or come up with complicated methods that also could introduce errors in feature engineering. On the other hand, we propose the approach on the right, where we passively collect the data set, acquire the images, and then we have the outcomes, which are the labels or attributes from the demographics. And we directly predict from pixels to the outcomes from a large data set. And at this point, we don't need any human labels yet. And then we explain the model after we have trained them. And then the explanation should give us a hypothesis on what's different about these two classes of images. And since there's no human curation involved in the collection of these data, these typically can be a very large data set. So here are some few examples of what we explain as human-driven hypothesis testing. These are expensive studies to start with a very narrow hypothesis to capture only a sliver of possible features in the human eye. They're also small, like we explained below, and the feature engineering could introduce more errors. Compared to this, I think our approach can scale much better. We aren't limiting ourselves to a small set of features, and one can incorporate millions and millions of images. And much work remains to be done in this hypothesis-free way of explaining models to discover, but we think we're only scratching the surface. There's a lot of low-hanging fruits we could tackle. And to be giving you examples about specific work we've done, I'm going to start with one of them. So just to give you a little bit of background about different imaging modalities. So on the left, you see optical tomography, coherence tomography slices. These are called OCT, and they are sort of volume images, where they're slices of pictures taken. And these are provide cross-sectional view of the retina. And so they're all single color, and they're usually much more expensive to acquire than the images on the right, which are fundus photographies. You can also see a small box on the fundus photography of the region that's covered by the OCT image on the eye. So the colored fundus photography is usually much cheaper to acquire, and it provides a view of the rare view of the eye. It doesn't provide you a cross-sectional view of the eye. And these are usually much cheaper. There are even handheld devices these days that you can use to collect the color fundus photography. And so one of the problems we worked on was diagnosing diabetic macular edema. It's a disease characterized by the swelling of the macula of the eye, as shown on the pictures on the right. And the gold standard for this is to measure the retinal thickness in the OCT slices. If they're swollen, you tend to, you need experts to actually diagnose these, but they are used to come up with a diagnosis based on OCT images. But OCTs are, of course, much more expensive to acquire. By some measures, in developing countries like India, it takes about 10x more price to acquire OCT images. And the doctors could only refer patients for OCT based on correlated hints they find from the fundus image, typically. So, for example, they look for what's called the hard exudate, which are yellow lesions that you find around the macular region that you can see in the colored fundus photography. And if they do have that attribute, they're usually referred, and then you collect the expensive OCT slice. So we published a paper that showed that a model trained on fundus imagery alone with the labels that were acquired from OCT labels. So these were paired images in some sense. And you can use a label from OCT images and pictures from fundus imagery, and we can train a model that can predict the diabetic macular edema much more accurately than just the rubrics based on correlated features like hard exudates. As you can see in the graph, the blue line is our model's performance. The green line is the one that is trained only on the fundus image. And then you can see the dots on retinal specialists fall on the model's performance, because that's the rubric that people usually go with on fundus imagery. But we didn't know how the model worked. So there's a big performance gap between the human graders and ML model that we needed to explain. So to begin with, we tried a few obvious saliency map-based methods and ablation methods. Here you can see two graphs of different levels of ablation that we did based on two different landmarks. And you can see the change in the AUC of the model as the different regions are exposed to the model. So each one of them, to be sure, is an independently trained model, and then we measure the performance on a held-out test set. And as you can see, most of the information that the model acquired seems to be coming from the macular region of the image, which is reassuring because that's also the place where the doctors actually do the diagnosis on the OCT slices. But we only knew where that signal was coming from. We actually didn't know what the model was looking for. So to close this gap, we proposed a new method that's based on image-to-image translation methods. And these models take an unpaired set of images, for example, pictures of zebra and pictures of horses, and show, using a trained classifier, how these classifiers change from one to the other. Right. And this model involves a whole bunch of classifiers. For example, it needs two discriminators to determine whether it's a real image or fake image. So there are two GANs on both ends. And there is also a generator function that goes from one class to the other and the reverse. And these functions operate on the two different classes of images. And as a model trains, it can show within the same context how a horse would, for example, change to a zebra without changing a pose or anything like that. So we did an important modification to this model to start with. We made all these functions, G and F, to be residual functions so that they don't have to touch parts of the picture that are irrelevant to the problem. And we train these models with two classes of images, which are essentially a no DME class and a DME class. And there are two independently trained functions, F and G, and there is a whole reconstruction loss associated with this. Informed by our previous ablation experiments, we focused our efforts only on the macular region. That is the region around the fovea, the central part of around the fovea. And the most important thing to note here is that this is a form of explaining the data, and it's not an explanation specifically for a trained classifier. Although this is the case, we'll show later that this sort of training actually encompasses all the classifiers that you possibly train on. in an ideal setting. And we also show empirically that for a given trained classifier, we can show that the classifier was able to be fooled by our generated functions on both sides. And you'll see the quantitative studies in the following slides. So the first question we wanted to ask ourselves was, does this actually work? So we first trained a model that can go between one class and the other, and then we have an independently trained classifier. So we were able to show that the model was able to successfully fool an independently trained classifier that was originally trained to distinguish these two classes. At first, it wasn't obvious to at all what these models were doing. It was like very subtle changes the models were doing. And so we applied these functions over and over again to the same image. So we computed g of x and g of g of x and g of g of x and so on, all the way till to the fifth degree. And we were able to exaggerate these changes that are made by these generated functions. So on the left, you can see the median scores of the classifier, which to be sure was independently trained from our generated functions, steadily dropped as we applied these functions. So the model was faithful to the problem at hand. And we also were able to show that it's not just a score, but the AUC of every successive application, as shown on the table on the right, was considerably dropping. And so we knew the model was doing the right thing, even when applied successively on the same image. So the default rubric that's typically used is to look at the hard exudates, which are yellow lesions around the fovea. And so the hard exudates was obvious to most people, most ophthalmologists, as they were part of the standard rubric, but we think they don't tell the full story. So you can look at the pictures here as we apply the different functions G and F on a source image. And as you go, there's sort of an exaggerated amount of changes. And we think there's a second attribute that the model uses to extract more signal out of it. So our explanation method actually clearly told us that we have to look for how dark or light the macular region was. And that's actually a strong signal as to whether the person was having a DME or not. So here, I'm going to show you a set of videos that actually show these changes. So here we are having a change from one class to another in an alpha blended way. And we're going to show you in a sawtooth fashion. So it goes from no DME to DME and back and so on, back and forth. And there's a gallery of pictures. And the first thing that should strike you was this change in the brightening and darkening of the macula as we go from one class to another. Let me show you a couple of additional videos showing these transitions. So these are like slightly higher resolution, as you can see, focused on the macula, going from one class to another and back in a sawtooth fashion. And as you can see, there is the addition of like yellow lesions, and there's also the darkening and lightening of the macula as we go towards the center of these pictures. So we think what the model is doing is actually trying to exploit how light reflects from the surface of the fovea. Although you cannot see the swelling of the fovea, you can actually exploit how the light bounces and try to extract that from basically the darkening and lightening of fovea. So the fovea is actually the foveal dip is quite deep. Then you see some more darkening of the fovea region. But if it becomes slightly lighter, it becomes paler, and then it's probably has a signal for DME. And these are subtle cues that humans find hard to pick up and humans find hard to calibrate. So one thing is that this was just a theory at this point, right? So the darkening in macula was pretty obvious to us, but how do you know it's true? So to be sure that this is quantitatively true, we also verified by taking the macular region and reducing it to a very set of simple features that we can compute, like average color around concentric circles around the fovea. And then an SVM was able to recover almost most of the AUC along with the hard exudate as an information from this. So as you can see on the table below, we recover most of the AUC based on these two features alone, whether the concentric circles average color in those concentric circles, plus whether there was a hard exudate or not. So this needs more clinical validation, of course. But we think that's pretty cool that the model was able to teach us what to look for in this data set. And we hope to improve our understanding of the fundus imagery from these models. So to summarize, we started out by describing our earliest work developing a model to do diagnosis of diabetic retinopathy. And we explained our models using integrated gradients. And these are useful in addressing the concern that our models are black box, and also to gain doctors' trust. We then discussed our work on predicting novel signals like cardiovascular risk, age, gender, refractive error, etc. We have some clues on from the model explanation, but nothing is very clear on how these how these attributes are being predicted. And this remains an unsolved problem. And one such novel prediction that we were able to explain was a diagnosis of DME based on the fundus imagery, much better than the original rubric. We provided a human interpretable explanation based on the image translation models, and we believe that the exaggeration of these changes is actually critical to highlight subtle differences. And these are much more important in novel predictions because by definition, these are things that are not obvious to humans when they look at these pictures. And so exaggeration of these changes based on model explanation, I think, played a key role in trying to explain these models. So I'd like to conclude my talk by saying there's a lot of work needs to be done in explaining all the predictions and across different areas of science. And we are very early in this process. And I think we as a community need to develop some good interpretability benchmarks. And the tools we need to build to explain these models should also go beyond confirming known facts. The typical explanation tools I've seen are most about finding or presence or absence of objects like cats or dogs and natural images and so on, but there is a lot of confirmation bias that we want to overcome. And we also want these explanation methods to teach us some basic science that we already didn't know. And that'll be pretty cool. Thank you so much. At this point, I will stop and take some live questions. Thank you.
Okay, thank you. Wait, I thought there was going to be another, like a 2-minute break, but okay. So I think we can still wait for some more participants to come in because we have some extra time in the schedule for this. But actually, I failed to introduce Arun before his talk, so let me do that now. Arun is a colleague of mine at Google, and he's worked extensively on explaining medical imaging data, as you just saw in the talk. And he's done a lot of things that's gone into the work that Google Health uses in terms of both validating the model and also explaining to understand if the signal is correct. But before that, even prior to his work at Google, he spent a lot of time working on computational biology data, like applying vision for microscopy images. And yeah, not just microscopy images and medical images in general. Thanks, Arun, for your wonderful talk. Let's wait and see if we have questions that come up in the Zoom chat, and I'll also look up at other venues, and maybe we can start Q&A in 30 seconds or so. For the data, and in some sense, maybe can you comment more on how important was it in terms of getting the right labels to figuring out that, okay, to actually make something, to discover something new from the data when you're making predictions. Maybe you can comment more on getting the right labels. I think this also in the thread in the problem that you presented, where getting labels from the 2D data was quite different from the same model being trained on labels where doctors look at the 3D scans. So Arun, it'd be great to get your comments on that.
Yeah, I skipped over some of the details on the grading for this particular problem of DME. In a typical medical imaging problem, what we have found, as I said earlier, even in the DR work, it's much more important that we reduce the variance on the test set. We wrote in the paper too, where we showed that you can have quite a lot of noise in the training data, and that doesn't seem to affect the classifier as much as measuring its performance. We need the test set to be deeply labeled. So that's one of the key things. And the other thing is, in terms of labeling, it's not just about consensus. We have published some work showing that adjudication, which is actually agree with each other on what their differences in grades are, and they will like argue with themselves and come up with a perfect answer that they can convince each other of. So typically, when you have false negatives, it's easier for someone to point out, that you missed this lesion, and then you can correct the label for the image. And so that's one way to get a better grade by deeply labeling it, or adjudication. In terms of the OCT grades themselves, in this case, at least, the grades from the OCT were not very subjective. It's essentially measuring some fixed position in the OCT slice. At least I'm not a doctor, I'm just paraphrasing what I've heard from the experts. So it's measuring a fixed distance on the image based on the OCT cross-section, and then using a threshold to determine if you have TME or not. And that is typically the way it's diagnosed in OCT, which has less subjectiveness, as you can imagine, than even fundus imagery, where like there could be several level amount of lesions, and people have to come up with rubrics to determine what determines referability versus not referability for TME.
Thanks, Arun. And also one more question in terms of like, how does this, I think, yeah, actually, is that, okay, I think Scott is saying there might be a question on YouTube live stream, is that it?
Sorry, no, that's not what I meant. I actually was just trying to make sure that the live stream was also reflecting what's happening here in Zoom. But if you don't mind, I also had a general question about this research that I could interject. So yeah, I think you touched on this a little bit in your talk. This is kind of just a high level question about, as this work is obviously, it's very translational and impactful. And I'm wondering about your experience, getting these tools into the hands of physicians and, getting that buy-in and that trust that these methods are helpful and not going to do more harm than good or as much.
Yeah,
so,
absolutely.
Anyway, it's a high level question, but I think an important one.
Yeah, so one of the things in this project, at least, we were fortunate enough was to work with the doctors right from the beginning. As I said, we have a couple of hospitals in India. We right from the earlier DR work that we did. We worked with two eye hospitals in India, and they were very helpful in giving us grades, helping us understand what it means to get a wrong answer and why it's wrong, and like going through the training data over and over with us, because we are total novices in this field. And so we closely work with doctors, and also we have a whole bunch of ophthalmologists right here at Google we work with to go through some of these images and try to understand if these saliency maps help us look at the right things, what is it missing, what would they rather prefer? Because sometimes, as I showed, you can show a saliency map and try to assist the doctors, and it can actually backfire in some cases, because you might be actually confusing the doctors by showing spurious things or distracting them from the actual thing that they should be looking on. Even if you had because some of these saliency maps are not exhaustive, they look for positive signals, but they're not necessarily exhaustive, and that could actually backfire in some cases. So we still, I think, in the assist phase, at least, we are early. We don't have a strong conclusion on whether what's the right thing to do to help doctors, other than referring them using a better algorithm that on the average works better. But we still have to find better tools to assist doctors in this case. I don't have a good answer there.
Right. Yeah, I thought that was quite an interesting finding, that the algorithm could be better than the doctors in many cases, but then when you try to use it to suggest where in the image to look, it actually can have the opposite of the intended effect. It's kind of surprising.
Yeah, this is one study. I think we want to replicate this across many things and see if this actually holds up in other problems and other verticals and so on. This is one of the studies that we did. Yeah, it was surprising to us too the first time we saw it.
Cool. Actually, yeah, my question is also going to be very similar in flavor to what Scott just asked. So that's good. I think now we don't have any more questions, and following up next is actually going to be a break. So thanks, Arun, for your talk and for taking time to answer the questions. And we will see you again in the panel discussion in a little while, I guess. So for now, I think we're going to go on a break. But during the break, we'll be playing videos of a bunch of posters. So everyone who's watching, I'm not sure. So I think you might be able to. I'm not sure if you'll be able to hear audio here on the Zoom webinar, but you should definitely be able to view the posters both on the YouTube stream as well as you look at the recorded sections. You might be able to see it. Okay, so please go on YouTube or the ICML website to view the poster videos. So they should just play. I can actually share my screen and show you how that works. And you should be able to see all of the poster videos as a single joint video. Is it this one? Okay, so if you go down and it's a recorded posters, you can click and see the videos, or you should just like take a break for half an hour and come back for Katie's talk in 10:50 a.m. So this is going to be in Central Time. So sorry, this is going to be in Pacific Time. Yeah, it's like 35 minutes from now. Come back for Katie's talk. And that's about it, actually. So we will see you all in about half an hour or so. And we're going to take a break now. Thanks. Bye.
Hello, my name is Peter Liu, and I'll be telling you about work with Sam Kin and Maren Solicic at MIT on extracting interpretable physical parameters from spatial-temporal systems using unsupervised learning. For more details, please see our paper on arXiv. In this work, we aim to develop a method for analyzing datasets with spatial-temporal dynamics that are affected by uncontrolled variables. Our method is able to extract relevant hidden parameters that correspond to these uncontrolled variables and characterize the dynamics of the system. Previous works include direct sparse identification methods, which aim to explicitly determine the dynamical equations, and other unsupervised learning approaches that focus on discrete particle dynamics. We propose a method that uses a variational autoencoder architecture combined with a physics-informed encoder and decoder designed for spatial-temporal dynamics. The encoder extracts the relevant parameters while the decoder acts as a physics simulator and predicts the future propagation of the system. For more architecture details, please see our paper. We first test our method on a variety of simulated PDE systems. We also add around 10% noise to test robustness. After training, we can extract the learned parameters and compare them with the ground truth values. Shown here are the R-squared correlation coefficients from a linear fit. As you can see, our method accurately identifies and extracts the correct parameters without any prior knowledge about the governing dynamical equation. To test our method on a more realistic dataset, we use an ab initio solver to model a highly dispersive nonlinear optical fiber. After training, our method identifies 3 relevant parameters, 2 of which correspond to the group velocity and dispersion of the fiber. The third was initially unknown to us, but we were able to interpret it as a spurious phase velocity. In conclusion, we have successfully demonstrated an unsupervised learning method for extracting relevant and interpretable parameters from complex spatial-temporal datasets. All our code is available on GitHub at the link shown below. Finally, we would like to thank our funding sources for making this possible and our colleagues for useful discussions.
Hi, I'm Vikram, and today I'm going to talk about my work on attribution for fingerprint-based virtual screening or protein ligand binding models. Virtual screening machine learning models are developed to identify molecules that very tightly bind to particular proteins and are usually applied for drug discovery. Attribution on these models can be done using toy datasets, where the binding logic is known. Specifically, certain fragments are required for a particular molecule to be active. Given a pre-trained model on this toy dataset, we can then carry out attribution by using a method called sys that identifies which fragments of the molecule are necessary for the model to assign the score that it did. By identifying these fragments, we can then determine which atoms are more or less important and compute an attribution vector, which has 1 element per atom, for which atoms are more or less important for the particular attribution of this molecule. This can then be compared to the true attribution from the known binding logic. Doing this, we find that even very high-performing models tend to misattribute their findings into low attribution scores. This is important because it results in adversarial examples or molecules that are similar to those found in the real data, but are misclassified by our model due to the poor attribution. We can break down the attribution into false positives and false negatives to better understand why models are making these mistakes. False negatives can be fixed by adding fragment match decoys, or inactive molecules that have similar fragments to the actives but are actually found to be inactive, and this significantly improves attribution scores for all models. False positives end up being the result of spurious correlations in the background or the data set where the toy datasets are drawn from. These spurious correlations result in fragments that are not part of the binding logic still being very highly correlated with activity in our toy datasets. And by eliminating these spurious correlations, we can also improve the attribution scores. Thank you.
Hello, I'm Vishal Udandrav, and I will be presenting the paper Discount Self-Supervised Visual Attribute Disentanglement using Context Vectors. This was joint work with Sarthak Bhagat and Shagun Uppal. In this paper, we tackle the problem of learning disentangled representations from visual data in a self-supervised manner. Following Locatello et al.'s impossibility result, we aim to exploit the spatial and structural inductive biases prevalent in visual datasets for the task of unsupervised disentanglement. Our major contributions in this work are fourfold, namely 1. Introducing a self-supervised multiple attribute disentanglement method called Discount. 2. Highlighting the utility of stochastic transformations in learning rich disentangled representations. 3. Presenting the idea of attribute context vectors to capture the intra-attribute variability. And 4. Applying attribute clustering objectives to promote disentanglement. I will now briefly go over the method. Given a batch of images, we construct an augmented batch of images using a set of positive and negative stochastic transformations. We then encode both of these image batches using a fixed encoders, and the encodings of these original image batches are then sent to construct attribute context vectors. Based on these embeddings, we apply a set of losses both in the image space as well as the latent space to enforce high quality disentanglement. We enforce clustering among the attributes in the latent space using a center loss. We do this by providing self-supervision in the form of pseudo-labels obtained from the context vectors. We further apply an augmentation consistency loss to ensure proximity between the latent representations of the original image batch and the augmented image batch. To quantitatively evaluate our model performance, we use the informativeness score metric. We outperform the state-of-the-art unsupervised disentanglement model mix on 4 benchmark datasets. We further provide qualitative evaluations in the form of latent visualizations and attribute transfer. For more details, please see our paper and the open source implementation on GitHub. Thank you.
Hello, my name is Joshua. Today, I'm very glad to talk about work, feature extraction on synthetic black hole images. This work is accepted by Machine Learning Interpretability for Scientific Discovery Workshop at ICML. So the story begins with last year. The Event Horizon Telescope collaboration announced we just have a first image of a supermassive black hole. As an astrophysicist point of view, we would like to understand whether it's possible to directly infer spin or mass from this black hole image. With this motivation, we turn our attention to one of the greatest tools in computer vision, the convolutional neural network. In order to train our neural network, we generate a bunch of synthetic black hole images since we only have one real data at this moment. On the left-hand side is full resolution of our synthetic black hole images, and on the right-hand side is with a blurring effect on our synthetic data that you would observe with the current Event Horizon telescopes. So we use a ResNet 18 as the baseline of our neural network model, and we further modify the last layer so it outputs the 7 parameters that we are interested in, such as spin and mass. We build blind datasets to test our neural network, and we show that our neural network are able to recover the spin parameters with good accuracy. With this good prediction, we're really interested to know what kind of features the neural network learned during the process. It turns out that using feature map of basic block 1 is particularly helpful because we discover lots of new features that look significantly different from the original input, while we can still recognize the ring-like structures. And one of the ways to tell that it learned some new feature is by comparing the feature map of our trained ResNet with an untrained ResNet. And we notice that the biggest difference is that the trained ResNet learns that there's a lot of information in the low surface brightness areas, as known as these spiral patterns, isolated in the green box. So this shows that these kind of features could be correlated to the physical parameters of the black hole that we are interested in. With these feature maps, please check the link below to read more about the paper. We look forward to meeting you at the virtual workshop. Thank you.
the scientific community has been increasingly interested in harnessing the power of deep learning to solve various domain challenges. However, despite the effectiveness in building predictive models, fundamental challenges exist in extracting actionable knowledge from the deep neural networks due to their opaque nature. The traditional attribution explanation approaches for the convolutional neural network often produce a per-pixel saliency map to highlight the region in the image that contributed the most to the prediction. Such an approach may work well for natural images, for example, highlighting the head of the cat when predicting a cat image. However, this pixel-based explanation is not particularly insightful when trying to explain why certain material has higher peak stress, as the image pixel space does not correspond to any meaningful or understandable material science concepts. In this work, we introduce techniques for exploring the behavior of deep learning models by injecting domain-specific actionable concepts as tunable knobs in the analysis pipeline through generative adversarial network-based image editing methods. For example, we facilitate the direct modification of various material attributes such as the crystal size in the input image without significantly altering other important features. We then feed the synthesized image to the model and observe how the new prediction is influenced by changes in the corresponding material attribute. By incorporating the domain concepts in the data processing pipeline, we can ask questions such as what material attributes need to be altered in order to achieve a more optimal peak stress prediction, which not only enable us to better evaluate the behavior of the black box models, but also provide scientists with actionable insights that can potentially lead to new discoveries.
In this video, I'm presenting Reaction Mapper, unsupervised attention-guided atom mapping. Chemical reactions describe how precursor molecules react together and transform into products. Using SMILES, a text-based molecule representation, chemical reactions can be represented as text and tokenized atom by atom. The SMILES representation makes it possible to apply transformer models to chemical reactions. Transformers are made of stacks of self-attention layers, and every layer has multiple heads to learn functions to attend their input tokens. We trained transformers on millions of unlabeled reaction SMILES. For the training, we used a mask language modeling task, where input tokens are randomly masked and then predicted by the model given the context. Once we had a trained model, we visually inspected the attention weights. The mind-blowing discovery was that all the models had captured how atoms rearrange during a reaction. This challenging task is also called atom mapping. We used the atom rearrangement signal to build Reaction Mapper. On a test set of 49,000 chemical reactions, we found that Reaction Mapper mapped more than 99% correctly, sometimes even better than the ground truth. The atom maps are not only of high quality, but can also be generated much faster than previous tools. Atom mapping is crucial as it allows to automatically extract reaction centers, bond changes, and reaction rules, the grammar of chemical reactions. Therefore, atom mapping affects many downstream applications. We discovered that transformers trained self-supervised on chemical reactions capture their hidden grammar, and developed Reaction Mapper, which efficiently produces high-quality atom maps. For more information and the demo, visit reactionmapper.ai.
Hi there, my name is Tegan O'Brien and I'm from the University of Victoria. Today I'll be giving a short presentation on our work interpreting stellar spectra with unsupervised domain adaptation. The main motivation for our project is what is known as the synthetic gap. This is the gap between our synthetically generated spectra and the data that we observe on the telescope, which can result from instrumental effects or improperly modeled physics. To minimize the synthetic gap, we use a cycloGAN to create a shared latent space between the two domains of data. Once this shared space is created, we can easily map a synthetic spectrum to the latent space, then decode this representation into the observed domain to create the observed representation of that original synthetic spectrum. Here we show an example of how the synthetic gap is minimized by making a direct comparison between the original synthetic spectrum and its observed counterpart, as well as a comparison between the synthetic spectrum mapped to the observed domain and the same observed spectrum. The spectrum that our network produces is a much closer match to the real observed spectrum. To make a similar comparison on a more global scale, we use a t-SNE analysis to first exemplify the synthetic gap, then show how this gap is mitigated by our network, and lastly, we compare the latent representations to show that the latent space is indeed shared. Lastly, we can use our framework to discover missing information in the synthetic spectra. To test this, we created a mock dataset where certain information was masked in the synthetic spectra, but not in the observed spectra. When mapping the synthetic spectra to the observed domain, we correctly recover the missing information. Furthermore, by tracking the derivatives of the translation back to the original labels that produce the spectra, we can identify the source of this information. Using techniques like this can effectively improve the synthetic models by providing insight into the physics that we currently are missing. In summary, we use a cyclogan to minimize the synthetic gap in stellar spectra. By doing this, we created a shared latent representation between the synthetic and observed data. Lastly, we can utilize the derivatives of the network to interpret where the network learns new information from. Thank you very much.
Hello, I'm Avanti, and I'm excited to present joint work with my colleagues Mara and Anshul on methods to identify when the feature interactions learned by a neural network are likely to be false positives. In this work, we consider neural networks trained on genomic sequence inputs. Throughout our genome, proteins known as transcription factors, or TFs, bind to DNA to ensure that the correct genes are active in the right cell types. These TFs recognize short patterns in DNA called motifs that are 6 to 20 positions wide. TFs typically bind in a highly cooperative manner, meaning that different motifs can interact to have a nonlinear effect on TF binding. Several recent works propose ways to infer these types of motif interactions using neural network models trained to predict TF binding strength from DNA sequence input. However, the susceptibility of these methods to learning false positive interactions has not been extensively studied. In this work, we devised a simulated TF binding dataset to investigate the tendency of neural networks to learn false positive interactions. Strikingly, we find that models consistently learn large interaction effects even when the ground truth simulation contains no interactions, and these interactions are significantly larger than those present in previously proposed null distributions generated using shuffled sequences. However, we show that even when a model learns large magnitude interactions, the interactions are not always beneficial to the loss. Specifically, if we create a new set of predictions on held-out data by subtracting away the predicted interaction effect from the original model output, we find that the new predictions often do not have a significantly worse loss. We also find that in the absence of a ground truth interaction, model performance tends to be inversely correlated with the average magnitude of the interaction effect. Thus, we propose that checking whether an interaction effect significantly improves model predictions across different model architectures is a promising approach for distinguishing true interactions from artifactual ones, both in genomics and in other domains.
Many-body physical systems often change its internal state when subject to external driving forces. In some cases, such change is long-lived, and we say that the system has formed a memory of its drive, or the system has learned the drive that is used to push it. Examples of such systems include polymers, viscous fluids, and even crumpled sheets of paper. Traditionally, physicists use various thermodynamic measures to diagnose such learning behavior. However, due to the complexity of many-body systems, simple thermodynamic measures are often found to be inadequate. In this work, we study the learning behavior in a prototypical many-body system, a driven spin glass. We apply different drives to the spin glass and ask, based solely on the response of the spin glass, whether one can classify the drives. We make progress by using the variational autoencoder, a popular architecture in representation learning, to identify spin glass learning behavior. Meanwhile, we also perform statistical analyses on thermodynamic variables to try to identify learning. We show that not only the variational autoencoder outperforms traditional thermodynamic measures, but it also uses an interpretable latent space that has physical meanings. If you are interested to know more, please take a look at our paper to find out what the variational autoencoder found. Thanks.
Hi, I'm Aish, and today I'll be talking about some of our work on modeling brain microarchitecture with deep representation learning. Modeling brain structure and organization is fundamental to many tasks in neuroscience, such as studying brains across different stages of age, development, and health. Despite its importance, however, modeling brain architecture is far from being solved. Neuroanatomy varies quite a bit across areas and subjects, and modeling it is a multi-scale problem that involves analyzing microscale substructures as well as entire brain regions at the macroscale. Consequently, this process requires a lot of domain expertise and human involvement, and given the ever-increasing sizes of new neuroimaging datasets, and for us to be able to identify new architectural motifs in the brain, there is a real need for automated, robust ways of discovering anatomically linked substructures and patterns both within and across brain areas. Our work, Deep Brain Discovery, addresses this very gap by leveraging the power of deep learning to build a rich model of neural structure and organization across diversified brain regions. We force a CNN to learn biologically meaningful characteristics of the data by training it to solve a brain area classification task, but using only local views of the brain structure. Once the network is trained, we collect representations across many samples at its last hidden layer, which provide us with useful cues about the relationships between different brain areas, as well as their inherent characteristics. But these representations are still rather high-dimensional and entangled. We therefore project them onto a lower dimensional, more interpretable space via non-negative matrix factorization. We then use this new low-dimensional embeddings to find microstructural motifs and anatomically linked ROIs in the overall brain sample. At the macro level, we find that the NMF factors distinctly align with different areas or specific neuroanatomical features. We even find that a method successfully picks out regions of sparse axonal innervation in the cortex, which isn't easy to do by eye. At the microscale in the cortex, we find that the NMF embeddings loosely correspond to cortical laminar divisions. On fitting a GMM to these embeddings, we find that the mixture models cluster regions by cell density. Moreover, it successfully highlights barrels and barreloids in the somatosensory cortex. You can find our paper, code, and data, as well as other material related to the project at nerdslab.github.io/deepbraindisco. Thank you.
Okay, thank you. We're already live. Okay, good. Welcome back from the break, everyone. So, oh, okay, looks like we are live. Okay, I'm actually excited to have Katie as our next speaker here. Just a moment. Okay, yeah. So Katie Bouman is a Rosenberg Scholar and Assistant Professor of Computing and Mathematical Sciences and Electrical Engineering at Caltech. Her research focuses on computational imaging, designing systems that tightly integrate algorithm and sensor design, making it possible to observe phenomena previously difficult or impossible to measure with traditional approaches. Her group at Caltech combines ideas from signal processing, computer vision, machine learning, and physics to find and exploit hidden signals for both scientific discovery and technological innovation. Interesting, and we're really excited, and it's great to have you, Katie. Maybe I think should we give another minute for you to start, or okay, because participants are still coming in from the break. Maybe we can give another 30 seconds per minute, and then you can get started. Maybe we'll wait for 30 seconds before you go. Thanks.
So start now, you're saying?
Yeah, I think you can just start. Yeah.
Okay, sounds good. Yeah, so thank you for so much for inviting me to this workshop. It's a really exciting workshop. I wish I could be there in person, but maybe on the bright side is with being virtual, we can share the work across more people who wouldn't have been able to join otherwise. So thanks again for organizing. It's been I've been impressed at the organizational ability of the group as well. So today I'm going to be talking about interpretability for the future of black hole science. So black holes are one of the most mysterious objects in the universe, and scientists have been studying them ever since they were first predicted by Einstein's theory of general relativity just over 100 years ago. And in particular, for decades, scientists 87, and it's 55 million light years away from us. And it's very special because over 100 years ago, someone discovered the streak of light on the sky, which ended up being a galactic scale jet of plasma that was shooting out of the center of the galaxy and marking the spot of a supermassive black hole. So over decades, scientists have tried to develop better and better instruments to try to study the supermassive black hole predicted to be at its center. And in April of 2017, an Earth-sized telescope connected together and collected the data necessary to make the first picture of a black hole. And 2 years later, after processing the data, this is what we saw. So this black hole image was the result of a group of hundreds of scientists from around the globe that worked together to build a computational Earth-sized telescope necessary to take this picture. And so today I want to tell you a really a bridge story of how we were able to capture the picture of the black hole. But more so is getting this image of the black hole was a huge milestone, but really it's just the beginning of this endeavor. And now that we have this new extreme laboratory of gravity, we're thinking towards the future in ways that we can improve our instrument and algorithms to learn even more. And so today, I'm mostly going to be focusing on the future of black hole imaging, extracting scientific parameters from the data, and how interpretability will play a crucial role in the success of these techniques by helping to design the next generation of the telescope. So in the first half of the talk, I'll talk more about some methods currently being developed in my group at Caltech to push the Event Horizon Telescope past its current limits to see environments around black holes that are invisible to us now. In particular, I'm going to talk about developing methods to extract information, not just about the static structure, but also about the stochastic motion of a black hole. and also developing machine learning methods to optimize the placement of the next telescopes we build into our Earth-sized telescope array. And so both of these approaches are inspired by a desire for interpretability in our solutions. However, I'll be honest that they're not really the interpretability in the same way that a lot of people in the machine learning community talk about it these days. But in the last part of my talk, I thought it'd be fun to tell you about what are the big remaining questions in black hole science? Why do we really want to take an image of a black hole, and what are we trying to get at? And discuss ideas that we have for using modern machine learning methods to help us in answering these holy grail questions. Okay, so great, but before getting started, I think it's also just important to give you a quick explanation of how we were even able to get an image of a black hole in the first place. So light propagating near a black hole doesn't follow straight lines. It's actually curved because the black hole is curving space-time. And photons can even go in complete circles around it. So the space around a black hole is lit up by this hot gas that's spinning around, heated to hundreds of billions of degrees. And so we have photons flying around everywhere, and some of the photons fall in, but other ones just graze it so that they're kind of bent around it, and the net effect is essentially that the black hole casts a shadow on a backdrop of hot material that's almost perfect, a bright material that's almost perfectly circular. And if Einstein was right about general relativity, this ring would be bent into a ring, this light would be bent into a ring whose size and shape tell us about the mass and the spin of the black hole, and referred to as the black hole shadow. And here is a more realistic simulation of what we'd expect to see if we had infinite resolution. Somewhat like infinite resolution, and we could see it around a 1 millimeter wavelength. But it turns out that even though we are able to see the imprint of the black hole, this ring kind of should exist, taking this picture seems still nearly impossible. And that's because the size of this ring is incredibly small. It's about 40 microarcseconds in size, which is about the same size as a grain of sand. But when we're viewing it from where I am in California, in Los Angeles area. And so taking a picture of something that small is really difficult. And it all comes down to diffraction. So if you plug in the necessary wavelength and required angular resolution that we need in order to see the black hole shadow into the diffraction limit equation, you can calculate that in order to see the black hole ring, we would need to build a telescope that's 13 million meters across, or essentially the size of the Earth. And if we could build this Earth-sized telescope, we could just start to make out that distinctive ring of light that's indicative of the black hole's event horizon. Okay, so although building a single dish telescope the size of the Earth wasn't possible, by joining telescopes located around the world, the International Event Horizon Telescope Collaboration built a computational telescope the size of the Earth, capable of resolving structure on the scale of a black hole's event horizon for the first time. So how does this work? How do we use disjoint telescopes to make a picture? Well, unlike with a regular camera, the Event Horizon Telescope doesn't capture a picture in pixel space, but instead in its frequency space. So we essentially take measurements of the black hole image's Fourier transform. And if we put telescopes all across the globe, we'd sample all these spatial frequency measurements. But since we only have telescopes at a few locations, that means we only get a sparse number of measurements. And it turns out that for every 2 telescopes in the telescope array, we get a single measurement of the underlying image's 2D spatial frequency related to the projected baseline between the telescopes. But in 2017, we only observed with 8 telescopes, but actually only 5 of those telescopes were at different locations and could see the black hole in M87. So that would have been 5 choose 2, which is only 10 points to make an image. So that would be a really small number of points. But luckily, as the Earth rotates, we actually obtain other new measurements. So since the baselines between telescopes change, as the Earth rotates, this amounts to carving out different elliptical paths in the spatial frequency plane. Where telescopes that are close together probe the low spatial frequency, so they give you information about the broad structure in the image. And to get the high frequency detail we need to see that ring, we have to put our telescopes as far apart as possible. So at this point, we have all this data, and we can kind of abstract away the astrophysics of the problem and look at it as purely a mathematical problem. We have this sparse data, and our challenge is to find the image that caused it. But since we only see a few samples, that means that there are an infinite number of possible images that are perfectly consistent with the data that we measure, because the areas that we haven't measured could have any value there, right? And on top of that, the fact that there's a quickly changing atmosphere above every telescope across the globe causes our data to also be really noisy. So this measurement sparsity and the extreme noise makes the problem of recovering an image incredibly challenging. So how do we actually, you might say, okay, well, how are we even supposed to go about solving for an image in this case? Well, of the images that are consistent with the measured data, there are some images that are more likely than others. And thus our goal becomes to find an image that doesn't just fit the data, but is likely under some sort of model of likely images. And so using this idea, we developed a number of techniques, Bayesian image techniques, to tackle this challenging event horizon telescope data. But these Bayesian methods required us to inject additional information into the problem about what images look like. And then that's not specific to the Bayesian methods. If we developed a fancy new machine learning method, that would be true too, based upon the kinds of training data we use. And so this kind of additional information about what we expect images look like could have significantly biased our final picture. For instance, we wouldn't accidentally want to inject a preference for ring images and then be super excited we got a ring back in the end. So we had to be incredibly careful when making images. And we therefore tackled the imaging through a multi-step process that included many synthetic data tests, strict blind imaging procedures, and objective learning of imaging parameters. So unfortunately, I don't have time to talk about them today. I'm happy to discuss more during the Q&A period if people are interested. But I just want to say that basically we just tried, trying many different kinds of assumptions, plugging in different kinds of bias, and make sure that it didn't change our scientific conclusions we made. And these tests took us over 1.5 years to complete. But once we had done many tests and analyzed our uncertainty in multiple ways, many late nights and internal reviews, we finally felt confident that this ring wasn't just our imaginations. The data was telling us the truth, and that this was actually a ring. And at that point, we released the image to the public last year. And so it's estimated that over 1 billion people have actually seen this image of this light circling the black hole. But what a lot of people don't know is that this black hole is actually evolving. So actually, we took data of the M87 black hole over 4 nights of a week, and we independently imaged each of those nights of data. And it turns out if you string the recovered images and play them as a movie, you see some variation, some evolution. So I'm going to play it for you here. It's a bit hard to see, so I'll play it again. At the beginning, you can see it looks a little brighter on the bottom left, and then it moves to being brighter more on just the bottom. I'll play it one last time. So we don't have a good idea of what exactly is causing this variation, if it's rotation or just material lighting up. And I would say we're not even sure exactly where it's appearing in the image domain. But what we are really sure of is that time variability does exist in the data. And that's because it appears in the raw measurements we take. If you look at the measurements across the 4-day span, you'll notice that the measurements are very different. So here the yellow and the purple points come from 2 different days at different points in the week. And you can see that there's a statistically significant difference between them. And getting a handle on this variability turns out to be important for a number of reasons. It doesn't only give us a window into the dynamics around a black hole, but it also helps us in mapping the space-time and constraining our predictions of general relativity and just understanding how black holes are evolving. And so as awesome as the M87 black hole has been, there's actually another black hole that the EHT is also interested in that might be able to help us better understand these questions. So that black hole is called Sagittarius A star, called Sagittarius star for short, and it's the black hole that resides at the center of our own Milky Way galaxy. And Sagittarius star is teeny compared to M87. So M87 is 6.5 billion solar masses, and Sagittarius star is a measly 4 million solar masses. And because it is so much smaller, that means the gas is spinning around it much more quickly. So whereas it takes 4 to 30 days for gas to make a complete orbit around M87, for Sagittarius star, gas makes a full orbit every 4 to 30 minutes. And so it's evolving really quickly. And we'd love to recover the dynamics of the gas flowing around Sagittarius star, because using the gas dynamics to map the space-time around the black hole would be a huge scientific gold mine. And also Sagittarius star is closer to us, so we can get independent measurements to help us confirm other constraints. But recovering these dynamics is really challenging. And that's the difficulty with Sagittarius star, is the data that we're collecting from a dynamic black hole image, which is simulated in blue here, over the course of a night is very different from how the data would look if the source was static, as shown in magenta. Remember, we're collecting data over a night as those telescopes sweep out those different baselines. And so, you know, now all the data that we've collected overnight doesn't all correspond to the same picture in the sky. And so how do we recover the evolution? Well, we could break up the night into little segments and image each region of time to, separately, and then join them together to form a video. However, if we just apply the same static imaging methods as before and try to reconstruct each snapshot, since a small number of measurements is seen at every time, the data is just far too sparse to use our standard imaging approaches, and this approach results in uninformative images. So how should we go about recovering this underlying motion? Well, we've been developing methods that propagate information through time to try to use all the measurements simultaneously, with the knowledge that although the image is changing, there is some continuity to the changes. And one approach is to model the data as part of a graphical model with 3 components, where the first component says that each image should be consistent with the data collected at that time. You know, second component says, okay, every image should look like how we expect images to look, be a sample from our image prior. And the third is that neighboring frames in time should look similar to one another. And so by maximizing the probability of this joint distribution, we're able to change images from looking something like this to like this. And this has been good because then we can, you know, start to get out even basic features about the black quickly evolving sources from this data. So here on the left shows a synthetic ground truth video, and on the right is the video reconstruction obtained from this method, assuming measurements from the EHT array. So this is a synthetic example. And okay, although this looks like a lot of improvement from the random gibberish we were seeing before, it's still not really what we want. You know, here we have recovered something like the average structure, which is great. It gives us the ring size. We see that there's a ring there, but this video has lost all the information about how gas is actually flowing around the black hole. So the dynamics are pretty much totally lost, at least here. And so although imaging is crucial in making sure we give ourselves the flexibility to see something surprising, perhaps if our ultimate goal is to understand the underlying motion field, imaging itself is not the right approach. And so, for instance, maybe we want to recover some parameters of a persistent flow field. Looking at this movie, maybe the simplest thing we might want to do is ask ourselves, is the gas spinning counterclockwise or clockwise? However, even more generically, we could try to recover an underlying flow field that describes the evolution in the source, similar to doing something like optical flow, but learning it from this kind of weird data rather than an image itself. And a non-parametric flow field like this would give us flexibility to describe arbitrary evolution in the underlying source, lots of flexibility, but then it ends up being an even more ill-posed problem because it's fitting more parameters than a movie itself. You know, you have the direction of the vector at every instant in time. And so that's probably pretty tough to do as well. And on top of that, I'm not sure how interpretable the final result would be in terms of describing the underlying black hole parameters. And so alternatively, you could also imagine wanting to describe the underlying flow using a physical model of black holes. So for instance, we could search in the space of parameters that produce different synthetic black hole videos and try to find the parameter setting that best matches the data we've observed. And this approach would be maximally interpretable since the motion parameters we are searching for have physical meaning. However, while this is theoretically maybe possible to do, in reality, this ends up being too computationally expensive. So simulating just 60,000 static image snapshots of the black hole for the M87 papers was done by teams at UIC, Radboud, Frankfurt, Harvard, and Perimeter, and it took over 75 million core hours to complete. And so it's not something, it takes too long to generate these black hole simulations, and so it's not something we want to be naively searching over using something like a Monte Carlo sampling method. And so there's a spectrum of different approaches one can take in trying to recover the underlying motion of a source. On one end is recovering a non-parametric flow field, which is maximally flexible, but requires us to recover a movie of flow and is not necessarily interpretable. And on the other end of the spectrum is estimating the parameters of a physics-based black hole model. And while this result would be maximally interpretable, the approach has the disadvantage that we are tied to a black hole model that's not necessarily correct and therefore overly restrictive. So rather than picking one of these extreme options, we would like to find a middle ground. So maybe we can use a model that balances the pros and cons of these different approaches while being computationally efficient. And towards that end, we've been working on modeling the persistent evolution of black hole sources as a result of a stochastic advection diffusion process and solving for the underlying parameters of this model that best fit the observed measurements. So this work is led by Aviad Levis, who joined my group at Caltech at the end of January. So this is very much a work in progress, but I'm excited by it. And I also want to give you kind of a highlight of things to come. Aviad is leading this work, but we're also in collaborating with other members of the Event Horizon Telescope team on it. So the immediate option tries to find a compression, basically, of the motion field by asking ourselves to match the persistent motion statistics while still being flexible in what kinds of evolution it allows us to model. So since it's not, this advection diffusion process isn't tied to any particular black hole model, it still has that flexibility. So let's dive in to understand a little bit better. So here I show an equation of a stochastic PDE, partial differential equation, describing the advection diffusion process. And in this process, there are two parameters we really care about here, V and D, where V is the velocity field and D is the diffusion field, which describes how the material is spreading out over space. So for instance, in these two cups contain water with different non-isotropic diffusion fields, resulting in the dye spreading out differently in them. So therefore you could imagine maybe describing motion of dye like this using a PDE with different underlying parameters of the diffusion. And similarly, the gas that spins around a black hole follows a similar looking process. So the black hole simulations are actually a much more complicated forward model. But by looking at these videos of gas moving around a black hole, you can imagine that they might be still be able to be approximated by this kind of advection diffusion process. And so this is what black hole researchers David Lee and Charles Gammy at UIEC are starting to do. And this could be really useful because, as you can see, different black holes that have different underlying parameters, in this case, I'm showing black holes with different values of spin, result in very different looking gas flows. So maybe solving for the parameters of the velocity and diffusion that best fits the data we measure will allow us to actually extract some properties of the black hole we couldn't do otherwise. And so by solving for this rho parameter here, we can obtain movies that obey this advection diffusion process under different parameter settings. And so on the videos on the left has this swirling motion, and the video on the right has more of this radial motion. And this feeling of motion is defined by the parameters V and D here. And by tuning these parameters appropriately, you can see that we should be able to recover flow fields that mimic the smoothly evolving black hole simulations. Meaning that although this advection diffusion process is not at all a model of black hole evolution, it can be used to still model flow fields that are perceptually similar to those seen in physical black hole simulations. Okay, great. So then our goal is to find these parameters V and D that best describe our observed source motion. But there is a catch, and that's that this is a stochastic PDE, meaning that here on the right side of the equation, I have this epsilon term that is adding randomness into our resulting movies. Just like the gas flowing around was stochastic, it kind of was random, right? So we must first draw a movie of epsilon, perhaps from a white Gaussian distribution, and then under that fixed epsilon, solve for the video rho. And if we solve for videos under different epsilons, but with the exact same B and D parameters, we see that the general feel of the motion or the motion distribution is consistent between the two videos, yet the exact pixel values are different. So since we don't know this random unknown force, we have to develop methods that are invariant to its effects. And when we're recovering parameters of the stochastic PD. So we have to be invariant to this unknown random force. And so although we are still in very early days, we've begun to get some results on very, very simple experiments that show we might be able to extract these advection diffusion parameters from an observation without knowing anything about that initial forcing term. And again, this is very preliminary, but it's something that Aviat is actively working on now and something I'm pretty excited about because it means that we might have a chance of using these noisy and sparse EHT measurements to not just recover a static image, but to recover more about the dynamics of the underlying source in a way that is still interpretable enough to lead to more science. And so as important it is to continue pushing on the algorithmic approaches to squeeze more on the data, it's also important to recognize that in doing this, we have to think about not just how do we improve our algorithms, but also how do we improve our instrument that collects the data to begin with. And so that leads to some work my group has been doing on machine learning for optimal telescope design that I'll talk about next. So in 2017, as I said, we observed with telescopes it was 8 telescopes, but they were at 6 different locations. Making images was really challenging with that small number of noisy measurements. And so although in the end we were confident about the ring shape, it took us over a year to build that confidence. And so we've been building out our array to get more data, adding telescopes in Greenland, France, Arizona. And the one I'm most excited about right now is we recently got funding to add a telescope at Caltech's Owens Valley Radio Observatory. So I'm really excited that now I'm at Caltech and Caltech is becoming part of the EHT telescope as well. But imagine what we could do if we added 10 more. So maybe we would be able to improve our black hole image to extract more science. Maybe we could actually have a chance at making that video of the gas falling into the black hole where we could actually see the gas. But before we just plop down a bunch of telescopes, we have to be really careful. And that's because building a new telescope is expensive. At the very least millions, but usually tens of millions of dollars. And so we should try to optimize to find the location that will give us the biggest bang for our buck. But how should we go about solving for the location of telescopes in the next generation array? Well, typically the design of the sensor is idealized. So the design of the telescope is idealized and considered independently from the image reconstruction problem. And in doing so, many assumptions are made. For instance, ignoring any nonlinearities in the system or complexities in the noise. And especially for problems where the data is so correlated and the noise is so challenging, it's a huge missed opportunity to not consider these effects. And so because these problems are so intimately related, we really should be trying to optimize the telescope instrument and the algorithms simultaneously. And so how should we go about solving this? Well, if you rearrange the blocks in this diagram, you'll notice that it looks really similar to an autoencoder, where the sensing system is the encoder that produces the measurements or codes, and then the imaging algorithm is the decoder. And so as you all know, deep encoders, where the encoder and decoder are neural networks, are often used for image compression, and you're going to solve for the weights of the neural network that encodes that picture in the small number of measurements at the same time as the network that decodes those measurements into an image, right? And in the image reconstruction problem, we can think of the same thing, as the sensing system is encoding the image in a small number of measurements. And so in the reconstruction problem, you could use an arbitrary mathematical function that takes you from the measurements to an image for the reconstruction. So we could use some neural network for the decoder in that way. However, unlike a generic deep encoder, the sensing systems we build must obey physical constraints. We can't just build an arbitrary mathematical, function. And so it has to be, how do we learn something that we actually can build? And so I've been working on this problem at Caltech with my postdoc, Hassan, who's been leading this effort. And the approach we propose is to develop a physics-constrained encoder that we train with the decoder end-to-end. And so let's zoom in to understand it a little bit more. So from an input image, Z here, we can define a forward model of the measurements that we expect to collect. So in this example, we pretend we have 3 possible telescopes to sample from. And these measurements we make can be complicated, nonlinear, correlated, noisy. And so here we're showing all the possible samples. And we could pass all these samples off to the image reconstruction decoder. But that means we'd actually have to build all of these telescopes to make these measurements. And our goal is to choose among them to find the best to build. So to do that, we can put a trainable block here that selects which telescopes should be turned on. And this will mask out the measurements that were not produced by those telescopes. So if we train, we're going to mask out those measurements. And so we can train this to find what combination of telescopes is best. But once we train this, you can ask yourself, like, well, why are those telescopes best? Or are we in a local minima? Or how stable is the design? If I remove one telescope, for instance, due to bad weather, does it all fall apart? And so we really don't want to just figure out what is the best combination of telescopes. What we want to understand why that is. And so because of this, we asked ourselves, well, how do we not just recover the best design, but an interpretable design? And so to do that, we replaced the telescope selection with what we call a sensor sampling distribution that characterizes the probability that we sample from a particular telescope. And so we can sample telescopes from this distribution and mask out any measurements that aren't from those telescopes. So here we just have the red and blue dots remaining. And every time we sample from this distribution, we may sample different combinations of telescopes and obtain different measurements. So we want to find the joint probability distribution over telescopes that leads to measurements that, when passed on to the decoder, actually recover an image and produce a good image with high probability. And so we can string all the pieces together into a single differentiable network with trainable blocks here highlighted in red. And our ultimate goal is to use this network to learn an optimal probability distribution of telescopes to build. And so to do that, we do that by maximizing the similarity between the input and output images, also trying to impose sensor sparsity to use as few telescopes as possible, and something that tries to encourage sampling diversity to explore many different possible designs. And so from this, we learn a sensor sampling distribution parameters during an end-to-end training. So we are co-designing the instrument with the image reconstruction method. And then we can visualize the distribution after we have learned it. So here is an example of a telescope distribution we have learned. So here on the left, on the bottom, we are showing these are the names of possible telescope sites. And on the left, we're saying this is something like similar to the marginal probability of each telescope, how important each telescope is. And on the right is showing the correlation it finds between pairs of telescopes, where red represents positive correlations, where those telescopes, the network wants them to be sampled together, and blue represents negative correlations. So from the recovered distributions, we can see some patterns that we expect to see. So for instance, in the case of uncorrelated noise, in many cases, co-located telescopes are heavily penalized from sampling together. And that makes sense because in the case that the telescopes have no noise, those telescopes are just giving you the exact same measurements. So why would you take the same measurement twice? But my favorite is not the things we would expect to see, but the things we don't know what we expect. So for instance, by changing the types of noise we train with, we can see how this affects our results. For instance, this is a result of the telescope distribution that we would like to sample from if we only have purely Gaussian thermal noise included on the measurements. But when we include difficult atmospheric noise that happens because of the fact that there's different atmospheres above every telescope, which is going to completely plague and scramble our measurements, the array preferences change fairly drastically. And so notice that when I flip back and forth, the correlations here have changed. And we find that this tells us about structures in the geometry that are preferred in the case of this challenging data correlation. So for instance, one thing that we see that we're looking more into now is it seems to prefer more telescopes with smaller baselines, whereas that isn't as essential when you don't have that noise, that kind of very complicated noise. And so complicated and correlated noise is not something that people can easily consider analytically. And so I'm really excited about how we can use machine learning and this kind of more interpretable machine learning to learn about what structures are preferred in the telescope arrays we should build in the future. And so using this approach, we're starting to understand where we might be able to, where we might want to put our next ground-based telescopes to improve our imaging capability. And I'm also excited soon we may even be applying it to a variety of other scenarios, including maybe, even considering the placement of telescopes in space. I also want to highlight, I'm going to do a little bit of an aside here, but highlight that this could be a powerful tool in a number of applications, not just black hole imaging. And the technique that I presented for jointly learning the sensor sampling distribution with a reconstruction design is actually very general. So now we are working on applying the exact same technique in other imaging applications. For instance, fast MRI is something that we've been working on, and Fourier topography microscopy. And by applying the same idea of probabilistically co-designing sensors and algorithms, we're beginning to get results that inform us about how to design sensors in other kind of challenging computational imaging scientific applications, and seeing patterns we didn't even realize before. So here was something that we got this result from Fourier topography, and now we're talking to the experts to understand why, for instance, here, it prefers to always rotate the structures of LEDs in a microscope, basically. Okay, anyway, I just wanted to sneak that in because I think it's kind of fun to see how these methods apply to many different domains. But getting back to black holes, perhaps our goal is not just image reconstruction, but let's say our goal is instead to extract some feature or classify some black hole property. So for instance, maybe we want to classify between two different models of black holes. In this case, our optimal telescopes to sample might be different from if our goal is to make an image. And we do see that this is actually the case. So here's an example when we actually trained on different kinds of, we tried to have the network classify between two different kind of models of black holes. And we see that the classification task does end up with a different telescope array than the regular imaging task, although it's not substantial at this point for this task. And one thing that is different though is for instance, the activity here, this is showing the activity of the telescope in the South Pole, does seem more important for the classification task. And so one thing that we can then think about, or way we can interpret that is, well, maybe because the South Pole provides much high, because the baselines to the South Pole are so large, it's providing high frequency information in the image. And so maybe fine details are more important for classification than they are for just imaging. Then to optimize the future telescope array, this is just one example. And really we should be asking ourselves, well, what are the big questions in black hole science that we haven't been able to answer with our current array? And if we work these black hole questions into our network loss, we could try to find a telescope array that gets us closer to answering these kind of holy grail questions. And so I think it's interesting to discuss them with a big disclaimer that I'm not an astrophysicist. So, be easy with me on questions, but I think it's just good to show you what are the big questions and how can machine learning help in answering them. So I like to separate the questions, the black hole questions as I know them, into three main categories. The first category is the one that most people are most likely aware of, and that's, do black holes obey Einstein's theory of general relativity, or do theories break down when you get to this extreme curvature of space-time? The second question is black holes launch these galactic scale powerful jets of plasma, like I showed you at the beginning of this presentation. So how do they exactly do that? And then the third question is, how do supermassive black holes grow and evolve? And so let's dive into each one and show how interpretable machine learning can help us in answering these questions. So the first question of, do black holes obey general relativity, also abbreviated GR? And to answer this question, we need to search for signatures of GR in the data and see if what we observe actually matches these predictions. But what are the signatures of general relativity, of GR? Well, the cleanest signature that we know of is related to the size and the shape of the photon orbit. So the photon orbit is the orbit which photons are orbiting around the black hole. And the space-time around a black hole, by something called the no-hair theorem, is defined fully by only its mass, its angular momentum, also called its spin, and its charge. And in effect, there are no other hairs or details about the black hole that we have to take into account when we're seeing how a black hole affects its environment. Astrophysical black holes are expected to have neutral charge, so really only the mass and spin matter in determining the shape of this sharp ring around the black hole. The mass of the black hole has the main effect on the shadow. It affects its size, but the spin also changes the shadow, but only by about plus or minus 4%. So it's a really small effect on the shadow size. And so, although the Event Horizon Telescope allowed us to show that the shadow is at least roughly consistent, the image we made has a shadow that's at least roughly consistent with general relativity, and we've shown that by comparing the shadow size to other known weights of the black hole and confirming a rate of about 6.5 billion solar masses. Using this image, we were still not able to show consistency on a level where spin would really have an effect. And one big reason for this is that although the bright ring where photons orbit a black hole should be really well defined, what's really messy is all this gas that's flowing around it. So you see here, these are different black hole simulations. I think they all have the same mass, but the gas around them is quite different. So it's obscuring the details of the ring that we care about for answering these fundamental physics questions. And so in that case, are there signatures of general relativity that are cleaner than this that we can use to probe our understanding of gravity using black holes? And so after we had published the results, there was this big discussion in the broader astrophysics community about the results of the Event Horizon Telescope. And following these discussions, actually Michael Johnson and colleagues discovered that there wasn't just one ring circling the black hole, but actually a series of subrings that get successively sharp. And from this, they realized that maybe a potential approach to get around all this messy gas that's flowing around is to go to incredibly large baselines to probe really high frequencies using space telescopes, very high spatial frequencies. And this would probe really, as I said, very high spatial frequency structure that would have the signature of the ring, but all the messy gas and astrophysics would kind of be washed out. But without knowing about this feature, we probably wouldn't have thought of this alternative approach for future telescope designs and missions. So this new signature like this, signatures like this are still being discovered. And we would like to know if there is something else out there that we're missing that could even be better at handling this fundamental question. So for instance, what if we could train a neural network that could tell the difference between black holes that were obeying general relativity and those that weren't? And maybe if we were able to train a network to do this, we could inspect the features that the network uses to make the prediction and see if this is a robust feature we could actually search for in our data, or if it's just an artifact of the limited data sets we're training with. And doing computer-aided feature discovery like this would be hugely important. Just to drive this home, here's another example from black hole science that we could use this approach for. So also in answering the question of how do black holes launch jets, probably the first step that a lot of astrophysicists would like to ask themselves or answer is which of the two black hole models are we likely observing in the black hole that we see? There's something called a sane model and a mad model, and they have different kinds of magnetic field structures that lead to different kinds of jet formation. But if you look at mad and sane simulations, it turns out they can actually look awfully alike. So in fact, here I'm showing a good fitting model from a mad simulation on the left and one from a sane simulation on the right, and both look really close to our M87 images. And so these high-level features don't seem to be able to distinguish between the two models. And in fact, it turns out that if you are even a black hole researcher, a black hole theorist, or computational theorist who works with these simulations, it's still nearly impossible to tell. So it's not just that we have the untrained eye. Even people who work with this have, it's hard to train themselves. So these images were given me by black hole graduate researcher George Wong, and he knows one is mad and one is sane, and he simulated them, but he can't remember which one, he can't tell from the images either. And to make it that much worse, the in-class variation across the different black holes is even more varied than the variation across classes, where the in-class variation is just due to dynamics over time and initial conditions and things like this. So again, maybe we can use a neural network to find the separation between the MAD and the same black holes, and then use that to identify the features that we can then probe in our data. So actually, we have found that neural networks seem to do surprisingly well at this task. But without showing what features the network is using, I am really skeptical of these kind of results, because I feel it could just be picking out artifacts in the simulations we have. And so it's really important to have to interpret what features it's using, so so that we can interact with humans and determine, are these reasonable features to use. The same is true for the last question. One way of understanding how supermassive black holes grow is by seeing what the distribution of black hole spins is over a collection of many black holes. But extracting the spin, as I talked about, is really difficult. So are there better ways of extracting spin than we know of right now? And so I think, again, this is just a huge area where machine learning, and in particular interpretable machine learning, can help make significant scientific progress. I just want to emphasize how important the interpretable part is, because without interpretation, I don't think any scientist will trust the features that a machine learning network is using. And so I just want to plug some work that is being done on this topic, actually being presented at this workshop. This work is done by collaborators of mine, although I'm not on this work. And they trained a network to predict the spin of the black hole from a simulation. Joshua is leading it. And by looking at the features, they could see that compared to an untrained network, the network trained on a collection of black hole simulations found this kind of features in the outward spiral may be strongly correlated with the spin of the black hole. And so I know this is ongoing work, but it's really interesting results and pushing us forward in trying to use these methods to help us identify what features we should look for in the future. So although I'm not as far along as this team is, I've also been working on this question with collaborators at Caltech, including Bean Kim, who's one of the organizers of this workshop. The project actually started as an undergraduate project in machine learning, a course project that continued beyond the course. They're really great students, and I'm really excited for the future of the project. And so although I'm not confident enough in our results to show any at this time, I do think it's worth discussing some of the challenges with interpretability methods for computer-aided scientific feature discovery that we've had to tackle as part of this process. The primary challenges we have faced so far with interpretability from scientific images is, one, there's very, very little data for training. So even if we could make synthetic data, as we are doing for the black hole, who says we've explored this parameter space sufficiently well to be invariant to some kind of features that we don't care about? As I said, those black hole simulations that were done for the M87 papers took over 75 million core hours to complete. And that didn't even produce that many compared to what is usually used for machine learning methods. Not even close. The second challenge is that unlike most interpretability for computer vision tasks, the features in the images are not really things. So they aren't like wheels on a car or a nose on a face that when you highlight a region, you say, oh, that makes sense, right? These are tiny features, small changes in, for instance, the distribution of the flux in the image, the compactness in an image, maybe stochastic features in the motion. And so it's very difficult of how do you even highlight these features in order to train a human to see what they are. And the third and somewhat related challenge is that these features may not even be perceptible to the human eye. They may be too small or occurring at temporal frequencies outside of human perception. So in that case, how do we draw attention to them so we can teach humans what to look for in the data? And so these questions, I hope you all are, help find interesting and help us kind of address, because I think that pushing these computer-aided feature extraction methods forward is hugely important for the future of scientific analysis. And once we're confident in these results, then we can confidently plug them into our optimization for the next generation of the array and build a telescope that we can see more of the invisible than we can now. And so to close, I think it's clear we have learned a lot from this image of the black hole in M87 already. But as I said at the start of the talk, we're really just at the beginning. And we're thinking towards the future in ways that we can improve our instrument, our algorithms, improve them jointly with the help of ideas from artificial intelligence to learn even more. And so hopefully one day we'll not just be able to show you a static image of a black hole, but perhaps a movie of a black hole as the gas is flowing towards its event horizon and all of its underlying parameters. So with that, thank you, and I'm happy to take any questions.
Thanks so much for the wonderful talk, Katie. I think we're a bit over time, but are there any questions? So I think we want to also make sure that we're set for the panel, but so we have a few minutes. Sorry,
yeah,
okay, wait. Oh, it's okay. No, we still have like a buffer time before the panel discussion. So in that respect, we're good. So I do actually have, there are like a number of points that you brought up that I do want to like bring up again during the panel discussion, which is great. But maybe one thing that's more specific to this, to the first part of your talk, in terms of like, how did you incorporate like, so I think you talked a bit about like biases that you could bring in when you were generating those images, like for example, the ring shape and things of that sort. So can you touch a bit more on like how you try to overcome biases in human priors when you build your simulation models, and also like how you validate them afterwards when you get these images? Maybe if you can touch a bit about those.
Sure. So yeah, I didn't talk very much about how we dealt with biases in the imaging during this talk. So so as I said, the data that we have is very sparse and very noisy. And so and there's an infinite possible number of images. So we had to incorporate image priors when reconstructing the images. But depending on the prior you use, you'll get slightly different images, right? So what we did, well, this is a multi-step process that we went through to try to remove bias. So we can't remove bias. There's no way of removing bias. But what we wanted to see is that no matter the way that we biased our image, our images through different kinds of methods or different kinds of priors, that it would always kind of result in that same ring structure, and it wouldn't change our scientific conclusions. And so what we did is we used many different methods with different priors, many different people, and then we went through a number of different validation procedures. For example, one is that we trained our method. So we did like these huge parameter surveys, and then we chose parameters based upon, for instance, data generated from a disk model. So we pretended the black hole in the sky looked like a disk with no hole in the center. And then we tried to say, what are the best parameters that recover this disk shape? And we'd say, okay, once we had found those parameters of the imaging methods that best recovered that disk shape, then we transferred it onto the actual M87 data. And we saw, even though we had tried our hardest to find imaging parameters that produced a good disk, when we used the actual M87 data, it put a hole in the center. So things like this, where we trained and tested on different kinds of synthetic source structures to see how it affected our results. And of course, every time we train, every time we use different parameters, it's slightly different. But we saw for the M87, the data was so beautiful that 99% that we show the public, and the ones that I showed in this talk, it's actually the combination of images from multiple methods. So you can have many images that are consistent with the data. And so we went through this whole philosophical discussion of what do we publish? Well, we published it all, but what do we show to the public? Because the public doesn't really understand, you think of a camera, you get one image. So we ended up merging them and showing that at a resolution where all these images were consistent with one another. And so, yeah, so we did lots of different testing, and then the image we showed, that blurry one, is really what is consistent across all those methods.
Thanks, Katie. And thank you very much. I think, yeah, we're going to go into the panel discussion very shortly. So maybe we'll take like a 5-minute break to help set things up for the panel, and we'll follow up with more questions there. Okay. Thank you. Okay, welcome back everyone, and we're here for the panel discussion now. I think we'll do a quick roundtable of the reintroduce the panelists a little bit, and okay, I'll let the panelists introduce themselves, like so that we can make sure that you can all hear them and see them, and then we'll open up the floor for questions to the panels. Okay, so first up, Eun-Ah.
Hi, I'm Eun-Ah Kim at Cornell University, and I'm a physicist.
And next, I think is Barbara. Yeah, you've all heard Barbara's talk earlier in the morning, but can you unmute yourself, Barbara? We can't hear you.
Yeah, I'm Barbara Engelhardt. I'm a professor at Princeton, and I'm in computer science, machine learning. I do work related to genomics data in medicine.
Thank you. Next, Sendhil.
I'm Sendhil Mullainathan. I'm a professor at the University of Chicago. I do work on sort of behavioral science and increasingly on medicine and applications of machine learning to these topics.
Thank you. Finally, Arun.
Hi, I'm Arun. I'm a software engineer at Google in Google Research. My work is at the intersection of machine learning and science. We look at a lot of scientific applications, and I'm mostly focused on trying to understand how we can use explainability to make scientific discoveries.
Finally, okay, yeah, Katie.
Yeah, I'm Katie Bouman. I'm assistant professor at Caltech in the Computing and Mathematical Sciences and Electrical Engineering department. And I work on different kinds of computational imaging problems, but mostly recently a lot of astronomy, specifically black hole imaging.
Cool. Thank you. So that's all of our panelists, and we also have a bunch of our organizers, the Scott, Michael, and Mukund, who's on the steering committee as well. He's also here on this talk. But I think I'll actually just jump right into one of the points that Katie left off in her last slide, which I think is a great point of discussion for for this workshop. But I think, yeah, more Jay, it's like what we're seeing with machine learning interpretability has been like, so initially when people built prediction models and they thought deep neural networks are great because like you can learn from like raw data, and then it'll learn features that are like, it will automatically learn whatever feature is necessary to solve the prediction task. It is really cool. And then like we needed interpretability because now we wanted to like really understand what the model was picking up on, like what are the things that it's learning, what are the features that it thinks are important for doing this task. And the initial studies that were done with interpretability were more focused on like building trust with the model. And it's only like now more recently that we're seeing a lot more work that's come up with actually moving towards not just verifying things that we know or help identify biases, but moving towards enabling discoveries of some sort. And it was wonderful to see a whole range of examples that came out from in this particular workshop so far from all of our speakers. So I think there were like points that several of you brought up during your talks. I think one in terms of like, there's definitely like signal, but it could be like weak, and we kind of need better tools to identify this weak signal and pull that out to really understand. So because like we're throwing in a lot of like rich features at our data, and then we see that, okay, it's doing quite well, but we want to pull out that weak signal and make it stronger, so that we can make some statement with a high confidence. So this came out in Sendhil's talk and Arun's talk in terms of how do we get weak signal and turn it into a stronger signal. And also in Katie's talk towards the end, where we want to like also validate these signals and overcome human bias. So I know maybe I'm going to ask Sendhil to probably start commenting on it, like how you see that makes how that plays an important role, and then if each of the panelists could also address, because I think even in Eun-Ah's talk, I think in Barbara's talk, she introduced like new ways of modeling so that we could like really understand the signal, and also being able to use visualization tools to come up with it. So I think this would, yeah, I would actually love to hear from all of the panelists on this question, how you did it in your own work, and what do you think the ML community should do better?
I'm happy to. Should I start, Subha?
Yes, please.
I'll start because I don't have an answer, so I can just rephrase your question and then punt it to other people to answer. I think if I were to rephrase the question, what I would say is something like the following, which is we have one set of techniques, holdout sets and other sorts of validation techniques for assessing how close is y hat to y, the prediction to the ground truth. We have another set of techniques, which are interpretability techniques, which actually begin with y hat. If you think about most interpretability techniques, they are about interpreting the predictor. So if you imagine y equals y hat plus some delta, or rather y hat equals y plus some delta, I think what you're saying is completely resonant to me, which is we don't want to interpret y hat, we want to interpret the part of y hat that's related to y. And I think I'd love to hear if anyone has found interesting ways of doing that piece. It's almost composing interpretability with evaluation and holdout. And I certainly haven't seen a good way to do that, but the problem is very crisp, and the way you stated it is a very crisp problem. So I think it's probably the first order problem in this area that I'd love to see some solutions to.
I can address that. I don't know if this is the right answer, but one thing that I saw your talk, Katie, and it was very, very cool. I really liked it. I have no idea anything about black holes, but it was incredible. And one thing that it actually brought up is sort of the use of these models, specifically the sort of posterior predictive distribution as a way to sort of spot check everything that's going on, right? So you build this model, you fit the model to data, and then you just run it a whole bunch of times. You generate a whole bunch of data and sort of eyeball it in some sense, right? It's a different type of interpretability, but it specifically generates the y hat star and lets you sort of compare that to y. And maybe interesting and useful in that context. So there are a lot of models, obviously, that draw, or methods, I should say, that draw on really evaluating carefully the posterior predictive distribution. And I think they're really useful.
I can jump in and give my little bit of perspective on what I see. So one of the things that I've noticed in the model explanation sort of world is that once you train a model, at the end of it, you just have these weights, and you have a function that maps from like R power n to some scalar, let's say, and then you're trying to understand what this model does, right? So there is the space in which the data manifold lies, which is, of course, in like a lower dimensional manifold that exists in this Rn space. But once you get rid of the data set itself, and then you look at just this function, so imagine a point in this Rn that you're looking at, and you're trying to understand why this image or this input gave this particular prediction using this function space. And so you'd be typically looking at some sort of like a tangent space that goes near this point on the image manifold. But unless you know the data manifold itself, just evaluating this function on Rn around this point can give you completely bizarre results, because it is just an arbitrary extension outside the manifold, whatever the function desired to fit. And so in cases where I've noticed are where the models are extremely more complex than what the data lies, or in some cases, the model are like completely underfitting what the data actually represents, like some projection of the data in some low-resolution manifold. The interpretation of these weights alone, absence of the data manifold, does lead to bizarre results. And some of these, I have a like a pet example that some of my colleagues talked about, where you have these. Let's say you have a picture, we have a list of like some spheres that are moving around here, And then there's a class of data where these spheres are moving around in like different distances and so on. And one class is drawn from a lower, like a shorter distance, and one class is drawn from a farther distance. And so you build a model on it, then you try to understand what this model represents. And this model will give you some weights on the neural network, right? So what you can get at the end of it is something that, like, what you truly expect as an explanation is that when there are two spheres, to move from one class to another, you want the spheres to move closer or further. That's sort of the physical interpretation of what it needs to do. The model needs to understand from the data. But what ends up happening is that the model might be either, if it underfits, like it could just look for, if you would need two spheres that are farther apart in an image, you generally expect at least one sphere to be closer to the center, because that's an easier way to place these spheres. So the model might be just looking for some, like a Laplacian filter on the image that is focused on the center of the image. And when you get a gradients of this sort of model, which is completely a simplistic way of looking at the thing, you would get a gradient that is like rings around the center of the image, as like a very simple example, right? And so looking at this function space and the gradient space devoid of the data manifold usually leads to very poor interpretations of the physical phenomena. Like it tells you nothing about what should you do with this sphere image that contains multiple spheres that are moving around in an image. So some of these techniques need to be careful that we shouldn't make conclusions of what this function is outside the data manifold. And that can lead to either like adversarial perturbations or other sorts of phenomena that I can explain.
Yeah, I totally agree with that. That's one of the big challenges that I'm having now in the work that I'm trying to do on the computer-aided feature discovery, right? Because especially a lot of times, the features that we want to see are not, like, it's not perceptible. Like, they're tiny little things, right? And how do you draw the eye to it? And so one thing that we've been thinking about is doing kind of exaggeration. Like, is there a way that we can exaggerate these features? But then that's kind of moving outside the manifold of possibilities, right? So then what does exaggeration mean? What is even the definition of what does it mean to exaggerate something? Because exaggeration is assuming an underlying model, right? So what is, so that's something that I would really love if there was like some method that would exaggerate difference. And it's not just, people are doing these things where they shift between males and females and GANs and stuff like that, but that's still within the space, right? What does it mean to be like a male? What is a male face? Can you exaggerate a male face? And what does that even mean to exaggerate a male face? And it's the same kind of thing. That's what I think is needed for scientific images, to train a scientist to know what to look for, and also to pick out, is that an artifact or not? I think that this exaggeration is necessary too, or something like that. Because it's not like the same as pointing to a feature, an object in an image and saying, oh yeah, humans know that wheels are correlated with bikes and cars and stuff. It's a different kind of feature.
I think Arun's talk also brought up this other thing where, so as you mentioned, this exaggeration is necessary, but to identify, let's say it's something visual and you can see it, but then if you wanted to understand why is that happening, to even generate more hypotheses, maybe we need even tools further than exaggeration to some extent, it seems like, based on Arun's talk. And maybe there's something more to elaborate there in the sense of like, so ML interpretability is looked at saying, oh, like lots of these models say spatially localized features, say, hey, here's where things are changing. And then exaggeration will tell you what is changing there. And then you also need some more physical parameters that come into the space. So I think both your talk, Eun-Ah's talk, and also Barbara's talk earlier came like, so you need the kind of like input space that you're taking from the rich free parameters that you're looking at. You want the interpretability method to also incorporate, let's say, the underlying physics or things of that sort in the parameter space to help you explain why is that happening in some sense, so that you can combine that with the hypotheses that you generate from that, just like looking at what is changing. So that's, I guess, then, okay, it's not a question per se, but I think if I were to phrase this as a question, in terms of like all of the data that you've been looking at in each of these different cases, it's I think the point that Sendhil brought out was like, it's also important to look at the right labels. So we want to look at data that's like more from the full raw data as we can, with as less human supervision as possible. And on the other end, we also want labels where we want to pick not just the kind of classes that you would normally predict for a prediction task. For in Sendhil's example, it was like, okay, don't just predict the score that a doctor will give you, but the actual score that the patient gives for that pain in some sense. So the label space is also important. And I think maybe you can all comment on that as well. I think that would be nice. So how do you look at the input feature space and also the label space in terms of interpretability? Yeah, what do you think are more important for each of the problems that you've looked at?
So can I jump in?
Sorry, Eun-Ah, I think you're muted.
Yeah,
I can't
yeah.
hear you.
I'm not muted.
I can hear. Yeah.
Yeah.
Yeah, I can hear you.
Can you hear me?
Yes.
Suba, can you hear me?
So is it just me, or is Eun-Ah muted for others?
It's just you.
That's kind of inconvenient.
Oh, okay. Sorry, just me. Okay.
Can you hear me now?
Yeah.
Yes.
Okay, good. Okay. So I just wanted to comment about bringing like two things that I learned hard way. One is I think one thing is the interpretability is really important, especially in scientific domain, when often you don't have a large volume of training set, large copies of the training set. So we often combine some model simulations with experimental data because there's only so many experimental data, and then you want to make sure that the things that that your algorithm learned to be the feature in the model are the same things it's looking at when you look at the experimental data. But then inevitably, what we simulate is always not quite the same as experimental data. Then it could be looking at it fires for some reason, but if we don't know why it's firing, we wouldn't know when it is firing for the wrong reason or the right reason. So we actually went through this exercise in the part that I didn't have a lot of time to talk about, this looking at the correlation functions, because there was earlier work when people used simulation data trained, so synthetic data trained neural network, and looked at the and then fed it the experimental data, and it said something about the experimental data. So there's hypothesis A, there's hypothesis B, and it said the experimental data looks like hypothesis A. So they said, okay, yay, we've learned. That's great, we got a verdict. Because in human eyes, the difference between two hypotheses were not easy to pick up. So we wanted to approach it from some objective measure, and that was the trained neural network. But then once we started looking at what it was learning, which is what was part of our project, there were two approaches that we've taken. First, we looked at the weights, like Arun mentioned, which he mentioned is like it can be kind of not very useful. And indeed, when we looked at the weights and went to the filters that had high weights, we couldn't make sense out of them. Like why are these filters being associated with option A versus option B? Because of all the nonlinear steps that it goes through in convolutional neural network. So we changed the feature extraction from just the usual convolution and fitting in through layers of convolution pooling, nonlinear convolution pooling nonlinear. But we built a feature that's starting from convolution and a whole set of correlation functions. And then when we sent it through the analysis of branching, lasso type analysis, we were able to see, okay, this is a correlation that it is picking up. So then we looked at how the trained neural network was responding to the simulated data, and it was doing the right thing. That was really rewarding. It was looking at right features of the hypothesis theory A and theory B. But then we looked at how it was calling the shot with the experimental data. We realized it was really picking up on something that we didn't think was all that important, but that's a kind of artifact that experiment has real life noise and simulation doesn't. So that was very disappointing. And that whole exercise taught us the potential danger in combining synthetic data with experimental data, because real life is inevitably much more complex. And so therefore there are differences, right? But then what it was picking up was something that's really trivial and not the right thing. So so that whole experience, which is pretty recent for me, taught me personally that interpretability is really important, especially when you have to combine synthetic data with real data. And we might have to strive to go beyond the off-the-shelf architectures, off-the-shelf feature extraction, because taking the off-the-shelf thing and just looking at the weight, because of the whole going away from the manifold of where the data is living in and the projection that you've done, may not be very informative. So that's my own lesson that I've learned over the course of the last couple of months.
So my name is Scott. I'm one of the co-organizers. And, as I was listening to the talks today, I was thinking about how to place these different approaches to machine learning methods for interpretable machine learning methods for scientific discovery into some sort of organizational space. And, I think the best panel discussions evoke some differences in opinion here, maybe not outright disagreement, but certainly differences in how we approach these problems. And so one of the axes along which I think that there were differences among these talks were, when thinking about approaching interpretability, it seems like there's sort of two ends of this spectrum that you could approach this from. One is by trying to compose interpretable or simple pieces together into some model for a complex phenomenon. We do that when we stitch together linear models, when we invoke sparsity assumptions, decision trees, hierarchical models, all kind of have this flavor, some sort of regularization of the model. The other end of that spectrum is to say, well, I'm gonna not make strong assumptions. I'm gonna take the most flexible model that I have at my disposal. I'm gonna fit it to solve some task, and then I'm gonna investigate that model and try to figure out how does it do so. Maybe I can interpret that complex model that's got state-of-the-art accuracy. I can interpret it in some local way. I can figure out how it behaves at this part of the input space. Do we have ideas of prototypes and criticisms and Shapley values and things like this from the recent machine learning literature on interpretable models? And so these strike me as two different ways of trying to get an interpretable result. And I think to lead to this discussion, I would say, Barbara, your talk was very much starting from that first end. I'm going to put together this model in a piece by piece way such that I maintain interpretability along the way. I think that Arun, your talk was maybe a good example of the other end of the extreme, where you're going to take this, the best-in-class neural network and then try to figure out how it makes its predictions. And certainly everybody's talks were at some point, or we're using some parts of both, or somewhere on this spectrum. And I'd like to hear your opinions on the relative merits and demerits of these two approaches. I'll point to Barbara first, since
Good. Okay, yeah, I completely agree, Scott. I think that's sort of the crux of interpretability. I mean, to maybe sort of say, let's all be friends here as opposed to start a fight, I think actually the two go hand in hand. So, when I throw sparsity on the models and add a lot of hierarchical structure, you can't do that without, sort of fitting the models, generating results, and then, looking at those results and interpreting what they actually mean. So maybe it's a little bit different than exactly what you're talking about, but I think the point about discovery in these systems is that we have to label the signal. And I think a lot of the models that we use, unless we very specifically parameterize a specific a question, a rate, you know, a shape of something, we often need to go in and sort of do the post hoc labeling, the interpretation of those things after we fit these models with structure on them. So yeah, again, I think they're sort of hand in hand. At least if you do it from the perspective that I'm talking about, which is start with the model with some structure.
So I can weigh in. Thanks, Scott, for putting it that way. I think one of our priors in this is to come up with, like, obviously, you know, the biggest and the biggest kitchen sink we can throw at it, and then find what sticks, right? So what we try to do here is that we actually try to look at the other way too when we try to find a interpretable model. So I think fundamentally, I think there's two ways to know if you have actually interpreted the model correctly. So the one way to do it is to say, I'm going to show an explanation to a human, and I'm going to explain what it is that the human should actually look at, and then the human learns the fact, able to reproduce what the model does in a different way, in their own way, right? So you've completely distilled the information from the model by teaching a human how to do the same task that the model is able to do, that they were previously not able to do. That's one way to do it. And the other way that we think is to come up with what you can define as simple features that you can then feed it to something that is much more interpretable, like a linear regression, logistic regression, or so on. And then you can compute these features using a very fixed, defined way that you've derived from the model explanation. So the model explanation, let's say, says that you have to measure these sort of features along this image and then use some sort of reduced form of representation, and then you can learn that. For example, in the DME case, we were able to reduce it to a simple vector of few numbers, and we were able to show that that is enough for us to recover the signal. So I think at the end, we still need some of these simpler techniques with either feature engineering or explaining it to humans to be completely sure that what we're doing is the right thing. Because at the end of the day, if your model explanation depends on showing saliency maps on top of the image, there is a gap in, I would say, the amount of explanation you've done, right? So if you're still at the end of it showing these are the regions I think are most important, it's not very convincing for everybody. And so in that sense, going to a simpler like a SVM or something would actually convince a lot more people and be more sure that you're doing the right thing, because you can interpret the results more. But to arrive there, we start with something much more complex. I think at least that's how we did it, because we frankly didn't know how to engineer these features. And even if we tried hard enough in some of these problems, like I said, we're still not able to engineer the features that we need to reach the kind of accuracy the model reaches. And perhaps it's because we're not like being creative enough in trying to come up with better methods, or in fact, like I hope not, but the nature is extremely against us in finding extremely small amount of weak signal in like hundreds of features that it slowly accumulates around the image, so that moving to the other end of the spectrum is actually very hard in our problem. So I don't know which part of this phase we are in, but that is the sort of challenge we face here.
Yeah, I thought it was a great question, and to pose it, where do you lie on the spectrum? And I think at least what I presented, I think I kind of laid squarely in the middle. I used both the model fitting kind of stuff for interpretability, and also hoping to do some more of this machine learning kind of stuff that I'm doing now. And I really
think you actually had a slide with the spectrum on it.
Yeah,
so.
That's right, exactly. And so I do think that the approach you take also really depends on the goal and how much you already know and are confident of, and because they have pros and cons, right? The purely modeling based approach, as you now said, that you're going to have model mismatch always from your simulations to the final data, and how much is that biasing your result? And if you purely are relying on models and data and fitting those models, you're not going to be able to tell very easily. But then going to the other extreme is like you're completely throwing out everything you know about the world and about physics and biology or whatever you're working on, and not making use of what you already know. And that you have the other extreme, and where you really need tons of data, and then you go into these problems with biases and everything. So I think it really depends on the problem. And so I'm kind of the opinion that use what you know and learn what you don't, and you know, even learning maybe model mismatch and stuff like that in the process. And I do think that using, like, when you are on the extreme of learning everything, I think that learning the basics sometimes can be good for sanity checking, right? Like, even if you want to plug in the basics, you know that this property of the data is there and we should eventually put it in. Remove that assumption, see what it learns, and see that it's able to do this basic thing first, I think is also important.
One thing I've agreed with everything people have said. I might make two comments, which is, I think we're treating the various we're not sufficiently differentiating the various kinds of scientific activities we could be undertaking. I think it's noteworthy that it varies dramatically by application or discipline how firmly you should trust the knowledge that you come in with. There are obviously some disciplines where there's just like some, as you said, they're just basics, like if conservation of energy is violated, or they're just things. But as you move away from things like physics, most disciplines have the aspect that realistically, if we look at it, even things they believe strongly today, you want that to be up for debate. You want that to be overturnable with some pretty strong possibility. This is definitely true, I think, in medicine. It's true definitely true in social science. And so I think that distinction is worth making, because it feels like where you lie on the spectrum very much depends on that core question about how much are you trying to overturn what we already believe. And in some places, that's just a foolish thing to ask. The second thing is, I wonder if there's a different way to break up this dimension that you've laid out, which is, what are you baking in your priors into? I think I thought the discussion that you made, Eun, about simulated data was intriguing, because I think, and that connects to what you said, Arun, which is one way we could bake in our priors dramatically without affecting the supervised learning part is to use the priors to model the data distribution. Now that's just an unsupervised activity. And so simulations can generate tons of data and they can give you something about the data distribution. It's also a more forgiving activity, because when you go to interpret, to your point, Arun, you know, if we can move from RN to something like the data distribution, plus or minus a huge amount of noise and error and whatever, that's a massive gain relative to just doing interpretation in RN. So even if we can, so it seems like even weak models or models we're not that sure of, we could use to great effect for simulating the data distribution and then being able to move gradients on the data manifold or the approximate data manifold. And so it seems like there are these two things where, yeah, anyway, that's all.
Yeah, I actually agree. Some of these, talking about the data manifold, at least I feel like the techniques that we use, for example, GANs, right? They're trying to like approximate a very, very complex manifold. And what happens is when you have class of data, like you described, with a very low AUC, so you have these manifold that you want to represent. Of course, GANs are not perfect. So they have errors in like how much of the manifold they cover. Sometimes there's mode collapse and you only talk about like if some part, subset of the data that you focus on and so on, right? And so when you have that in conjunction with a very, very small differences in the manifold between the data, a discriminative model, of course, can easily distinguish these two because it's very well suited for that. But GANs, especially, the way I've actually experienced it, it's on a very weak signal, it's very hard to learn a image translation model or a cycle GAN or something, which is going to give you very good interpretable translation between the two sets of data. And I think that's one of the things we have to like try to figure out. How do you take some of these GAN works and move it to a point where you can use it on very weak signals? We haven't found a good way to do that.
So would anybody else like to chime in on this topic before pivoting?
Oh, actually, there's one participant question that's very relevant to this. In terms of, they're asking like how much of Eun-Ah's observation is due to the fact that neural networks fail in out of distribution generalization versus how good the interpretability method is. So I think it's kind of like relevant to the discussion that was just happening. If Eun-Ah can take that, and then we can move to another topic.
Yeah, sure. So well, that's hard to tell. But, yes, indeed, neural networks are not very generalizable. But then once we were able to interpret, we could tell that it was a feature that's in the data. Just we have all our layers of knowledge to not look at certain things. Like we kind of filter things out, but neural network didn't have that knowledge, right? So it was less of a generalization issue, but more that it was it was a difference between the two data set, the synthetic data and the real data, that we wishfully hoped that wouldn't matter, but turned out that was the most easiest thing to grab on. It was just the ready right there to grab on, so he grabbed it. So what are you going to do? As a parent, if kids go after something that's not safe for them, and if I left them lying around, that's my fault, right? So that's kind of how we took it. Just it was so invisible to us until we tried to do this. And another lesson that we learned, which was kind of interesting in light of what Sendhil mentioned, is that we looked at two data sets that were generated by synthetic data sets that generated following two conceptually pretty different ideas. So the advocates of each of those ideas would say that my idea is infinitely different from, idea A's, parent would say idea, and idea B's parent, each would say we have nothing to do with each other, right? And then you look at the data, and in actually many respects, they are indistinguishable. But we wouldn't have known, and they wouldn't have known. And what we learned was that, okay, they are distinguishable, but if you go to fourth order correlation functions, so now we know if you want to be able to tell these apart, you want to go to fourth order correlation functions, which we didn't know going in, because each of these advocates who invented the ideas said my idea is just different from his idea, period, right? So this is kind of the idea of the notion of focusing on what's in the data is not so prevalent in physics, because physicists are used to being able to derive everything, starting from Newton's equation. You send human to moon, right? So we're used to deriving things. Although the equation that sent human to moon actually was a result of a lot of empirical observation. To get to that theory took a lot of looking at the data, but we've forgotten that part of history. We start our physics lesson in whatever school where you start from F equals to ma, that's kind of God-given, and we derive everything. So what I feel is very helpful is sort of in line with what Sendhil said. I want to be ready to overthrow things if the data says so. But that has to be also balanced with the way how I come up with my statement has to be understandable to my community. So I have to be able to fold it back to the framework and the language that's the standard language. And then I can convince them, look, actually, this has to be overthrown. But there's this like constant balancing act and sort of optimization given the task, where do I want to set my needle in balancing over the spectrum, which is challenging, but it's very interesting and it's really fun. Yeah. I don't know whether I answered the question, but that's all I have to say.
If I could jump on to what you're saying, that example you gave is super interesting, that there are these two theories, people thought they were quite distinct, but then like when you look at it, and I think for me, it opens up a whole category of things that we haven't talked about, which is what are other uses in science that these tools allow us to do that change the nature of hypothesis testing completely. So I'll give one example from work we've been trying in game theory and decision theory. One thing that we rarely look at is how much of the explainable signal do the existing models explain. And so this is another way to take the two ends of the spectrum, Scott, which is you could say, for the same phenomena, I take the existing models and I form the best fit structural model I can. then I do a black box predictor, and then I take the ratio and I say, how close are our theories right now to the signal that's in the model? That's not even an interpretability thing. It's just a way to test hypotheses differently. It's not on your accuracy, but it's on your completeness. And so I'd love to hear if people had found other ways of using these models that are just, that even change the nature of the scientific enterprise, that ask us questions like, are two theories really the same? Are they really different? I find that such a fascinating use of ML in science.
So we're not doing that exactly, but one thing that I feel is a little related is we're seeing some data and we're trying to say, okay, ignore the theory. We don't make any assumptions about that theory. Is this data consistent with this theory, and up to what level is it consistent? And let's keep pushing that closer and closer, and in which direction should we push at to get the boundary closer? So it's not what you were saying exactly, but I think it's kind of related, you know. Yeah.
I think that idea of using your so-called black box model, non-parametric model, as a way to sort of upper bound performance of your interpretable model is one that's quite interesting. I think one other place where I've seen that is Cosmo Shalisi has a really nice book on elementary data analysis. What's he called? Advanced data analysis from an elementary point of view. That's what it is. Where he's got a chapter kind of espousing a similar approach. And I think I don't see that done all that often, but I think it's a really useful technique. Another theme that I saw running through some of the talks today had to do with experimental design. So Katie, you talked explicitly about this with the problem of figuring out where to build your next microscope, telescope. I'm so used to looking at brains that, like, I think everything you want to look at is really small.
Scope in there too.
Yeah. your next scope, whatever type. And so obviously that's a huge investment, and you want to make sure you get it right, and you want to make sure that your return on investment is as large as possible. I think, Barbara, you may have mentioned this in your talk. If not, I know this is also a thing that your lab is very interested in. And I'm sure many of us here are thinking about how to use our work in progress models, given the data that we have so far, to guide our next experiment, to generate our next hypothesis in such a way that it'll be maximally informative about the way that the system under study actually works. And so I think we all kind of agree that this is a thing that we should aim for. This, again, though, it seems like a thing that we're not necessarily all doing uniformly, or not doing with the same amount of success. So maybe this would be a good point of conversation. What are the best ways to do this, and how successful have we actually been in relative to just a random experimental design, for example?
Can I start? Because this is something, like you said, Scott. Okay. I mean, a few thoughts on this. I think if I'm being, completely honest about what I think the role of machine learning is in scientific discovery, it's, epsilon improvement in 100 different places. It's not one like major home run. And one of those huge places, I think, one of those epsilon improvements, I think, is experimental design for sure. That's why my group's been working on it a lot. But but the epsilon part of it is, what I can tell you when we do experimental design is that we won't do worse than random design. And that's pretty much, the guarantee that I can give you. And it's not a strong guarantee, honestly. But the reality is, in practice, the experimental designs that we come up with end up doing much, much better than random. But really hard to show that, without a really, really deep data set to sort of work off of. So I firmly believe, I agree with you in that, this is one place where I think we can really work together with scientists. But also, this is one place I feel like without strong proof of the ability to improve, experimental pipelines and things like that, it's sometimes really hard to convince experimentalists, in my world, experimental biologists, to work with you. So right now, as an example, I'm working in a positive way. I'm working with Jared Tocher, who's building these optobodies, which are essentially modifications to individual cells, where if you shine a light on a particular organoid or organism or tissue, particular transcription factors light up, and then a whole bunch of stuff happens. So basically, optical control of tissues, and designing these optobodies is quite hard. But actually, in reality, they sort of just shoot darts at the problem. But there's about 8 experimental design parameters, and we can do it very straightforward using simple models. And so it's cool when you have someone who's like, look, I have to do this. I can't avoid doing it. Can you just, pull something together to help us out? And it fits, what machine learning can do really, really well. And it's very useful for them. So good example of when it works, but other situations don't fit so nicely.
Any other thoughts on experimental design here?
I'll mention some stuff that I think that I would like to explore too moving forward. So I talked about like, oh, how do you optimize the placement of telescopes to get the best bang for our buck and everything. But this was kind of like a one-shot deal. And in reality, things don't happen one shot, right? We're not going to get $500 million to put down all the telescopes we want. It's going to roll out over time. And so I really think it's a sequential decision, right? Where's the first one you want? Where's the second one, so that we can make progress as we go along? And I think that this is the case in many kinds of problems, like conditioned on even medical imaging, like conditioned on the observations I've made up to this point. What's the next one, best one? So I reduced my dosage to a patient or something like this. Right, so that, I think sequential decision making is really important. I also think it's related to how people explore the parameter space. When they look at features and they say, so it's a little bit different, like sequential for optimization versus sequential for this is how people learn and explore a space. Maybe this tiny feature tells me if I see this, then the policy is the best thing for me to do is make this decision next, and stuff like this. And so I do think that we should draw on like things like reinforcement learning and sequential design and stuff like that for experiment design as well.
I have a different sort of opinion touching upon the experimental design. I'm not an experimentalist, so I actually don't do any of this. But one of the things we really worry about when trying to use data from experimental biology, we use a lot of images from microscopy, pictures of cells by fixing and staining and taking pictures of them using cell painting. One of the things that we worry about a lot is like having a good experimental design that does not lead to any of the confounders. And anyone in experimental biology understands the effects of confounders, but as a ML researchers, it's really hard to wrap your head around. So I can give you an extreme example where in one of the things we worked on for quite a while, we sort of assumed we had a really good grasp on what the classes were and how we were able to classify it. Our models were doing very well. And then we realized that the way that the data is generated is you have these vials, you expand them, and then you fix and stain them, and you let them mature, take pictures, and so on, right? So there's a lot of steps and many weeks of experimental design that goes on in this. And if you're not careful in any one of these steps, even the subtle variations in which the way people handle plates, for example, right? Like the way you keep a few plates above one another in the incubator and so on can lead to different evaporation coefficients for these plates. And even that introduces errors that ML models can pick up. So one of the things we need to be very, very careful in designing this is to be absolutely sure that there is nothing else extraneous in the data that the model picks up just because of the way in which you did the experiments as opposed to what's actually in the data. And since you can't interpret these machine learning techniques in the beginning, at least when you're looking at pictures of cells, for example, you don't even know what you want to look for, what the model looks for. I mean, humans have an intuition on what organelles to look at and so on to understand what the differences are. But these models do pick up on any simple feature, like mentioned before, any simple feature they can latch onto, they latch on. And so it's very important, at least from the ML perspective, to clearly do an experimental design. So whenever we work on any of these projects, we go through several months of back and forth, just doing controls over controls over controls, just to make sure that we're not doing the wrong science there.
I just want to say I totally agree with that perspective as well. And I think it's something we have to keep in mind when doing this. I think I mean, it's just similar to machine learning, right? Like machine learning doesn't work unless you have bias. And it can be a really powerful thing. You can do amazing things with machine learning, but when you have too much bias or you're not careful with it, then it can lead you in bad directions too. So yeah, totally.
That's a great segue into a question that was asked on the ICML chat just now for you, Katie. So when thinking about experimental design, how sensitive are your proposed experiments to prior bias? And this is from Suvayu. It says, the approach probably relies heavily on simulations, presumably of your current model. And then if you rely on those simulations to suggest your next experiment, then does this potentially lead us astray? What are the caveats? I paraphrased the question a little bit, but that's the...
Yeah, sure. So, so for the actual imaging stuff that we did, that we presented that we released the image last year and stuff, we don't, in the imaging, there is no assumption of black hole models in there. We were very careful to not have any kind of assumption because we didn't want to get into a circular argument where we we're assuming something that we want to verify, so you don't stay away from anything black hole in the imaging process. But there are things that we assume, like light travels, and I mean things about the telescopes that we assume about physics of that, not about general relativity. Yeah, so for these models, I think it kind of depends on where this is, where the choice comes down to. So the results that I showed you were when we were training the models that I showed with the distributions of what telescopes are good and all that kind of stuff, these were trained with images from fashion MNIST, because we want to say, I want to be don't want to have anything about black holes when we're deciding where to put telescopes down. We want to be able to come up with a large picture of a shoe in the sky, I guess, or something like that. But when you're really getting down to the questions of is this black hole this model or this model? What is this? You might want to have a series of different solutions, and you have to optimize between them of what do you really want. You need to give yourself the flexibility to see something surprising while still the power to extract that feature. And so it's something where I think the interpretability is so important, because you don't want to just spit out an answer of this is the best telescope array. You want to explain to the scientists why it's good. This one is good. What's good about this one, so that people can come to a compromise. Because I think that that's going to be necessary, that you have to involve humans in the loop.
I totally agree with Katie. So one part of my talk was that one of the models that we designed, as I said, allows you to have sort of dense components, non-sparse components, and sparse components. And again, the reason is that there's a lot of evidence, especially in genomics, that the structure is both dense and sparse. And some examples of dense structure, which, try as we might, we're never going to get out of the system, include, who does the pipetting, the lab technician that actually does it. Also, library prep, batch. And these are by far the biggest contributors to variation in gene expression data. So, there's nothing we can do to sort of fake that out of the data, but we can explicitly model it and control for it. As long as, like you're saying, when we build these experiments, we build them in such a way to try and minimize the effects of that bias. It's going to be there. We're going to need to model them and control for them. But I think about it in terms of sort of a minimize the effects problem. So, all of the males in your experiment shouldn't all be the smokers, for example, or else you have sort of a complete confounder and there's nothing you can do. So that's how I think about it.
In terms of experimental design, sorry. I think in terms of design is a big word, but there are things that that we're going to use ML to look at the data can make a difference. So there's my colleagues doing image type measurements didn't always make the images of the same size. They didn't make it same resolution or same size, because they might want to do something quick and they would have lower resolution. But when you want to put it through a neural network, you want your input vector to be of the same size, because that's part of your design. Or when it takes not that much effort to take 3 more temperature data, before we started using ML on these data sets, my X-ray experimentalist colleagues would take maybe 3 points, and 3 points do not give me enough of a structure. If they just take 10 more points, that gives me enough structure to learn from. So at least this is not quite a design, but think having that forethought that we're going to use ML to look at this data can actually affect how people do measurements. That's one thing. Another thing is when we learn certain aspects of the data makes a meaningful feature, sometimes my experimentalist colleagues can just actually go after that feature themselves. So that can actually change what people measure. That is not so much changing the instrument, but what you measure. That's a way in which we can have an input. And then there's a whole different layer of how you optimize experiment. That's more complicated.
Yeah, I want to pick up on, actually, you made the point that I was going to make better, but I want to tie back to this distinction between, I think when Katie, you were talking around reinforcement learning, it's really worth, I think, distinguishing between the times when we're trying to kind of change the learned model, and that's why we're trying to collect data, versus the situations where we're expanding the vector of inputs, like adding a new telescope. Because expanding the vector of inputs is just such a different, even if we do reinforcement, whatever we do, it's like online learning, whatever we do, expanding the vector of inputs is such a different problem, even conceptually, because the old model actually can't really work with the new data, or rather it can, but it can work with a subset of the new data. And then a lot of the issues like exploit, explore have a very different flavor here. And I think they take on a much harder problem here. So for example, exploit would mean try to collect, the placement of the telescope, try to pick the one where your current model tells you it might get a lot of signal were it to get another measurement, another input from the X's. And explore would mean try to pick one where it says there's no signal, but that's a weird thing because it's going to say there's no signal pretty much everywhere. So even rethinking what exploit explore looks like when you're expanding the vector of inputs, I think requires some amount of thinking and some way to even formalize that problem. And it seems to me like the second thing you'd kind of need is we often have models of X to Y, but if you're going to solve this expansion problem, you actually need a model of X to X. Like you need a way to understand how the things you've measured project to other things you haven't measured. So these things of geometry are the easiest place to see there are X to X maps. So that's at least structurable. But when you start doing other contexts, the X's could look so different from the X's you have, it's not even clear how you'd structure those relations, that it seems to me that category of problems is actually quite interesting. And it's a subset of experimental design. It's not the only thing. It's almost like new measurement. And I find that problem very interesting. Sorry?
It kind of reminds me of cross-validation, right? Like
right.
over time.
Yeah. And it excites me because in a way, if you look historically at what a new theory really provides, I think the most important thing that new theories really provide is an impetus for a new thing to measure that we might not otherwise have thought to measure. Like it's easy to imagine what theories do in science is to explain the data we already have, but often the best theories, they do that next step, which is really what makes them very interesting. We didn't really think to measure heat as a thing, but now we knew to measure heat. And so I think there's something exciting about thinking about science as the expansion of the measured.
I think that, Subhashini, did you say Mukund has a question?
Yeah. Mukund, you want to go up next?
Yeah. So I'm Mukund, I'm from Google. So I have this question. It seems that model-based discovery cannot fully be automated. And clearly the human at the other end has to do some work. This was implicit in what Eun was talking about. This was explicit in what Katie said about knowledge of physics and biology required to do discovery. This was also implicit in the fact that interpretability techniques often lead you off the manifold that you care about. So the question is, could someone speak to the distribution of labor between the human and the model and the interpretability tool. So the human on one side and the model and the interpretability tool on the other side.
Was that the, I think you may have frozen for a second there, but does anybody want to answer that?
I can give my small perspective on that. I think it's at least in the physical sciences, that at least the problems that I've looked at, it's extremely hard to have it fully automated discovery of such a thing exists, right? Like unless you have a problem where you're discovering a law of physics. Let's say, for example, there's some infallible thing that you can write down as a symbolic expression that you can write down that describes the data in a way that you had no idea. I think it's at least in physical sciences problems, there's always a room for trying to simplify using human intuition as opposed to just writing the model spitting out the answer by itself. So at least I've found that there is a small human element at the end of it. Even if you've shown the answer right in front of you, there needs to be a human there, at least for now, to make sense out of the data. And that barrier has not been crossed yet.
This also ties to another question that Sendhil got during his talk that we didn't come to, which was, I think, what does it take for, let's say, the model that you have to predict pain, for that to be clinically usable, in the sense of what would it take for you to convince doctors to actually use this model-based measure as a measure of pain as opposed to their current rating score? So this is also what do you need at the end from the human side?
And it's related to something you mentioned around, in the end, I need to be able to convince my colleagues. And there's just this pure sociological element to the whole thing, which is, I think, it's not just a pairwise, it's ultimately, we have science, and each discipline has their own sort of port for taking in new information. And you can't plug anything into that port. You can only plug things in that match the port. And I think one of the challenges is, even if you're sure personally you've gotten a piece of new information, how do you even get it into the network of scientists and convince them this is worth thinking about?
You know, this question kind of reminded me of this quote that I read it's in Karl Popper's his Logic of Scientific Discovery, right? He has this line that stuck out to me that I pulled it up here: bold ideas and unjustified anticipations and speculative thought are our only means for interpreting nature. And I think what he's saying is, look, try as we might, there is no automated solution for this little element of human creativity. He was writing this in 1935. I feel like nowadays we're a little bit more bold in our anticipations of what machine learning algorithms will be able to do and their creative abilities. But I think there's something to this, right? That these methods can give us new ways to look at the existing data that we have. They can guide us toward new types of experiments we might run to collect new data. But ultimately, the onus is on us as scientists to have that critical insight that says, aha, here's the unifying principle that binds all of these things together. I mean, maybe, like I said, maybe it's hubris that I feel like me as a scientist still has a critical role to play in scientific discovery, but at least I feel like we're in good company here. What do you guys think?
Yeah, I wish we had a way of other than sparsity, I don't have good ways of encoding simplicity into models, or the preference for simplicity, like the Occam's razor. I think that's what humans are good at. They're like, what is the thing in common, the most basic feature in common? And right now, these machine learning methods are these complicated things, and actually the more complicated, the better, because then the more local minima and all this kind of stuff. And it's like the opposite of what that humans use. And sometimes even putting sparsity just makes it worse, right? So how do we encode some sort of the basic unit that connects all these things? I wish that there were methods that were able to do that.
I have an interesting anecdote that I want to share. Once I was giving a talk, machine learning talk, and one of my senior colleagues said, the Ptolemaic view of universe, that Earth is at the center, actually fit the data that was available at the time much better than, even our solar system, sun at the center of the solar system, right? A Copernican idea was more appealing to human as we look at it now. But if you look at which one did better in fitting data, the Ptolemaic idea, that a theory fit the data better. Nowadays, we have a lot more data, and we know that old idea was wrong, but it held on for a very long time. And if we only relied on how the best we capture the data, then one could make the same error. So that's just intrinsically there, I think. So perhaps even when we feel like machine can do a lot of things automated, we want to keep our eye on it. That's my view. I don't want to become obsolete, but yeah.
Going back to the sparseness point, I think there's something very revealing in the sparseness thing, which is I often think of the sparseness thing as in part an optical illusion. And let me explain what I mean, which is it's sparseness in some representation of the input data, where that representation is chosen for us as humans to think, oh, that's just one variable. Like if you even just take computer vision as an example, like if I said it's sparse, oh, I'm counting pencils, presence of pencils, and a feature is number of pencils. But that's an enormously complicated torquing of the input vector of images. And so you tend to see this all the time, is that like there's already a pre-built representation either in our heads or in the data set, so that when we impose sparseness, what we're really saying is it's sparseness relative to my representation of the input space. And so I've never seen a better answer to your question, Katie, but I do wonder if that might help us get to a slightly better answer. That is there a different representation of input space on which sparseness is also obeyed, but just not the same representation we have? Now, of course, this is hard to formalize for obvious reasons, but it does seem like that's part of why when we use the word simplicity, we're almost kind of misusing it. It's like it's simple relative to what we already believe, and that's what we mean.
I guess another way to think about this too, I like this idea of wanting the most simple explanation, obviously, in terms of the interpretation. I think, sometimes the models that we use, it's already been said in this discussion, are overly complex. And, maybe we don't need nonlinearity. Maybe we don't need, as many features or, versions of those features projected onto some other space as we actually use. And, simplicity could be actually just in the experimental design of our analysis as well, which is, start with logistic regression and go from there, which often is the right answer, even if it's not, necessarily publishable. But I guess the other thing that was brought up by a couple of people is that, the best predictive model is not necessarily the interpretable model. And when I think about a particular analytic problem, I always separate the two out extremely far from each other, just because it's in my experience, thinking about evaluating the interpretability, the sort of value of a model based on its predictive power is almost never the right thing to do. I'm thinking about this in particular, using genotype, let's say, to predict your disease risk, where right now, essentially using every single SNP in the genome is quite predictive. But, the reality is that there's probably, for most traits, probably many fewer than every single mutation that's leading to that disease.
Actually, yeah, I think both Scott and me have a couple of questions. We have like another 10 minutes or so. So I think I'll ask mine and we'll have Scott ask his next. So I think mine's going to be pretty short in terms of like the problems that you're working on right now. what is the ML tool that you would love to have already that will if it's there, you would just love to plug it into your problem and say oh, it gives you whatever you want. So if there's, I don't know, it's yeah, basically is there an ML tool that the community can develop that you would love to use in your next problem? And now maybe I'm going to ask, you should ask somebody specifically. So, Katie, because you said you wanted to see subtle differences, I think there was the phrasing of the problem was you said there's going to be lots of intra-class variation, and within class variation is much higher than between class variation, or maybe it's the other way around. And I think maybe this question applies there.
Yeah, so the tool that I really want, and I think I said this at the beginning, is this exaggeration tool. But this exaggeration tool requires the thing that we were just talking about, that you have some underlying model. Like humans are good at understanding exaggeration because of this model, just like the pencils thing. We saw the pencils, we say, okay, how do we exaggerate the number of pencils? Add a pencil, right? But that was under some model. And it's very hard to discover a model and exaggerate it. And so that's what I want, something that exaggerates differences in an interpretable way, but that there's so much baggage that comes along with that I think needs to be solved. So
Eun-Ah?
So one thing that we struggled is Michael mentioned when he was introducing me, the problems in my field often are hard to even simulate. I've talked about simulating, but I wasn't being so, that's kind of overstatement. We simulate some simple rendition of what we think is going on, and just to simulate is difficult. So what we generate, the synthetic models that we generate to try to train, are often overly simplistic compared to real experimental data. And we tried again to try to see if we can, go from our, handmade synthetic data to real experimental data. And that was also very difficult. So being able to bridge between what we can, by training, I'm a theoretical physicist, and often theoretical physicists like to discuss with a piece of napkin, because if you have an idea that's good enough, you should be able to scribble on a piece of napkin, while drinking coffee, right? But piece of napkin can hold only so much, right? So what you get out of piece of napkin is some really key essential feature, however you compress that knowledge, right? So from that key essential feature piece of napkin model, we make something up that's my synthetic data, and we can throw some, simple noise, Gaussian noise, white noise, whatnot. But it doesn't really come to the reality. But we also, although we know the laws of the motion of electrons, because they are complicated quantum mechanical objects, we cannot just simulate them either. So yeah, it would be really nice if there is a way to bridge between my piece of napkin, model, generated synthetic data, and real life data, which has all kinds of noise in it.
Okay, we have 5 minutes left here. I think that means it's time for bold ideas and unjustified anticipations. So we've had, over the past 5-ish years or so now, many workshops, not me personally, but we as a community have had many workshops on interpretable machine learning and such. And so in 1 minute or so each, I'd like to ask you guys to prognosticate about where we as a field are headed. Where will we be next year when we meet again, hopefully in person, for a conference on machine learning and interpretability. And I'll go around the clock here. So Sendhil, you're next on my screen.
Oh, I mean, I'm a very good prognosticator, so this should be very easy. I mean, predicting the future is the easiest thing, I think. I think the thing that I'd probably do is to make a macro observation, which is, I think that over the next 5 years, what I hope we'll see is the spread of these tools to, I think right now we've seen them primarily used in the very hard sciences, where the theories are extremely good, they work very well, and it's kind of, and I think, Barbara, to your point, it's like we might get epsilon gains on lots of margins. But there's a whole suite of other sciences where the existing theories are not very good and we don't have much understanding. And I think that what I would hope happens in the next 5 years is people start moving into those areas. And I think there's a chance for some potential for pretty massive changes there, simply because we're starting off at a relatively low base. And I think that sometimes includes even areas where the theories are in some fundamental sense, like Eun-Ah was saying, correct, like quantum mechanics is what drives material sciences, but the fact that the theory is in some sort of an unsimulatable, inaccessible sense true doesn't really help material. I mean, material science still has this huge bridge to gap, a huge gap to bridge. So I think there's a whole set of theory fields, I think, that are ripe for this area, and I hope what we see is a real expansion and a flow into those areas.
Barbara, 1 minute of posterior predictions.
Let's see. Maybe not a prediction so much as, you know, one thing I really liked about hearing your talks and this discussion is that, you know, we start with very simple questions about, maybe not simple, but questions about the scientific space we're working in, and ask questions about interpretability, you know, sort of to answer the questions that we're asking. I feel like a lot of the sort of generic work on interpretability is not really built on a specific question, but just sort of asking, you know, broad questions about interpretability. And I also think, to the point that I sort of made before, but I'll just sort of repeat it because I think it's important. You know, these models that are extremely good at doing prediction are not necessarily the ones that we care about interpreting. So let's not try and put lipstick on a pig. Let's actually, you know, use simpler models, maybe models that are slightly less predictive, but that actually give us a real answer to the specific question that we're posing.
Katie?
Yeah, so I love these ideas and these comments that people have been making so far. I guess the one thing that I hope for and that I think I see in moving in this positive direction is, like, when I was doing my PhD, there was almost no which was, you know, not too long ago, there was like almost no connection with machine learning and the sciences that I saw kind of at least at the mainstream computer vision and machine learning conferences. Or I feel like it was kind of off in the corner and it wasn't as big of a deal. And I think I'm seeing much more appreciation for connecting machine learning to the sciences now, which I really love. It's something I really think that we can aid scientific discovery through this. And in people understanding the connection of AI and science, I think that things that people don't talk about now will become more important. Like, people are so used to having these massive data sets of millions of images with no noise in them. Like, error bars are really important for science, and it's ignored in a lot of the work in machine learning. So I think, like, things like this are becoming increasingly important, and people are appreciating them. And then along with that, I think interpretability is another thing. We keep saying we have to convince the scientists. They're just not going to blindly believe these machine learning black boxes. And so I think as people are connecting more science and machine learning, and researchers from both sides are coming towards the middle. I think I'm really hopeful for the future and these approaches that will come out of them. I guess that's what I have to say.
Okay, Eun-Ah.
So 5 year didn't seem to be a long time in my field. Condensed matter physics has long history, and some problems have been kicking around for 30 years until I started doing machine learning, which was only like 3-4 years ago. Okay, and I never knew I will be doing this. So I don't know what I will be doing next 3 years, not to mention 5 years. But what I am really hoping for, and I think it's quite possible, is this interpretable ML efforts leading to new understanding, new knowledge, giving us new insight, and going beyond until now, like last year, 2019, 2018, there are papers saying, yay, I've done machine learning. Yay, I've done, this. People do this on computer vision. I've done that on my data. It was all validating, benchmarking, but gradually we're starting to actually make discoveries, actually, gain new insight. And I think that's really exciting. And because I am a scientist, I have to convince myself before even convincing my colleagues. And when I can convince myself that, okay, we've learned something new, that I can make sense out of after fact, because we're always smarter in, after fact. That's very, very satisfying. So I'm optimistic and really excited about the progress.
All right, and then Arun.
So for me, it's more of a wish that I want to see. There's a lot of complexity in the kind of models we build, and I think there's a lack of good language vocabulary for us to talk about what kind of explanation we're coming up with, what is the complexity of the model we're talking about. Is this something that we can actually interpret with a very simple thing, or a very complex thing with some very complex nonlinearities between the features and so on? So I would like the community to come up with a better taxonomy of these things. Like, this is a simpler problem, this is a much more complicated, and it's like, diverse in this way and so on. Better language, better taxonomy, and better maybe benchmarks to understand if the tools actually do the thing for each sliver of thing. But because I don't think there's going to be a silver bullet that solves everything. So if there are all these three things that are better in the next few years, that'll be great.
All right, I am excited hearing all of these ideas and hopes for the future. I just want to say thanks to all of you again for some really amazing talks and for taking the time to share your ideas and opinions and thoughts with us on this panel. And then I'll hand it back to you, Subhashini, to wrap us up.
Yeah, thanks everyone for joining in. I thank all the I think Scott already thanked all the panelists. I think up next is our poster session, which you can watch. It's going to be a recorded video, so you'll have to watch it on the screen. Sorry, by the screen I mean like you have to go out of Zoom and watch it on the ICML page. And then you can come back here and we'll have a short closing remarks and how we go about the parallel poster session. But thanks to all the panelists, and thanks everyone for listening in, and across YouTube and also here in the Zoom session. Cool.
And thank you so much for organizing everything.
Yeah, thank you for organizing.
Yeah, it's great.
Thank you so much.
Hello, my name is Peter Liu, and I'll be telling you about work with Sam Kin and Marin Solicic at MIT on extracting interpretable physical parameters from spatial-temporal systems using unsupervised learning. For more details, please see our paper on arXiv. In this work, we aim to develop a method for analyzing datasets with spatial-temporal dynamics that are affected by uncontrolled variables. Our method is able to extract relevant hidden parameters that correspond to these uncontrolled variables and characterize the dynamics of the system. Previous works include direct sparse identification methods, which aim to explicitly determine the dynamical equations, and other unsupervised learning approaches that focus on discrete particle dynamics. We propose a method that uses a variational autoencoder architecture combined with a physics-informed encoder and decoder designed for spatial-temporal dynamics. The encoder extracts the relevant parameters while the decoder acts as a physics simulator and predicts the future propagation of the system. For more architecture details, please see our paper. We first test our method on a variety of simulated PDE systems. We also add around 10% noise to test robustness. After training, we can extract the learned parameters and compare them with the ground truth values. Shown here are the R-squared correlation coefficients from a linear fit. As you can see, our method accurately identifies and extracts the correct parameters without any prior knowledge about the governing dynamical equation. To test our method on a more realistic dataset, we use an ab initio solver to model a highly dispersive nonlinear optical fiber. After training, our method identifies three relevant parameters: 2 of which correspond to the group velocity and dispersion of the fiber. The third was initially unknown to us, but we were able to interpret it as a spurious phase velocity. In conclusion, we have successfully demonstrated an unsupervised learning method for extracting relevant and interpretable parameters from complex spatial-temporal datasets. All our code is available on GitHub at the link shown below. Finally, we would like to thank our funding sources for making this possible and our colleagues for useful discussions.
Hi, I'm Vikram, and today I'm going to talk about my work on attribution for fingerprint-based virtual screening or protein-ligand binding models. Virtual screening machine learning models are developed to identify molecules that very tightly bind to particular proteins and are usually applied for drug discovery. Attribution on these models can be done using toy datasets, where datasets where the binding logic is known, specifically certain fragments are required for a particular molecule to be active. Given a pre-trained model on this toy dataset, we can then carry out attribution by using a method called sys that identifies which fragments of the molecule are necessary for the model to assign the score that it did. By identifying these fragments, we can then determine which atoms are more or less important and compute an attribution vector, which has one element per atom, for which atoms are more or less important for the particular attribution of this molecule. This can then be compared to the true attribution from the known binding logic. Doing this, we find that even very high-performing models tend to misattribute their findings into low attribution scores. This is important because it results in adversarial examples or molecules that are similar to those found in the real data, but are misclassified by our model due to the poor attribution. We can break down the attribution into false positives and false negatives to better understand why models are making these mistakes. False negatives can be fixed by adding fragment match decoys, or inactive molecules that have similar fragments to the actives but are actually found to be inactive, and this significantly improves attribution scores for our whole models. False positives end up being the result of spurious correlations in the background or the data set where the toy datasets are drawn from. These spurious correlations result in fragments that are not part of the binding logic still being very highly correlated with activity in our toy datasets. And by eliminating these spurious correlations, we can also improve the attribution scores. Thank you.
Hello, I'm Vishal Udandrav, and I will be presenting the paper Discount Self-Supervised Visual Attribute Disentanglement using Context Vectors. This was joint work with Sarthak Bhagat and Shagun Uppal. In this paper, we tackle the problem of learning disentangled representations from visual data in a self-supervised manner. Following Locatello et al.'s impossibility result, we aim to exploit the spatial and structural inductive biases prevalent in visual datasets for the task of unsupervised disentanglement. Our major contributions in this work are fourfold, namely 1. Introducing a self-supervised multiple attribute disentanglement method called Discount. 2. Highlighting the utility of stochastic transformations in learning rich disentangled representations. 3. Presenting the idea of attribute context vectors to capture the intra-attribute variability. And 4. Applying attribute clustering objectives to promote disentanglement. I will now briefly go over the method. Given a batch of images, we construct an augmented batch of images using a set of positive and negative stochastic transformations. We then encode both of these image batches using a fixed encoders, and the encodings of these original image batches are then sent to construct attribute context vectors. Based on these embeddings, we apply a set of losses both in the image space as well as the latent space to enforce high quality disentanglement. We enforce clustering among the attributes in the latent space using a center loss. We do this by providing self-supervision in the form of pseudo-labels obtained from the context vectors. We further apply an augmentation consistency loss to ensure proximity between the latent representations of the original image batch and the augmented image batch. To quantitatively evaluate our model performance, we use the informativeness score metric. We outperform the state-of-the-art unsupervised disentanglement model mix on 4 benchmark datasets. We further provide qualitative evaluations in the form of latent visualizations and attribute transfer. For more details, please see our paper and the open source implementation on GitHub. Thank you.
Hello, my name is Joshua. Today, I'm very glad to talk about work, feature extraction on synthetic black hole images. This work is accepted by Machine Learning Interpretability for Scientific Discovery Workshop at ICML. So the story begins with last year. The Event Horizon Telescope collaboration announced we just have a first image of a supermassive black hole. As an astrophysicist point of view, we would like to understand whether it's possible to directly infer spin or mass from this black hole image. With this motivation, we turn our attention to one of the greatest tools in computer vision, the convolutional neural network. In order to train our neural network, we generate a bunch of synthetic black hole images since we only have one real data at this moment. On the left-hand side is full resolution of our synthetic black hole images. And on the right-hand side is with a blurring effect on our synthetic data that you would observe with the current Event Horizon telescopes. So we use a ResNet 18 as the baseline of our neural network model. And we further modify the last layer so it outputs the 7 parameters that we're interested in, such as spin and mass. We build blind datasets to test our neural network, and we show that our neural network are able to recover the spin parameters with good accuracy. With this good prediction, we're really interested to know what kind of features the neural network learned during the process. It turns out that using feature map of basic block 1 is particularly helpful because we discover lots of new features that look significantly different from the original input, while we can still recognize the ring-like structures. And one of the ways to tell that it learned some new feature is by comparing the feature map of our trained ResNet with an untrained ResNet. And we noticed that the biggest difference is that the trained ResNet learns that there's a lot of information in the low surface brightness areas, as known as least spiral patterns, isolated in the green box. So let's show that these kind of features could be correlated to the physical parameters of the black hole that we are interested in. With these feature maps, please check the link below to read more about our paper. We look forward to meeting you at the virtual workshop. Thank you.
The scientific community has been increasingly interested in harnessing the power of deep learning to solve various domain challenges. However, despite the effectiveness in building predictive models, fundamental challenges exist in extracting actionable knowledge from the deep neural networks due to their opaque nature. The traditional attribution explanation approaches for the convolutional neural network often produce a per-pixel saliency map to highlight the region in the image that contributed the most to the prediction. Such an approach may work well for natural images, for example, highlighting the head of the cat when predicting a cat image. However, this pixel-based explanation is not particularly insightful when trying to explain why certain material has higher peak stress, as the image pixel space does not correspond to any meaningful or understandable material science concepts. In this work, we introduce techniques for exploring the behavior of deep learning models by injecting domain-specific actionable concepts as tunable knobs in the analysis pipeline through generative adversarial network-based image editing methods. For example, we facilitate the direct modification of various material attributes such as the crystal size in the input image without significantly altering other important features. We then feed the synthesized image to the model and observe how the new prediction is influenced by changes in the corresponding material attribute. By incorporating the domain concepts in the data processing pipeline, we can ask questions such as what material attributes need to be altered in order to achieve a more optimal peak stress prediction, which not only enable us to better evaluate the behavior of the black box models, but also provide scientists with actionable insights that can potentially lead to new discoveries.
In this video, I'm presenting Reaction Mapper, unsupervised attention-guided atom mapping. Chemical reactions describe how precursor molecules react together and transform into products. Using SMILES, a text-based molecule representation, chemical reactions can be represented as text and tokenized atom by atom. The SMILES representation makes it possible to apply transformer models to chemical reactions. Transformers are made of stacks of self-attention layers, and every layer has multiple heads to learn functions to attend their input tokens. We trained transformers on millions of unlabeled reaction SMILES. For the training, we used a mask language modeling task, where input tokens are randomly masked and then predicted by the model given the context. Once we had a trained model, we visually inspected the attention weights. The mind-blowing discovery was that all the models had captured how atoms rearrange during a reaction. This challenging task is also called atom mapping. We used the atom rearrangement signal to build Reaction Mapper. On a test set of 49,000 chemical reactions, we found that Reaction Mapper mapped more than 99% correctly, sometimes even better than the ground truth. The atom maps are not only of high quality, but can also be generated much faster than previous tools. Atom mapping is crucial as it allows to automatically extract reaction centers, bond changes, and reaction rules, the grammar of chemical reactions. Therefore, atom mapping affects many downstream applications. We discovered that transformers trained self-supervised on chemical reactions capture their hidden grammar, and developed Reaction Mapper, which efficiently produces high-quality atom maps. For more information and the demo, visit reactionmapper.ai.
Hi there, my name is Tegan O'Brien and I'm from the University of Victoria. Today I'll be giving a short presentation on our work interpreting stellar spectra with unsupervised domain adaptation. The main motivation for our project is what is known as the synthetic gap. This is the gap between our synthetically generated spectra and the data that we observe on the telescope, which can result from instrumental effects or improperly modeled physics. To minimize the synthetic gap, we use a cyclogan to create a shared latent space between the two domains of data. Once this shared space is created, we can easily map a synthetic spectrum to the latent space, then decode this representation into the observed domain to create the observed representation of that original synthetic spectrum. Here we show an example of how the synthetic gap is minimized by making a direct comparison between the original synthetic spectrum and its observed counterpart, as well as a comparison between the synthetic spectrum mapped to the observed domain and the same observed spectrum. The spectrum that our network produces is a much closer match to the real observed spectrum. To make a similar comparison on a more global scale, we use a t-SNE analysis to first exemplify the synthetic gap, then show how this gap is mitigated by our network, and lastly, we compare the latent representations to show that the latent space is indeed shared. Lastly, we can use our framework to discover missing information in the synthetic spectra. To test this, we created a mock dataset where certain information was masked in the synthetic spectra, but not in the observed spectra. When mapping the synthetic spectra to the observed domain, we correctly recover the missing information. Furthermore, by tracking the derivatives of the translation back to the original labels that produce the spectra, we can identify the source of this information. Using techniques like this can effectively improve the synthetic models by providing insight into the physics that we currently are missing. In summary, we use a cyclogan to minimize the synthetic gap in stellar spectra. By doing this, we created a shared latent representation between the synthetic and observed data. Lastly, we can utilize the derivatives of the network to interpret where the network learns new information from. Thank you very much.
Hello, I'm Avanti, and I'm excited to present joint work with my colleagues Mara and Anshul on methods to identify when the feature interactions learned by a neural network are likely to be false positives. In this work, we consider neural networks trained on genomic sequence inputs. Throughout our genome, proteins known as transcription factors, or TFs, bind to DNA to ensure that the correct genes are active in the right cell types. These TFs recognize short patterns in DNA called motifs that are 6 to 20 positions wide. TFs typically bind in a highly cooperative manner, meaning that different motifs can interact to have a nonlinear effect on TF binding. Several recent works propose ways to infer these types of motif interactions using neural network models trained to predict TF binding strength from DNA sequence input. However, the susceptibility of these methods to learning false positive interactions has not been extensively studied. In this work, we devise a simulated TF binding dataset to investigate the tendency of neural networks to learn false positive interactions. Strikingly, we find that models consistently learn large interaction effects even when the ground truth simulation contains no interactions, and these interactions are significantly larger than those present in previously proposed null distributions generated using shuffled sequences. However, we show that even when a model learns large magnitude interactions, the interactions are not always beneficial to the loss. Specifically, if we create a new set of predictions on held-out data by subtracting away the predicted interaction effect from the original model output, we find that the new predictions often do not have a significantly worse loss. We also find that in the absence of a ground truth interaction, model performance tends to be inversely correlated with the average magnitude of the interaction effect. Thus, we propose that checking whether an interaction effect significantly improves model predictions across different model architectures is a promising approach for distinguishing true interactions from artifactual ones, both in genomics and in other domains.
Many-body physical systems often change its internal state when subject to external driving forces. In some cases, such change is long-lived, and we say that the system has formed a memory of its drive, or the system has learned the drive that is used to push it. Examples of such systems include polymers, viscous fluids, and even crumpled sheets of paper. Traditionally, physicists use various thermodynamic measures to diagnose such learning behavior. However, due to the complexity of many-body systems, simple thermodynamic measures are often found to be inadequate. In this work, we study the learning behavior in a prototypical many-body system, a driven spin glass. We apply different drives to the spin glass and ask, based solely on the response of the spin glass, whether one can classify the drives. We make progress by using the variational autoencoder, a popular architecture in representation learning, to identify spin glass learning behavior. Meanwhile, we also perform statistical analyses on thermodynamic variables to try to identify learning. We show that not only the variational autoencoder outperforms traditional thermodynamic measures, but it also uses an interpretable latent space that has physical meanings. If you are interested to know more, please take a look at our paper to find out what the variational autoencoder found. Thanks.
Hi, I'm Aish, and today I'll be talking about some of our work on modeling brain microarchitecture with deep representation learning. Modeling brain structure and organization is fundamental to many tasks in neuroscience, such as studying brains across different stages of age, development, and health. Despite its importance, however, modeling brain architecture is far from being solved. Neuroanatomy varies quite a bit across areas and subjects, and modeling it is a multi-scale problem that involves analyzing microscale substructures as well as entire brain regions at the macroscale. Consequently, this process requires a lot of domain expertise and human involvement, and given the ever-increasing sizes of new neuroimaging datasets, and for us to be able to identify new architectural motifs in the brain, there is a real need for automated, robust ways of discovering anatomically linked substructures and patterns both within and across brain areas. Our work, Deep Brain Discovery, addresses this very gap by leveraging the power of deep learning to build a rich model of neural structure and organization across diversified brain regions. We force a CNN to learn biologically meaningful characteristics of the data by training it to solve a brain area classification task, but using only local views of the brain structure. Once the network is trained, we collect representations across many samples at its last hidden layer, which provide us with useful cues about the relationships between different brain areas, as well as their inherent characteristics. But these representations are still rather high-dimensional and entangled. We therefore project them onto a lower-dimensional, more interpretable space via non-negative matrix factorization. We then use this new low-dimensional embeddings to find microstructural motifs and anatomically linked ROIs in the overall brain sample. At the macro level, we find that the NMF factors distinctly align with different areas or specific neuroanatomical features. We even find that a method successfully picks out regions of sparse axonal innervation in the cortex, which isn't easy to do by eye. At the microscale in the cortex, we find that the NMF embeddings loosely correspond to cortical laminar divisions. On fitting a GMM to these embeddings, we find that the mixture models cluster regions by cell density. Moreover, it successfully highlights barrels and barreloids in the somatosensory cortex. You can find our paper, code, and data, as well as other material related to the project at nerdslab.github.io/deepbraindisco. Thank you.
Hi, welcome back, everyone. I think we'll go over some of the poster session logistics, and I'll just do a quick closing remark. I think we had like a pretty good day. I really enjoyed a lot of the talks, and the panel discussion was also really interesting. Brought up a lot of points that uh, we've been thinking about for a while as, as people who are working on model interpretability. So that was also very refreshing and nice. And I think for all of you in this audience right now who are watching this, I think hopefully you got back a set of things that the people in the science community really want are looking for in like solving their problems, and then like how you can build tools or think about how reframe uh, the problem that you're working on so that you can generalize and develop tools that help address some of these problems and hopefully make new discoveries in science. With that, I will go over some poster session logistics. I'm just going to share my screen on how this works. So for the poster session, we're like going to close down this Zoom webinar room, but for every individual poster, there is a separate Zoom room. And we're doing this in 2 sessions, and we're going to be reusing those Zoom rooms. So to do the poster session, so first let me share my screen. This one. Workshop detail, I think. Okay, so hopefully you're all seeing the schedule. So we just got done with this. And for the parallel poster session, what we have is, for the first session, we have 9 papers. And for each paper, this link here will take you to a Zoom room where you can join and speak to the authors. So it's between like now and when we finish the session to the end time that's related. So it's half an hour. And for authors who are in that Zoom room, at the end of your session, please vacate your room so that the next session poster authors can join in and take on questions that they have. So apart from that, you can move the discussions that are happening in the poster session to this networking town. So this is how so once you click, you go to a gather town space, and then you'd say, hopefully you're all above 13, and you just say yes. And it will take you to a place where, oh, there are already some people in here. So you can meet other attendees in this room and chat with them. And you can just go around and say hi. That's for taking your poster sessions outside and coming back to this room. So note that the poster session, so after the half an hour slot, the Zoom rooms are still available, like for however long, I think for another 4 or 5 hours or something. So if there are other people who are interested in this and you want to continue your discussions afterwards, feel free to come back to the Zoom room and use it. Yeah, I think that's about it. Are there any questions about how this will work? So all of you know how to go about the poster session. Okay, then I guess that's it. So thanks everyone for participating. And hopefully I'll just meet all of you in your poster sessions shortly. Cool, thank you. Scott, any remarks that you want to say?
Nope, just want to say thank you to you again, Subhashini, for organizing and putting all the legwork into pulling this workshop off. It's been a lot of fun, and I really appreciate it.
Yeah, cool. Thanks, everyone. Bye.